Hi everyone. Hi everyone. Am I audible? So hi everyone, good morning. Welcome to Unacademy NEET English. Today we are going to complete plant physiology in one shot. So guys, you know that Aramb event is there, right? And it is on 7th of March at 7 p.m. So everybody just stay tuned, right? Just go to this particular link and click on the Okay, so go to this particular link and click on the notify me. This event is going to be really amazing because here you are going to get amazing offer for NEET 2026 aspirant, NEET 2027 aspirant, NEET 2028 aspirants, right? And trust me, everything will be better than last year. Okay, just say we keep growing, we keep evolving. Same way, Aramb event is also evolved in that way, and many students are going to get benefit out of it. So make sure you watch it. Okay. So let's get started. Let's start the session today. And today, what will be the sequence? Today, we are going to complete plant physiology, right? So after plant physiology, what should we start? I'm thinking to start cell cycle and biomolecules. What say? What say? Because I know these marathons, they are going to be, because class 11 students, class 12 students, they are about to end their board exams and they really need the sessions for the quick and the complete revision. So that is why this is what I have planned, right? That for 11 syllabus, I'll go slow, right? So that they can, understand everything in detail. So what say? Next unit, cell cycle biomolecules. Because if we keep less number of chapters, then we are able to complete the details in that better. After that, we'll go for the structural organization, right? Morphology, anatomy, and then we can just go for the human physiology. What say? What say? Sure? Okay, so what will be the sequence today? Today, the session will not be very lengthy, right? So we'll discuss plant growth and development first. Okay, so you can expect 3 questions from this particular part. Minimum 3 MCQs you guys will expect here. Then comes the photosynthesis, and then there will be respiration. This is going to be the sequence for today's class. Okay, so PGD will not take more than 3 hours. Okay, photosynthesis 3.5 hours, because we will discuss each and everything in detail, and then comes the respiration. So this is going to be the sequence today. So minimum 3 questions, minimum I'm writing 4 questions, 3 questions. So if 11 questions are there, minimum I'm saying, so you know that 44 marks. So today's session is for 44 marks, but trust me, it will go for 50 marks as well, right? So there used to be 5 chapters in plant physiology, but now there are 3. So from all the chapters, you will get the question, right? Done? Thank you. So just keep it in your mind, one thing. And secondly, let me tell you, just in human physiology, 6 chapters are there. But sometimes they usually, this is what I have noticed, that they don't consider all the chapters. Like you won't get the question from all the 6 chapters in the final exam. This time if they will do it, I don't know, but last year also 1 or 2 chapters they were not even touched, not even a single question was there. But from these 3 chapters you will for sure get the questions, right? So do not miss it. Do not miss it at any cost. So what are we going to start? We are going to start the plant growth and development, right? So I'll start from the basics here. Okay, so I'll start with few terms, then I'll start that. Okay, though you are going to discuss that terms even in the this chapter itself, but still I would like to start with that. What are that terms? See, firstly, you all know that we all arise from a single cell that is known as zygote. Yes or no? You all are well aware that we all arise from a single cell. That single cell is what? That single cell is zygote. And from the single zygote, if you talk about the plants, yeah, animals, a complete individual will arise. So what is this property? This property is totipotency. What is this property? This property is totipotency. So what is totipotency? It is the ability of a single cell, right? It is the ability of a single cell to give rise to a complete individual. That is the meaning of totipotency. Zygote is a totipotent cell, right? Why? Because a single cell will give rise to, right? It is the ability of a single cell. It is the ability of a single cell to give rise to a complete individual. To give rise to a complete individual. So that is what totipotency is. First thing. Now, we know that in the case of plants, right, when you talk about the plants, they grow continuously throughout their life. They grow, plants grow continuously throughout their lives. Yes or no? They grow continuously throughout their lives. Yes or no? Yes or no? Why is it so? We know the reason behind. The reason behind is the meristematic tissue. Yes, please. What is the reason behind? The reason behind is the meristematic tissue. It is the reason that we are able to grow continuously. The plants are able to grow continuously throughout their lives because they have the meristematic tissue. So what exactly is the meristematic tissue? Students, it's a group of cells, right? It's a group of actively dividing cells. So whenever you are going to talk about actively dividing cells, like they are dividing, okay, their size is going to be small. In the case of plants, or overall, whenever you talk about an active cell, the active cell, any active cell will be small in size, okay? So here you will see there will be a large nucleus, right? We will also use the term conspicuous for that, right? Conspicuous for that. So large conspicuous nucleus in the surrounding, you people will see dense cytoplasm, right? You people will see dense cytoplasm. Conspicuous nucleus will be there. It's a group of small cells, right, beta? So this is what we have when you are talking about the meristematic tissue. So because you are talking about the plant cell here, you are going to see, because it is actively dividing, it is having a primary cell wall. It is having a primary cell wall. You know that that is going to be thin. That is basically cellulosic. This is what you see. Now, when a cell is continuously dividing, when it is doing nothing else, it is just dividing, dividing, and dividing, right? They are adding more and more cells. They are adding more and more cells. They are adding more and more cells. So what does it mean, right? What does it mean? That cell, obviously, you will say meristematic, but basically, these are the undifferentiated cells, right? They are what? They are the undifferentiated cells. So what is the meaning of being undifferentiated? Undifferentiated means its fate is not fixed, right? It is not fixed that what is it going to do. It is just dividing. Like we all arise from a single cell, that is zygote. From that zygote, there comes a time that some cells, they will go, they will make the neurons. Some cells, they will make the muscle cells. Some cells, they will make some other cells, right? So when they are fixed that, okay, this is my job. I have to be a neuron now. I have to be a muscle cell now. Then you will say they are differentiated. If their fate is not clear, if they are just growing, they are dividing, then you will say undifferentiated cell. Undifferentiated cell. It is immature. It is just dividing. It is not having a fixed function, fixed role. Now, if let's suppose this is a meristematic cell, yeah, meristematic tissue. Now, in this meristematic tissue, now it is turning into parenchyma. Just giving you an example so that you can understand it. So if it is turning into a parenchyma, so what does it mean? Like it is acquiring a fixed function. So this particular tissue is differentiated. So this is the differentiation when a cell, when a tissue will acquire a permanent function. So differentiation results in what? Right? It's DM. DM is the technique. So differentiation results in the maturation. Is that clear? Differentiation results in the maturation. So when a cell is undifferentiated, means it is just dividing, it is just making other cell mass, it is not It is not having a fixed function. But when I'm saying a cell has acquired a fixed function, right, a cell is clear that, okay, now I'm parenchyma, this is my job, that is my job, means it is a differentiated. This is the differentiation. And sometimes what happened that these, so from meristematic tissue, how the permanent tissue forms? By differentiation. Okay, right. So this is what I can write here. So if it is meristematic, so how that meristematic tissue is turning into a permanent tissue. This meristematic tissue is turning into a permanent tissue by differentiation. Okay, by differentiation. So sometimes what happened that these permanent tissue, they regain, right? Now they are not dividing, they're just specific for a job. So now they regain their ability to divide. So this is the de-differentiation. What's that? It is de-differentiation. Am I clear? Right? Am I clear? So permanent tissue, right? Permanent tissue, when it regain its ability to divide, it is the de-differentiation, and that results in the formation of secondary meristematic tissue. Why secondary? Because it has developed from the permanent tissue. And yeah, with time, even this can also acquire the permanent function again. Then you will use the word redifferentiation. Am I clear? Then we will use the word redifferentiation. Am I clear? Right? Then again, it will have a permanent role. So these are few basic terms, right, that I believe you all should know. Any doubts here? Any doubts here? Yes? Sure? Okay. So let's start the chapter, Plant Growth and Development. Right? That's our title. That is Plant Growth and Development. Okay? That's our title name. So first of all, right, plant is growing. We know that, right? They grow continuously because of the meristematic tissue. But what exactly is the development? That's what we have to discuss now, that what exactly is the meaning of development here. So students, when I am talking about this word development, so development actually means, right? Development, it means what? It means the sum of two processes. Two things will result in the development, and that two things are growth and differentiation. Right? Growth and differentiation means there will be the development. So if I talk about the growth, now you understand the meaning of differentiation, right? What is the growth actually? So when I'm saying growth, growth is an irreversible change. It is an irreversible, right? It is an irreversible permanent change that will occur in an organ, tissue, or organism. It is irreversible. You cannot reverse it. It is a permanent change. And we know that when it comes to the growth, growth is the fundamental and the characteristic feature of living being. It's a characteristic feature. It's not a defining property. Why? Because non-living beings, they also grow by accumulation. So it's not a defining property, but definitely a fundamental feature, a characteristic feature of a living being. It is an irreversible permanent change. And when it comes to growth, growth is the result of metabolism only, right? It is the result of the chemical reaction which are going on in our body, and especially when in our body buildup is more than breakdown. You know that when it is the metabolic reaction, metabolism, I am talking about the chemical reactions which are going on in my body. Is that clear? I'm talking about the chemical reactions which are taking place in my body. So here what do we see? That the buildup, if buildup is more than, if in that reaction there is a buildup, if buildup is more than breakdown, if buildup is more than breakdown, obviously then it will result in the growth. Okay, so growth is there, then differentiation is there. That means the plant is showing what? The plant is showing the development. So here we can very clearly see this thing, right, students, that all plant organs are made up of variety of tissues. You are well aware of it, right? Cell will result in the formation of a tissue group of cell is tissue then form the organ then form the organ system, and finally there will be an organism. So all the plant organs are made up of variety of tissues. So all cells of a plant, they are the descendants of zygote, because ultimately they are arising from what? They are arising from the zygote. So ultimately ultimately all the cells, they are made up of zygote, right? So zygote, so that's why we are saying they are descendants of the zygote itself. So they develop into different structure and function, right? And how will they get these different structure and functions? Obviously due to differentiation, right? So differentiation is a process by which they are acquiring a particular structure, a particular function. Is that clear? So we basically, we are developed from what? Right? We are developed from what? We all are developed from zygote. So that single zygote has developed some neurons, some muscle cells, etc., etc. Why? Because in that muscle cells, there are different proteins, different enzymes, right? Or ultimately, the DNA is all same. But in their case, that particular, I would say the proteins will express. In the case of muscles, that other particular proteins will express. So that is how the things are. So ultimately, we all are arising from a single cell zygote, but then the differentiation occur, different structure and function will be there. Obviously, some genes, some proteins are going to decide that. In your higher classes, you will also study about the cytoplasmic determinants, etc., etc. But now, right, stick to this part here. So this is a fundamental biological process where what is going to happen, a less specialized cell will become specialized type. So if you will ask me that what exactly is the differentiation, that's how I'm going to elaborate. That the cell which is not specialized for a particular function is basically becoming specialized. Are you getting my point? Are you getting my point? Right? So becoming specialized. Is that clear? Is that clear? So why it is happening? As I said, same DNA is there, but different genes will express. So which genes will express, that is going to decide. And yeah, here in plants, also you will see it based on location and the signals they receive. Just say for an example, in the case of a plant, the cells which are at periphery, right, they will form epidermis. They will form epidermis. So that's how the things are connected. So the location also plays an important role here, right? So plant cell differentiation is both. It depends on the internal factors as well as the external factors, the positional cues, that where that particular cell is located, right? The gene expression, hormones, chemical signals, environmental factors, they all are going to decide this. Is that clear? Now, what do we have in a plant? Yes, please. What do we have in a plant? We have roots, stems, leaves, flowers, fruits, and seeds. We are talking about the flowering plant, angiosperms, of course. This is what we have here. So you will not see that they are going to grow in a random order. It's not like that. Yes or no? Is it? Is it? Are they growing in a random order? Of course not. They are not at all growing in a random order, right? They are not growing randomly. There is a sequence. There is a proper order that they all are following while growing. So we know that in a flowering plants, there is an orderly sequence during the development. So when it comes to the plant life cycle stages, you will see the seed will form seedling, will form plantlet, will form mature plant. So there is a sequence which is present. Are you getting my point? So which sequence is there, students? Which sequence is there? So sequence may we will see the seed, seedling, plantlet, and then comes the mature plant, right? So they'll keep on growing. The height will increase, right? The girth will increase. That is the diameter. The circumference will also increase there, okay? Okay. So girth will increase, that is the diameter increase, right, students? So for height, yeah, I would say when there is the primary growth of the plant, right? When there is the primary growth of the plant, like it is increasing in its length, you will see the primary meristematic tissue is going to play a huge role here, right? Primary meristematic tissue, which forms during embryonic stages. This is the tissue that develops during embryonic stages. And when it comes to the girth, you will see the secondary meristematic tissue is basically responsible for, right? Secondary meristematic tissue is responsible for increase in girth, and we know that it develops due to dedifferentiation. So as I already told you, your primary permanent tissue, when it regains the ability to grow and divide, this is the secondary meristematic tissue. So it is the dedifferentiation that results in this, right? Right. So that's how the girth will increase. So yes, they all will grow. They appear and fall periodically, sometimes repeatedly, right? So that's how the things are. So before the flower, vegetative growth is required. Can you tell me why? Yes. Can you tell me why? Why is it so? Like why vegetative growth is before the flower growth. Why? Because see, flower is what? Flower is the site of sexual reproduction. In the flowering plant, we know that it is the site of sexual reproduction. It's a place like flower, we know that seeds, it will, the ovaries, right, inside that ovules are there that will form the seed. Fruit, flower itself will turn into the fruit. So we know how things are related. So we know that flower is a reproductive structure. Flower is a site of sexual reproduction, right? That will result in the formation of seeds, and seeds are very important because they help in the dispersal. They help that plant to grow in other areas as well. They are very important for their continuity as well. So basically, if that reproductive structure, it needs a lot of energy, it needs the food, it needs the reproduction. So vegetative part, like all that root, stem, leaves, they grow first, right? Why? Because plant has to make sure that I have the food resources. It has to make sure that roots are properly developed so that, you know, they can just get attached to the substratum, attached to the soil. They can take nutrition out of that. Leaves should properly develop. Stem should be developed so that photosynthesis can occur, so that food can be prepared. So that is why when vegetative part is there, and obviously it will give rise to, after vegetative part, flowering is there. Why? Because vegetative part will collect all the essential things which are required for that reproduction, right? Right. So here you can see vegetative phase, it always precedes. So yes, they can, even if you don't read the statements carefully in the paper, you can make mistake. So it precedes flowering, like vegetative phase occurs before flowering, right? So why is it so? Because vegetative phase is a phase of growth. It's a phase of period of growth and resource accumulation, which is literally required for the successful reproduction. Yes or no? Yes or no? Yes or no? So vegetative phase will make sure that there should be necessary support, necessary energy reserve should be there, right? So for the reproduction. So photosynthesis, energy storage, even the nutrition, water uptake, right? They all will be done by the vegetative phase. It will provide the structural support as well. It will provide a framework so that further energy and the resources can be provided for the reproduction. Is that clear? Is that clear? So this is the diagram given in your book. So you should know the meaning of hypocotyl, cotyledon, seed coat, epicotyl, hooks and everything. Right? So do read about it, do check it, which is hypogeal germination, epigeal germination, you should check about it. Okay, so now let's talk about the growth, which I told you clearly that it is a fundamental feature, it is a characteristic feature, but it is not a defining property. Okay, it's given in living word. We have discussed that too in our previous marathon. Yes, so it's not a defining property, but definitely a fundamental feature, definitely a characteristic feature. So growth is what? It is regarded as one of the most fundamental and conspicuous characteristics of a living being. Right? One of the most fundamental and conspicuous characteristic of the living being. I'm repeating again, growth is one of the most fundamental and conspicuous characteristic of living being. So it is an irreversible permanent increase in size of an organ or its part or even of an individual cell. This is what growth is. Okay, so how this will be done? It is accompanied by medical processes, both anabolic and catabolic, but at the expense of energy. And for growth, the catabolism should be lesser than anabolism. Anabolism should be more. Build-up should be more. Is that clear? Is that clear? So now here they are saying that plant growth is generally indeterminate. So yes, there are parts which show the determinate growth. There are parts which will show the indeterminate growth. So now this is the thing that we have to understand, that what exactly is the meaning of this line. So plants, we know that they retain the capacity for the unlimited growth throughout their lives. And that is because of the meristematic tissue. So plant can grow continuously throughout their life. So this ability, right, is due to the presence of meristem. Okay, so such cells, they have the capacity to divide and self-perpetuate. They will divide, they will make more cells like them, they'll increase their number. That's what we know in the case of plant. Is that clear? Is that clear? So meristem will keep on adding. Meristem will keep on adding the new cells there. Meristem will keep on adding the new cells there. Yes or no? Yes and no. And here, any cell has the potential to divide and form new tissue or organ. So it is the open form of growth. So when I'm saying plant growth is indeterminate, I am talking about the continuous plant growth. Yes, there are certain parts where you will see the determinate growth as well. You know that leaves can grow only up to a certain size, fruit can grow only up to a certain size, right? So yeah, overall plant growth, yes, we say that it is indeterminate, it is continuous, plus it is open form of growth. So when you are saying open form of growth, indeterminate means continuous, open form of growth means meristem will keep on adding new cells, and that cells are also having the potential to develop into a new tissue or organ. Is that clear? So apical meristem of root and shoot, where cells are dividing, they are producing the new tissue. It is the example of indeterminate growth, continuous growth. So why do you say that the growth is open? Because yeah, you will see determinate closed form of growth as well, but here you are talking about the indeterminate open form of growth. So when I'm saying Omkar, when I'm saying open form of growth, because new structures can arise continuously from these undifferentiated cells. So meristem is adding more cells which are undifferentiated, and they are giving rise to any type of new structure basically. That's why it is the open form of growth. So it is important. It is important. So it is continuous, and new cells can give rise to, the newly added cells can give rise to new tissue or the organs. That's why it is an open form of growth. I hope I'm clear here, right? So when I'm saying closed growth, when the future fate of the cell is already determined, like it is not, you are not saying that it is pluripotent or something, right? So basically, in our case also, these are the words that you use. These are the things that you know. So that's what I am trying to tell you here. Right, that's what I'm trying to tell you here, right? That just say in our case, we say that cells are not pluripotent, right? Pluripotent cells, these are the stem cells which can give rise to any of that germ layer, any of that cells, right? Any of that part. Okay, so it is not always like that. Sometimes it is fixed. Okay, fine, these cells can develop into this type of structure only. So growth where the future fate of cell, that this cell is going to develop into this part, it is already determined in the meristematic region. So it is the closed growth. So here you are talking about specific group of cells which will develop into specific structure. So this is the closed form of growth. So in open, new cells were added which were forming the new tissue or organs, right? But here, new cells, their fate is fixed, that okay, you are developed and you have to make this, make these structures only. Just in our case, let's suppose some cells, they are destined to develop into only into the blood cells, right? They will develop only into blood cells. It is like that. Are you getting my point? It is like that. Okay. Okay, so that's what you have to remember. So in some lower plants, we see that, right? So growth is more deterministic and restricted here, right? So just in root and the shoot apices, tips, only a definite set of initial cells, they contribute to growth. So it is closed. Why? Because meristem is closed in organization. It is having fixed initials producing fixed tissue. Like it is clear that, let's suppose you don't know, I can teach you physics also, chemistry also, maths also, right? If I'm saying so, right? So what sort of thing it is? Like open form of growth. If I'm saying, no, my fate is fixed, I have to teach biology only, I have to teach this chapter only, I have to teach this topic only, it is fixed. So it is closed form of growth. That's how you can relate the things. So because the meristem is closed in organization with fixed initials producing fixed tissue, that's why it is closed. So I hope you are clear with the meaning, why, what is open form of growth and what is closed form of growth. Is that clear? Is that clear, everybody? Guys, is there any issue with the voice? Chali. So next, which the part that I have already explained you, differentiation, dedifferentiation, redifferentiation. So when you talk about the differentiation, so it's a process by which the cells, they are derived from the root apical meristem, ya shoot apical meristem or the cambium. They mature to perform specific function. So always remember a trick, that is DM. First, differentiation results in maturation. So differentiation, like the cells which are derived from the meristematic tissue, they will get mature, they will perform a specific function, they will have a specific role, they will have a specific duty. So they will undergo structural changes to perform that particular job, right? There will be change in their cell wall, there will be change in their protoplasm as well. I'll give you an example. Just you know that xylem is also the example of a permanent tissue, yes or no? We all are well aware, right, that xylem will conduct water and minerals, right? It is having the unidirectional movement is there, right? And it will conduct water and minerals. That is what we know. And xylem is the example of what? It is the example of complex permanent tissue. It is the example of complex permanent tissue. So we have tracheids here, we have vessels here, we have xylem fibers here, we have xylem parenchyma here. Is that clear? Xylem parenchyma here. This is what we have. So now here, you know that it needs to conduct water and minerals. So tracheids and vessels, which are main tertiary element, they are the one which are the main elements which will conduct this water and mineral, right? So because they have to conduct the water, so in their case you will see they are what? They are dead. Dead means they don't have protoplasm. They don't have protoplasm. That is the meaning of being dead here, right? And it is important for their function. So their cell walls are, you know, dead. They have lignified cell wall, because if they have to pass the water, right, from the roots to the stem tip, right, you know that the strength is also required, right? And there is no cytoplasm to provide any sort of hurdle there as well. Apoplastically, the things will go on. So that's how things are connected. So as per the structure, as per the function, the structure will be, right? As per the function, structure will be. Okay, so tertiary element, you are talking about the tracheids and vessels. So cells will lose the protoplasm. They have thick, elastic, lignocellulosic secondary cell wall, right? So that it can also provide the strength. So they are adapted to conduct water over long distances under tension. So structure is always linked to the function. That's how this differentiation is. That's how the things are related. Am I clear? Am I clear? Yes? Done? Just allow me 1 minute. Okay? Sure? Okay, so now the next part is what? Next part is what? So next part is dedifferentiation. So dedifferentiation, a very important MCQ, right? So how will you get your secondary meristematic tissue, your permanent tissue, which is having a fixed fate, right? Fixed job. When it regains the ability to divide, right? This is the dedifferentiation. So here you will see secondary meristematic tissue. What will you see here? You will see secondary meristematic tissue. Is that clear? Is that clear? So phenomena where living differentiated cells, the phenomena where, right, the phenomena where living differentiated cells, right, the cells which had lost the ability to divide, right, when they regain the power of division under certain conditions. So this is the dedifferentiation. That's how you get your secondary meristematic tissue. So we have the examples of interfascicular cambium, cock cambium, which is also known as phallogen. So they are the examples of, right? They are the examples of what? They are the examples of dedifferentiation. So dedifferentiation will result in the formation of secondary meristem, right? Dedifferentiation, it results in the formation of secondary meristem. And let me tell you, students, this secondary meristem, we also call it as lateral meristem. It is placed laterally. It is also referred to as cylindrical meristem, right? Lateral meristem, cylindrical meristem. Now, here I would like to specify something. Just say here also in this lateral meristem also we have the examples of primary lateral meristem, right? Which has, because if it is primary, it has developed during embryonic stages, right? I would say developed initially during primary growth. And we do have secondary lateral meristem. Now, what does it mean? So, if you know about the anatomy of flowering plants, you'll be able to understand it very easily here. I have taken that session few days back itself. So, if you remember the structure of a dicot stem, right? Just a dicot stem is having a characteristic feature. In dicot stem, we see that the vascular bundles, they are arranged in a ring-like structure. This is the speciality of the dicot stem, a characteristic feature of the dicot stem. In the case of dicot stem, this is what you see, that the vascular bundles, right, these conjoint collateral open vascular bundles, they are arranged in the form of a ring. So now when I look at this vascular bundle, right, obviously we have xylem, we have phloem, right? We have xylem, we have phloem, and in between we have xylem, we have phloem, and in between we have the cambium. So because we have the cambium, so it is the open vascular bundle, right? Open, rather I would say open conjoint collateral vascular bundle. Open conjoint collateral vascular bundle. Why open? Because it is having cambium, so secondary growth can occur, girth can increase. So this cambium, because present in within the vascular bundle, so we are calling it as intrafascicular cambium. This one is the intrafascicular cambium. So it doesn't develop due to the dedifferentiation, but it develops during the primary growth only. Are you getting my point? It develops during the primary growth only. Is that clear? Is that clear? Sure. So intrafascicular cambium is there, right? So characteristic, this is the characteristic feature of dicot stems, right? So basically I'm trying to tell you that when it comes to this dedifferentiation, no doubt it is giving us the secondary meristem, but that meristem in some cases you will see that out of some part of that meristem is also primary in origin. It has developed during the embryonic stages, early stages, primary growth. Now in this dicot stem, whenever there will be the secondary growth, so in between there is these are the right parenchymatous tissue in between, right? These medullary rays are present in between. So the cells here will get dedifferentiated and then they will give rise to the secondary meristem. And here within this part you will see this cambium will also form. And now this cambium here is what? It is interfascicular cambium. What's that? It is interfascicular cambium. Yes or no? It is interfascicular cambium. Inter means in between. So this green part here, it is secondary in origin because it is the result of dedifferentiation, right? It is secondary in origin. It is the result of dedifferentiation. And this one is what? This one is primary in origin. So this one is the example of primary lateral meristem. Am I clear? Am I clear? So that's how it works, okay? Sure? Sure? So then comes the redifferentiation. So re means again. Re means what? Re means again, right? So the process where dedifferentiated meristematic cells, they divide and later mature again into specific tissue. So when that secondary meristematic tissue, you know that when it acquires the permanent function again, then I am calling it as redifferentiation. Is that clear? I'm calling it as redifferentiation. Then again, it will be having a new permanent function. Right, so this is redifferentiation. Jaise for an example, jaise here you are saying secondary xylem mane wood part is actually that secondary xylem, secondary phloem is there, phalloderm and cork is there, right? So they are the examples of redifferentiated things. Jaise cork, cambium, phallogem, that will form the cork, right? So cork, cambium, which is also referred to as phallogem. It is an important word. It's the example of secondary meristematic tissue. It's the example of secondary meristematic tissue, right? It is the example of secondary meristematic tissue. So when it is forming cork, so that is what? Sorry, that is the example of redifferentiated tissue. Done. So next is here again, parenchyma cells that are induced to divide under controlled lab condition are totipotent. They can generate entirely new plant. Overall, I told you growth in plant is open, right? But it can be growth is open, that is new meristematic tissues, yes, cells when they are added, right? When they are added, they will give rise to any new tissue or organ, but it can be closed or it can be indeterminate or determinate. Like that is, it can be continuous or maybe it will not be. So similarly, differentiation, jaise growth in plant can be open or closed. So differentiation is what? Differentiation is also open because cells from the same meristem, they can differentiate into different structure. It depends on the position. So it is not sure. So differentiation, growth is open, that's a different thing, already elaborated that. But we are also saying that differentiation is also open because the cells from the same meristem, right, from a same meristem, cells can give rise to some other structures. It depends on their position, okay? It depends on their position, okay? Done. So this is the examples of open differentiation. Jaise cells, they are near root apical meristem tip, they will form root capsules. Cells which are pushed to the periphery, they will give rise to epidermis. Cells which are towards the inner side of vascular cambium, they will form secondary xylem, which are towards outer side form secondary phloem. Mesophyll cells, they will differentiate as pellicate and spongy in dicots. So these are the examples that how the position is also affecting. So I hope you all have seen this diagram in your book, right? So it is showing the locations of root apical meristem, shoot apical meristem, and the vascular cambium. It arrows exhibit the direction of growth of cells and the organs. So here you can see root apical meristem, shoot apical meristem. No doubt in diagram they are showing it like this, but always remember your shoot apical meristem is terminal, but your root apical meristem is subterminal, mane a little away from the tips. Because at the tips you have root cap, right? So here you will have root apical meristem, yeah, meristematic zone here. You remember then you have elongation zone and the maturation zone with root here. So it is subterminal. So vascular cambium, right, at the lateral position. So this is what you can mark. So this arrow here is showing that there is an increase in girth or in the diameter. So basically it's an example of secondary growth. I hope it's clear. So it's an example of secondary growth. I hope it's clear. Done? Done? Sure? So primary growth of the plants and principally contribute to the elongation of plants along their axis. And in dicot plants and gymnosperms, the lateral meristem, vascular cambium and cork cambium appear later in life, right? So appear in dicot plants and gymnosperms. What are they saying? In dicot plants and gymnosperms. In dicot plants and gymnosperms, what are they saying? The lateral meristem, right, vascular cambium and the cork cambium, they appear later in life. Later in life, mane, they are secondary in origin, right? So they will increase the girth of the organs in which they are active. Any doubts? Any doubts? Sure? Done? Done. So now the growth is measurable. We can measure the growth. Yes or no? Growth is measurable. We can measure the growth. Is that clear? Is that clear? So here you can see, देखो, at cellular level, what will you see? At cellular level, what will you see? Cytoplasm. That is, protoplasm is increasing. Okay. So guys, I don't know whether you know it or not. See, when I'm saying protoplasm, there is one word protoplasm, there is another word protoplast. Protoplasm means cytoplasm plus genetic material. Some students, they even confuse protoplasm with cytoplasm only. When you are considering cytoplasm plus genetic material, you are saying it protoplasm. When do we say protoplast? When your plant cell, right, it is without cell wall. When the cell wall from this plant cell is removed, cell membrane is still there, but you have removed the cell wall, then you are calling it as protoplast. Is that clear? Is that clear? So, increase in protoplasm is difficult to measure directly. We cannot directly measure it now. So, we will go for something with some quantity which is more or less proportionate to it. Okay, if this is the growth, if this is increasing, means this has also increased, something like that. Now, growth is measured by a variety of parameters. It depends what are you considering. Just say we can measure it in the form of, here you can see, do underline it as well. Fresh weight, dry weight, even the length, area, volume, cell number, everything is considered, right? The fresh weight, dry weight, even the length, even the area, even the volume and cell number, everything will be considered. So growth is expressed as increase in cell number. Are you getting my point? Growth is expressed as increase in cell number as well, right? So here this data, you really need to remember this, that one single maize root apical meristem, one single maize root apical meristem, right, can give rise to more than 17,500 new cells per hour. One single maize root apical meristem, you are getting 17,500 new cells per hour, right? Per hour. So yes, I can change it, right? It can change into minutes. And if let's suppose in the paper, they can give you the same statement, they can mention 10 minutes, 20 minutes. So you have to remember we are talking about 1 hour. Is that clear? So growth has increased in the size of cell. You know that cells in a watermelon, they can increase by up to 350,000 times. Data is important. So growth of pollen tube is measured in terms of its length. Increase in surface area denotes the growth in dorsiventral leaf. So there are different ways by which we can check. So because here talking about the pollen tube, so always remember, right, again a PYQ, again a PYQ. Yes, please. So this here saying area, increase in surface area denotes the growth in a dorsiventral leaf, right? Increase in surface area denotes the growth in a dorsiventral leaf, and growth of pollen tube is measured in terms of its length. So boron, right, now that this part is deleted, boron is required for, basically calcium, boron, both are required, but mainly it is boron is required for the pollen grain germination. This is a PYQ. I thought I should share it here itself. Okay, so you can even write it in your NCERT. Okay, you can even write it in your NCERT. Okay, chaliye, next part here is phases of growth. Just like you remember this part from morphology. This part from morphology, do you remember this part? Done? So you have meristematic zone, you have elongation zone. You remember this from morphology. And then we talk about the maturation zone, where you talk about the root hair. Remember? And this is obviously, this thimble-like structure is root cap. which is going to protect the tip. You remember this? So same way growth is also like this, right? Firstly, we have the group of actively dividing cells with dense cytoplasm, with very less or no intercellular spaces, with a large conspicuous nucleus, with dense cytoplasm, with thin cell wall, with abundant plasmodesmatal connections. Even with abundant plasmodesmatal connections. With what? With abundant plasmodesmatal connections. This is what I have, right? This is what we see here. This is the abundant plasmodesmatal connection. Are you getting my point? Right, so that further grow. So cells, they elongate, right? And then finally they will become what? They will become mature. So same way that we are also going to see the things here. So do not forget the diagrams. Say, if you will look at this, so what is it showing? It is showing the detections of zones of elongation by parallel line technique. So which technique it is? It is the parallel line technique. What is it showing? It is showing the zone of elongation. Is that clear? So this one is the parallel line technique. Am I clear? So zones A, B, C, D immediately behind the apex have elongated most. Am I clear? They have elongated most. Sure? Sure? So period of growth is generally divided into 3 phases. Right, sorry for this. It is generally divided into 3 phases: meristematic, elongation, and maturation. Right, these are the 3 phases. So constantly dividing cells, we know that they'll be at root and shoot tip. We are talking about the meristematic phase of growth. Right, we are talking about what? We are talking about the meristematic growth. Then cells in this region, what are the properties of these cells? Rich in protoplasm, large conspicuous nucleus. Cell walls are primary in nature. No secondary cell wall here, because secondary cell wall will be there in a mature cell. It is thin, it is cellulosic. They will have abundant plasmodesmatal connection. Moreover, these meristematic cells, guys, 1 second. Done. So here you will see either no vacuoles, or if vacuoles are there, either no vacuoles, or if vacuoles are there, they will be of very small size. They will be of what? They will be of very small size. This is also the property of the meristematic tissues. Is that clear? Is that clear? Is that clear? Sure? Done, beta. So this is one thing, that no vacuoles, or if vacuoles are there, they will be of very small size, plus no reserve food material. Why no reserve food material? Because beta, it is a meristematic cell. It is just dividing. So how can it conserve the food? Because cells are they are having very high metabolism, right? They are continuously dividing. So that's why there will be no reserve food. Am I clear? Am I clear? So that's how it is connected. Okay, so these are the other properties. So yes, they can ask you, they can mention these points, and they can ask you that it belongs to what? Done? Done? So next is what? Next is the cells which are proximal. Proximal means nearby. Next away from the tip, you know that they will form the meristematic zone. You can even relate it here as well, and here as well, right? So this is, next to it will be the phase of elongation, where cell will increase in length. There will be increased vacuolation as well. So there will be increased vacuolation, like vacuoles will form, they'll increase. The cell is enlarging, new cell wall is deposited. So these are the characteristics of the cell in this phase. So vacuole development will be there, cell wall will also enlarge, new cell wall deposition will be there. Any doubts? Yes, please. Any doubts? Any doubts? Okay. So further away from the tip, what do you have? More proximal to the phase of elongation. What is nearby to that? The maturation phase. right, the maturation phase. So cells of this zone, they attain their maximal size in terms of wall thickening and protoplasmic modifications. They attain their maximal size, right? They will have the maximal size. They will be mature cells, obviously. They will have, the cell wall will be thick, the protoplasm will be modified. Obviously, it is as per their function. So most of the tissue and cell type we study will represent this phase. So this part is clear. We have completed the NCERT. Now we'll talk about the growth rate. Right, so we will talk about the two things here, arithmetic and the geometric growth rate. Things are simple. If it is like this, 1, 2, 3, 4, 5, 6, like this. This is basically your arithmetic growth. Right, students? It is your arithmetic growth. So now one cell is dividing into 2, into 4, into 8, into 16. This is your geometric growth. So what I want to explain here exactly, here I'm talking about the rates, right? So in the case of arithmetic growth, just keep it in your mind. You have one cell. This cell is dividing into 2. This cell is dividing into 2 cells, right? Just a minute. So you see that this cell is dividing into 2 cells. One is in red color, another is in blue color, right? One is in red color, another is in blue color. So now if you will notice here, right? Now if you will notice here, right? What am I trying to specify here? This single cell is dividing into 2 cells. Here, this cell is undergoing growth. It is undergoing division. It is developing into 2 cells. Out of that, one cell will again divide, but this cell is not dividing. This cell is not dividing. This cell is just growing. This cell is also not dividing, growing and growing. Here also growing. So out of these 2 cells, right? Out of the 2 cells here, right? This is one cell. Out of the 2 cells here, 2 cells here, one is dividing, one is dividing, one is not, and is just growing. So if you will notice here, after one division, you have 2 cells. After second division, you will have 3 cells. How come 3? This will divide into 2. This one is still existing. It is just growing. Then after this third division, you will see this cell will divide into 2. These 2 will grow. So this is the arithmetic growth. This is what they are trying to show. So when there is the increased growth unit, what is growth rate first? That it is the increased growth per unit time. What is the growth rate here? It is the increased growth per unit time. Are you getting my point? It is the increased growth per unit time. So this is the growth rate. Is that clear? This is the growth rate. Is that clear? So rate of growth can be expressed mathematically as well. Right, rate of growth can be expressed mathematically as well. So an organism or a part of the organism that produce more cells in a variety of ways. So we have different type of growth rates. So increased growth per unit time, right? Increased growth per unit time is the growth rate. Is that clear? So next is the geometric growth. You have the zygote. Right, we know that all the cells will divide here. All the cells will divide here. Right, so one cell will divide into 2. These 2 will also divide. You'll have 4 cells. Right, then these cells will also divide. You will have 8 cells. So that's how it will just keep on dividing. That's what you have to keep in your mind, okay? Right, so zygote, it divided. Right, geometric phase, may all the cells, they divide. Right, so some cells, they'll still retain the ability to divide. They will come under this geometric part, and some will lose the ability to divide. Is that clear? These are the growth rates. Diagrams are important. So growth rate shows that increase can be arithmetic or geometric. So again, the arithmetic growth, following mitotic cell division, only one daughter cell continues to divide while other will differentiate and mature. So always remember this trick. It is DM. It is what? It is DM. First a differentiation, then maturation. First a differentiation, then maturation. So this part is clear? Yes? Sure. So simplest expression of arithmetic growth is what? The root elongating at a constant rate. What's that? It is the root elongated at constant rate. Are you getting my point? A root elongating at a constant rate. So this is the arithmetic growth. So when you plot that, the length of the organ against the time, you will get a straight line, a linear curve is obtained. So yes, they are PYQs. Okay. So firstly, you should know what this arithmetic growth is, and then example here, they are giving us the example of a root elongating at a constant rate, and on plotting the length of organ as well against time, we will get a linear curve. So mathematically, it will be LT is equals to L naught plus RT. So LT is here, length at time T, L naught here, length at time 0, right? Length at time 0. So do remember this. 1 second. So it's LT is equals to L naught plus RT. So LT, L naught, and this is your R here. So LT mane length at time T, length at time 0. R here is growth rate to elongation per unit time. R here is what? Growth rate to elongation per unit time. This is what you have to remember here, right? You have to remember here. Mathematical expression is also important. So you can say constant linear growth, a plot of length against time t. So we are not starting it from the origin. Origin mane 0. This is not how we measure it. We are talking about the growth, right? If you have to check the growth rate, there must be some growth first. So do not draw it from the origin part here, right? So here you can see, you should also know what is there on x-axis and y-axis. So x-axis is telling me about what? X-axis is telling me about the time. Y-axis is telling me about the what? Y-axis is telling me about the height of the plant. Is that clear? Y-axis is telling me about what? Height of the plant. Is that clear? Is that clear? Right? So slowly, growth is there. There will be the growth. Right? So mathematical expression needs to be very clear. LT is equals to L naught RT. Right? LT is equals to L naught RT. Is that clear? LT is equals to L naught RT. Okay. Next is geometric. So here you will have the S-shaped growth. Right? What will you have here? You will have the S-shaped growth. Right? So initially there will be the lag phase, then comes the log phase, which we also call as exponential phase, right, students? And then you know what do we have? We have the stationary phase. So you do have to remember that, right? You do have to remember that. This is what you are talking about in the geometric phase. So geometric phase mein what exactly is happening, guys? So geometric phase mein in most systems, the initial growth is quite slow. You are calling it as the lag phase, right? The initial growth is quite slow. Then it increases rapidly. Very quickly it will increase thereafter at an exponential rate. So log or exponential phase. Am I clear? Am I clear? So in most systems, the initial growth is quite slow. It increases rapidly thereafter. So it is increasing at an exponential rate, right? At an exponential rate. So log or exponential phase is there. So both the progeny cells, you know that it is a geometric growth. They are dividing. They will retain their ability to divide. They'll continually dividing. So because here the nutrient supply is limited. Right? So here we are not saying that unlimited supply is there, nutrient supply is limited. So that's why after a certain time, growth will result in the leading phase, stationary phase, right? Growth will, right? Growth will result in the stationary phase. So if you will plot the parameters of growth against time, right? Growth against time, you'll get a S-shaped curve, sigmoid curve. So yes, it is the sigmoid curve, which is the characteristic feature of a living being, which is growing in naturally, where obviously the nutrients, the space, we know that it is going to be limited. Okay, the competition will also be there. That's how you can also relate it. Okay, so it is typical for all cells, tissue and organs of plants. So yes, they can ask you that what is typical for a tissue, cells, organ of a plant. We know that what is for them. the S-shaped growth. Okay, the S-shaped growth. So that's what you have to remember. Okay, done. So W1 is equals to W naught ERT. This is the mathematical expression here, right? This is going to be the mathematical expression here. So any doubt so far? Any doubt so far? Yes, please. Any doubt so far? So it is like this, okay? So W1 is what? So W1 is equals to W naught ERT. Once there was a PYQ that what is E? We also studied that in ecology. Remember, base of natural logarithm. Remember this? So W1 is the final size. It can be weight, height, number, anything. Initial size at the beginning of the period. R is the growth rate. T is the time of growth. E is the base of natural logarithms. E is the base of natural logarithm. Okay? Done, bit? Done? Sure. So that's what you have to remember. So R is the relative growth. You are comparing here, right? It is the relative growth. So how do you define it? Measure of ability of the plant to produce new plant material, right? What is this R? R is the relative growth here. Just here, see, in geometric growth, R is written as growth rate. In your this part here, R is the growth rate to the elongation per unit time, right? R is the growth rate to the elongation per unit time. So we are basically considering absolute and the relative growth here. So here, if I'm saying R, so R is the relative growth rate. It is the measure of ability because it is the growth rate. It is telling me how much new plant material is added by the plant, right? So it is referred to as the efficiency index. What do you call it? Efficiency index. So here, the final size of W1 depends on the initial size W0. Final size W1 depends on the initial size W0, right? Yes, final size depends on initial size. So now we will talk about this, but firstly, just have a look. So this is the growth curve that's S-shaped, which is for majority of the organs. It is sigmoid. Naturally, this is what organisms will have, right? So x-axis is telling me about the time, y-axis is telling me about the size or the weight. So it's the sigmoid growth, right? Typical of cells in culture and many higher plant and plant organs, right? Many higher plants and plant organs. Done? Sure. 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 So now let's talk about the quantitative comparison. Here we will be talking about the absolute growth rate and the relative. Relative means you're going to compare. So quantitative comparison between the growth of living systems, we can do it in two ways. Firstly, measurement and comparison of total growth per unit time. Measurement as well as comparison of total growth per unit time. In given time, this is the growth, right? Absolute growth rate it is. Now, growth of the given system per unit time expressed on a common basis like per unit initial parameter is the relative. So relative growth rate mein obviously you will consider the initial thing as well. So two ways are there. We have the example of the leaf here as well. So if you will look at the leaf, dekho, 5 centimeter square, then it will grow to 10 centimeters. So if somebody will ask me the absolute growth rate, so I will say 5 centimeter square here. Now here also it is 50, then 55. So here also the growth is 5 centimeter square itself. Yes or no? In both the cases, if I talk about the absolute growth, right, which is measurement in the comparison of total growth per unit time, measurement and the comparison of total growth per unit time. So it is going to be 5 centimeter square and 5 centimeter square. But if I have to see the relative growth, so that's how you can understand. So growth of given system per unit time to the initial parameter multiply by 100. Yes, I have to consider the initial parameter here as well. So growth of given system per unit time is basically what? It is the absolute growth. So it was 5. One case in leaf A, it was 5 at first. So 100% growth is there. In leaf 2, the initial parameter was 50, then 100. So you know that here, what will be your growth? 10% only. Got that? So read it once. Quick, read it once. Yes, please, read it once. Quick. Done? So now what are the conditions for growth? We are well aware of it. Water is required, oxygen, nutrients, they are all are important, right? So cell enlargement will be needing the water, right? Turgidity of cell helps in the extension growth. You know that in the case of a plant, plant is having the cell wall, right? Plant is having what? Plant is having the cell wall. Now when water enters in that plant cell, it shows the expansion, it shows the extension. That's how a plant grows. It becomes turgid. It shows that extension as well. So this is very important when it comes to the plant water. So plant growth and further development is linked to the water status of the plant, right? So you know that water will also act like a medium for the enzyme activities, isn't it? So it is required. Oxygen is required, will help in releasing metabolic energy. We know that in plant respiration also you need that oxygen. Nutrients, macronutrients, micronutrients, they all are important for the synthesis of protoplasm and for the source of energy as well. So every plant organism, it needs an optimum temperature. Optimum mane best suited. Optimum mane best suited. Is that clear? Optimum mane best suited temperature, right, for its growth. So any deviation from this range can be detrimental to its survival. So environmental signal, light and gravity also affect. Light, so you know that photoperiodism, phototropism we see in the case of light. Gravity also, right? Plants are growing towards the land, they will take the nutrition from that. So everything is related. And why do we say that we need the optimum temperature only? Because beta plant also cannot regulate their internal conditions, right? They are also what? Conformers. So in their case, for the enzymes to act, obviously suitable conditions are required. Otherwise either denaturation or the denaturation or the deactivation will occur. Got that? Done? Chali. So next is the development. This table here is very important, right? This table here is literally very important. So just Remember this, right? So you can see here meristematic cell, they are showing the plasmatic growth. They are showing that, cytoplasmic or protoplasmic growth. Now when plant is showing this growth, plant cell is showing this growth, right? The protoplasmic growth is there. Definitely there will be the expansion and elongation. Cell will also expand. Cell will also elongate. So after this plasmatic growth, we know that what will happen? Cell division will occur. Cell division will occur. Is that clear? Right, the cell is going to divide, the division will occur. So after that, you will also see differentiation is there, right? Differentiation is there, then maturation. So it is DM, then mature cell will form, they will undergo senescence, finally the death. Senescence is basically the process of aging. Got this? Right, so development, you know that growth plus differentiation results in development. Right, growth plus differentiation results in what? It results in development, right? So development is a term that include all changes that an organism goes through during its life cycle from germination of the seed to senescence, right? From seed germination to senescence, right? So this table is very important. The sequence of things is very important. So now you know that plants, they grow, yes, but they follow different pathway in response to environment or the phases of life to form different kind of structure. Not just plant, we see that thing in the animals also, right? In the animals also, this plasticity is there, right? You will see in some insects also that how the change in temperature can affect them, their growth. Are you getting my point? So basically plants, what are they doing? They are showing different, so what that plants are doing, so that plants are basically showing different pathways in response to what? In response to the environmental or phases of life. In response to what? In response to the environmental or phases of life. Are you getting my point? In response to what? In response to the environmental or phases of life to form different kind of structures. So this is the plasticity. Are you getting my point? So this is what? This is called the plasticity. Jaise heterophily in cotton. Cotton plant mein hetero means different. Phily means leaves. Okay. So heterophily, hetero means different. Right. Phily means leaves. Phily means leaves. So having different leaves in the cotton plant. So it can be during their, life cycle also, that jaise juvenile form is having different type of leaf, adult is having different type of leaf. Sometimes in some plants, you'll see, jaise let's suppose if a part of plant is in aquatic habitat, you will see the leaves are going to be dissected, right? So that water can pass. If it is in which habitat? Aquatic. Now, above the water, the leaf lamina is entire. So that is also the case. That is also the example of heterophily, sometimes to environment, sometimes to the lifespan. So heterophily is there in the cotton. Cotton is basically gossypium. It is even in coriander. It is even in coriander, that is Coriandrum sativum, and even in larkspur, that is Delphinium. So development in plants, right? So development mane, already told you, growth and differentiation both is under the intrinsic and under the control of intrinsic and extrinsic factor. So development may internal, external, both the factors are there. So the former one, mane internal factors, intracellular factor. So they are the genetic factors, they are the chemicals, they are the growth regulators, and light, temperature, water, they all are affecting the development of a plant. Is that clear? They all are affecting the development of a plant. Done. So when I talk about this heterophily, which is basically the plasticity, so if I have to define it, what exactly it is? So it is the ability of the same genotype. Genotype is same, genetic composition is same, genetic makeup is same, but it will give different phenotypes. How? In response to environmental changes, environmental conditions. This is the meaning of plasticity. We do see it in the case of animals as well. This is what plasticity is. Are you getting my point? Yes? Done. So it is the ability of the same genotype. It is the ability of the same genotype, right, that they are giving, they are making different phenotypes in response to the environmental conditions. So heterophily is there, right? Just a cotton, coriander, larkspur showing heterophily due to plasticity. This buttercup, which is also the scientific name is ranunculus. It is also important. So here you see the dissected submerged leaves which are under the water. They are right. They are the submerged leaves which are under the water. They are what? They are dissected. And the broad floating leaves are also there, right? So it depends where are they present. This is also the example of heterophily, plasticity, because environmental response is there. Due to the environmental response, the same genetic makeup is giving us the different phenotype. So maize and coriander seedlings, in their case, what sort of example is there? Juvenile and adult leaves, they are bit different in shape. So this is the example of developmental plasticity. So yes, heterophily is an example of plasticity, right? And here sometimes morphological heterophily is there, right? Heterophily is more than one type of leaf shape on the same plant. On the same plant you have more than one type of leaf shape. So morphological means on same plants, their morphology externally they are looking different. Cotton, coriander, ranunculus. Sometimes it is developmental heterophily, same plant but different stages of plant growth. Again, adult, juvenile form are having different, just cotton, larkspur, coriander. Habitual or adaptive, which best example is ranunculus. The leaves under the water, they are dissected. Over it, they are having the complete margin. So this is the diagram here. This one is important. Got that? This is the diagram here. And as I said, this one is important. Got it? Got it? Yes? Sure? Sure. So you can see buttercup. It is ranunculus. Scientific name is important. Okay. So this is larkspur, delphinium. So even in coriander, you will see juvenile and adult will have different leaf shape, right? So the next topic is of plant growth regulator. Very important. 1 or 2 MCQs. But I said 3 MCQs minimum you will get from PGD, but it can be directly 2 or 3 directly from the plant growth and regulator. So this is going to be a very important topic. Is that clear? This is going to be a very, very, very important topic. Am I clear? Yes? Sure? Done? So let's start it. Yes, everybody, let's start it. Just allow me 1 minute. So next is the PGRs, plant growth regulators, plant growth hormones or phytohormones. They are very important for the growth of the plant, right? Some some are basically the growth promoter, some are inhibitor, because the word that we are using here is the growth regulator. They will regulate the growth. So yes, here we will see that some will work like an inhibitor and some will work like a promoter or can act as both. So now if you think that if it is the growth inhibitor, that how can it help? I'll give you a very simple example. Let's talk about a seed. If this seed will not get the favorable conditions, right? If it is not having the favorable temperature, favorable water condition or anything like that, if it doesn't have the favorable conditions, then obviously if it's this seed will try to germinate, it will not be able to grow. So in that time, in that case, it is good that it should inhibit its growth. It should not try to grow. So that plant hormones, in such scenarios, plant hormones help. For an example, so inhibitors are required. Let's suppose there is a plant, right? Let's suppose there is what? Let's suppose there is a plant. Now, in the case of this plant, if this plant is under water stress, right? If there is a water scarcity, in that case, what's going to happen, right? Plant is under the water stress. So if this plant will keep on losing the water in the form of transpiration, this plant is already under the water stress. If it is losing more, then it will destroy this plant, it will kill this plant, right? So in that case also, yes, we need the plant hormones. Which can make sure that the water loss should not be there. Are you getting my point? So just say for that water stress, for such conditions, we have hormone ABA, abscisic acid, which is also referred to as the stress hormone. So it is not the hormone which is giving the plant the stress. Rather, it is the hormone which will release in the stressful conditions so that it can protect that plant, right? So that it can protect that plant in the stressful conditions. Is that clear? Is that clear? So plant growth regulators, what exactly are they? They are small. They are simple molecules of diverse chemical composition. Okay, they are small. They are simple molecules of diverse chemical composition. They are the phytohormones. So in their case, they are the inhibitors as well as the regulators. So you will see your Auxin, your Gibberellin, and even the cytokinin, and even what? Even cytokinin. They comes under the promoter category, plant growth promoter. And which one will be under the inhibitor category? You have ethylene, right, which is the gaseous hormone, of course, and abscisic acid, the stress hormone. Moreover, this ethylene is included in both the categories, right? It is included in both the categories, right? The inhibitor as well as the promoter, right? But majorly it is an inhibitor. Is that clear? But majorly it is an inhibitor. So this individual topic, either from its discovery, either from what will make which plant hormone or their functions, it can give you 2 questions, right? Right? It can solely give you 2 questions. So please pay attention. Okay? Kindly pay attention. Okay? Done? Okay? Sure? So here you can see, okay, so and growth of, so the discovery of all the plant hormones, all the phytohormones, it is serendipity. Mane, it is by chance discovery. It's serendipity. It is the by chance discovery. That's what you have to remember. So let's get started. So firstly, the point is indole compounds, right? Indoles compound. So it is indole 3-acetic acid, right? Indole compounds, indole 3-acetic acid, IAA, right? Important to remember. Adenine derivative, N6-furfuryl aminopurine, my favorite name, N6-furfuryl aminopurine. You are talking about the kinetin, cytokines here. Derivative of carotenoids, abscisic acid, which is the stress hormone as well. Terpenes will give you gibberellic acid, and the gaseous hormone is the ethylene. Very important paragraph. Very important paragraph here. Okay? So I told you they are on the basis of Like let's some are promoters. So if it is promoter means it will do the growth promoting activities, जैसे cell division, cell enlargement, pattern formation, tropic growth, cell division, cell enlargement, pattern formation, tropic growth, flowering, fluting, seed formation. Right? So inhibitor mein, they will respond to wounds, they will respond to stresses of biotic and abiotic origin. So they are involved in various growth inhibiting activities. They maintain the seed dormancy and abscission. So ABA and ethylene is the example, but I told you already that we can put ethylene in any of the category. Okay? Okay, but majorly where do we keep it? We keep it as an inhibitor. So that's what you have to remember, okay? So let's start with the discovery. So all the phytohormones, as I said, all the phytohormones, they have the accidental discoveries, right? They have the accidental discoveries. So we use the word serendipity for it. By chance discovery it is. Right? What sort of discovery it is? It is the by chance discovery. So Auxin, Gibberellin, Cytokinin, Ethylene, Abscisic acid, which is also the stress hormone. So if I have to talk about the precursor, Auxin ke liye you need the amino acid tryptophan. You even need the zinc ion for it, for the enzyme, right? So what do you need here? So this is important. So you need the tryptophan here, amino acid tryptophan, okay? Okay. So it is, you know that it is made from terpenoid pathways, right? Terpenoid pathways, जैसे isopyrenes, which basically you get from what? From acetyl coenzyme. So it is made from terpenoid pathway, which are the isoprene units from acetyl coenzyme A, which are the isoprene units from acetyl coenzyme A. जैसे acetyl coenzyme. So this information is important. Tryptophan is your most complex amino acid. So cytokinin, we have discussed that. So it is derived from adenine. You remember, which is adenine is what? Adenine is a purine. And we discussed that. We mentioned there N6 purpuryl aminopurine. Remember this? N6 purpuryl aminopurine. So do write it. Right, so amino acid tryptophan for this is very important. Then you know that for ethylene you need the amino acid methionine. Right, methionine amino acid is required. So once again, I'm repeating this trick. CMS, Chief Minister Sir. What's the trick here? It's Chief Minister Sir. That is, C stands for your cysteine, M stands for methionine, S stands for sulfur. So these two are the sulfur containing amino acid. So methionine is a sulfur containing amino acid, which you know. And here you need what? You need the carotenoid pathway. Done. So this information is genuinely important. So do mark it, do underline it. Okay. Okay. So it is also, carotenoids is also the terpenoid pathway. Okay. Yes. Done? Done, beta? So its main site is the shoot apex, even the young leaves. But it acts on both. This auxin, it acts on root and shoot. Right, we'll see the root and shoot, the auxin and cytokinin ratio, which is required for tissue culturing, okay? Right, which is required for tissue culturing. So do write it. Done, beta? Sure? Any doubts here? So this is also young leaves, right? Even the shoot, even the seeds, There also you will see that. So yes, you should know which one is developing where. So this young leaves, instead of shoot, it is root, rather root. Young leaves में, seeds में, roots में. जैसे this ethylene. So this one mainly in, matlab the main site is the shoot apex, right? I'm not saying it is not in root, but main site is this. So it is mainly in the root, then transported to shoot as well, right? Transported to shoot. So this helps in cell division, right? So I'm just talking about the main sites here, right? So जैसे ethylene, it's very important for the fruit ripening. So in ripening fruits, I would say aging parts में, you will see this ethylene as well. Okay, you are going to see the ethylene. So there, young leaves, roots, seeds. Here you will see root apical meristem, then transported to the shoot. So ethylene, we know that ripening fruits में, aging tissues में, right beta? And this one in leaves, this one is in root, this one is in seeds as well, right? In seeds as well, to maintain the dormancy, okay? To maintain the dormancy, okay? Done. So leaves में, roots में beta, developing seeds में to maintain the dormancy. And for ethylene, do mention that in the nodes of stem, you will find this. In the nodes of stem also, you will find this. This is important, right? So it is in developing seed to maintain in developing seeds to maintain dormancy. Got that? So again, the important information, so you can take a screenshot here. So quickly do it. Quick. Doubt? Okay. Just 1 minute. Okay. Yeah, ethylene is a gaseous hormone. So let's talk about their discoveries. Okay, so we'll discuss few extra points here as well, but ultimately you just stick to the NCERT. So why do we say, just say Auxin discovery was accidental, because that scientists, they were actually observing what? They were actually observing the phototropism. So that is why it is an accidental discovery. They got to know about this hormone, but actually they were doing what? They were just looking for a, for what? They are just looking for the plant growth hormone, phototropism. So we will talk about the Darwin here, Boysen, Jensen, Wendt. But in NCERT, we have Darwin and Wendt. So because Wendt is the one who isolated the auxin. Okay, so auxin is what? Indole 3-acetic acid, IAA, very important. And it was the first plant hormone identified, the another important plant. So IAA, it is the indole 3-acetic acid and it is the first plant hormone identified. So Darwin means Charles Darwin and his son Francis Darwin. Firstly, they observed when? In 1880. What they observed, they were actually experimenting on canary grass, right? Colutiles of canary grass, they were experimenting on that, right? They were experimenting on what? Yes, beta, they were experimenting on canary grass. So that is what you have to remember. Done, beta. So they were experimenting on canary grass, which is Phalaris species, yeah, Phalaris canarensis. You can check the spelling. So Charles Darwin and Francis Darwin, they were studying that. So when it was exposed, when it was exposed to a unilateral, matlab from one side the sunlight is there, when it was exposed to that, so they noticed that the colutile tip, it is bending towards that. It shows a bent towards the light. This is the phototropism, right? So they concluded that there is something in the tip which is perceiving the light. This is what they observed. They were like, there is something which is there in the tip, right, which is perceiving the light. It is something which is mobile, movable. It is a transmittable thing which is moving downward, and that is why it is causing the bending. So they were not looking for the hormone. They were just observing the phototropism. Okay, so this signal is the one, because see, if let's suppose if this is the tip, I'm giving it one unilateral source of light. So there is something which is developing here at the tip, which is going downward, and due to which there is a one-side growth that is basically resulting in the bending, right? That is basically resulting in the bending. So it is moving downward from the tip, right? So this signal, actually this mobile signal is going downwards. So the shaded side is elongating more, bending the collotype towards the light. Is that clear? Bending the collotype towards the light. So that is how it is working. So Boysen-Jensen's experiment in 1930 demonstrated that signal was chemical in nature. This mobile thing which was going downward, it was a chemical, right, which is going downward. So he placed, he cut the tip, he placed the mica sheet which is impermeable for this chemical, gelatin blocks which are permeable for this chemical. So he saw that the signal could pass through gelatin. Matlab that signal is basically some chemical, because gelatin is permeable, but through mica it was not able to penetrate. So he observed it, that something is developing in the tip, right? Something is developing in the tip. If he was cutting the tip, he was placing the mica sheet, then that tip, then it was not able to diffuse downward, right? Because it is impermeable. Okay, so when he was putting the gelatin sheet, it was able to. So that is what he demonstrated, that tip is making something that's a chemical that is diffusing through the permeable thing, and that is causing what Darwin and his son has said. Then finally, Fritz Wendt's experiment, it is the Avena curvature test. It is even the bioassay of Auxin. It is one of the that bioassays of Auxin. So bioassay of Auxin, is that clear? This Avena curvature test, right? It is also the bioassay of Auxin. Understood? Understood, beta? It is a bioassay of Auxin. So Wendt is the one who isolated this from the oat. Oat, his scientific name is Avena. So that's why it is the Avena curvature test. So who isolated, right? If this is the question, that who isolated Auxin, then indeed, who was that? You know that the answer is Wendt, right? Fritz Wendt, yes, simply Wendt. So this is important information. Then he placed the colutile tips on the agar blocks, right? Firstly, he isolated that substance. He placed that colutile tips on the agar block, which will absorb the chemical. Then these agar blocks were placed on decapitated colluteals. So you'll take the tip, you'll place that on the agar blocks. Agar blocks are going to absorb that. Then he took that agar block, he placed on that decapitated, the one from where the tip has been removed. He placed that agar block and he experimented, like placed it in different manners, and he saw how it affects the growth. So the shoot continued to bend and elongate because agar was there, this diffusible compound was there in that. So yes, it was proving that there is some growth promoting substance in it. So what is the meaning of auxin? So auxin from Greek auxin meant to grow. Okay, to grow. So first discovered plant growth regulator it is. First discovered plant growth regulated it is. Done beta? Okay. So bioassay of auxin is your Avena curvature test. So bioassay, do you understand the meaning of bioassay? So it is measuring the concentration, even the potency of a substance by using a living organism. That's what the meaning of bioassay is, right? So measuring the concentration of a substance by using a living organism, that is what the bioassay is. Is that clear? So this is about the auxin's discovery. Then when it comes to the gibberellin discovery, quite interesting. Early observation was done by the Japanese farmers, right? They noticed the disease in the rice seedling. They called it as Bacan. So they called it Bacan. So that is foolish seedling disease. So here what was happening, that seedling which were affected with this particular disease, they were abnormally tall, they were thin, they were weak. So without even producing the grain, they were just falling down. Right, so Japanese farmer, first of all, they were the one who identified it, who noticed it. Then, who identified the pathogen which caused this disease? So in 1926, Japanese scientist, in NCERT, they have mentioned E. Kurosawa, so it is Ichi Kurosawa. He discovered that disease is caused by the fungus Gibberella fusiculae. So as per your NCERT, if I go over that, as per that wala classification, so Gibberella is again an ascomycetes. So he basically observed, right, discovered that the disease was caused by a fungus, that is Gibberella fusiculae, right, now it is called as Fusarium moniliforme. Now we know it as the Fusarium moniliforme. So he observed that even when they were taking the culture medium, the filtrates from that fungus, not just the fungus, when they were taking the culture medium from that fungus, even without the fungus itself, it induced the abnormal elongation in the rice seedling. Matlab, I'm growing a fungus in the culture, I'm just taking the filtrates, and when I was observing, it was causing the disease then also. Are you getting my point? So that's how it is. So this suggested that the fungus is producing a chemical substance, right? So later scientists, they succeeded in isolating this from the fungal compound. So it is on the name of fungi as well, gibberellin, right? But later on, subsequent research showed that gibberellin is also naturally present in the higher plants. It is not just in fungi. So yeah, initially scientists, they were thinking that it is only in the fungi, but actually it is not. Is that clear? Actually, it is not. Is that clear? Done? So more than 100 types of gibberellin have been identified in plants and fungi. So Ga3 is the gibberellic acid. It was the first purified and studied gibberellin. Which one? Ga3. So it is widely used in agriculture as well as in horticulture. So it is about their discovery. Next is the discovery of the cytokinin, which promote the cell division, right? So scientists, they were studying better factors that promote the cell division, that is cytokinases in plant, that which chemical is actually promoting what? Which chemical is actually promoting the cell division in the plants, right? Which one is responsible for that? So it was known that auxin alone, right? It was known by then that auxin alone could stimulate cell elongation. This is a very important line, I would say. So auxin alone could stimulate cell elongation. Auxin alone could stimulate cell elongation, but not sustained cell division in plants grown in a culture. Like if we are doing tissue culturing, then auxin alone could stimulate cell elongation, but for the sustained cell division in plant tissues grown in a culture, right, this auxin was not helping. So they suspected that there might be some another substance which we don't know. So now the first experiment that we discuss here is Cook's experiment with tobacco callus. First of all, I would like to explain what callus is. Callus is the undifferentiated cell mass. It is what? It is the undifferentiated cell mass. Who? Callus. Just when we go for tissue culturing, okay? Right? So in tissue culturing, what do you see? In tissue culturing, what do you see? Just we are talking about what? We are talking about the tissue culturing. We are talking about the callus here, the undifferentiated cell mass. So when you talk about the tissue culturing here, so you know that we have to take a plant part. We are calling it as explant. Explant is anything that you are taking for the tissue culturing. It can be root, stem, and whatsoever it is. You will use the word explant. Right? Any part of the plant which is taken for tissue culturing is the explant. Now you are growing it in a culture medium. Obviously, we need the sterile conditions. The conditions should not, like here, the microbial growth should not be there. It can affect the culture otherwise. Sterile conditions. You will put some chemicals here, the growth substances, the growth promoters, right? Different chemicals, carbon source, all that energy source and everything you are going to put. So we have to make sure, because if you are taking a plant material here, that is having a permanent role, that will be a permanent tissue. I need something, right? I need to develop it, I need to grow it into a complete plant. So what will I do here, right? What will I do here, guys? So here you are going to put the substances which will make sure that it regain the ability to divide, and then this plant, explant will develop into undifferentiated cell mass. Right, it is the undifferentiated cell mass, so it is not having a fixed fate, it is growing, then it will finally grow into that complete plant. So that undifferentiated cell mass is the callus. That undifferentiated cell mass is what? It is the callus. So Skoog's experiment with tobacco callus, 1940 to 50. So Fouquet Skoog and his team, right, they worked on tobacco pith tissue culture. So yes, they can ask you that they worked on which plant? Tobacco. So they observed that auxin was supporting some growth, but if we need the continuous cell division, we need some other factor. So when extracts from vascular tissues, even from coconut milk, even from yeast were added, cell division was enhanced. So it basically was a signal that there is something present. There is something which is present there. Is that clear? There is something which is present there. So the isolation of the active substance. So then later Miller and Skoog isolated the first cytokinin from autoclaved herring sperm DNA. Herring sperm is basically you're talking about a fish here, right? So Miller and Skoog, they isolated the first cytokinin, the substance that can promote the cell division from this autoclaved herring sperm DNA. This substance was a modified adenine derivative and was named kinetin. So this is not obviously naturally occurring. So kinetin is not a natural plant hormone, a very, very, very important line, because it was not found in the plant. It was in the sperm of a fish, of an animal. So it was artificially discovered, but it revealed the class of compounds which can do the same. Actually, when you read the story, right, it's not like that they were well aware that, okay, we have to add the substances. Actually, it was something out of frustration. Just let's suppose you are working in a lab, you're not getting the result, right? Out of frustration, a scientist did what? Some old bottles were there. Out of frustration, he placed everything. Then he observed that after placing chemical from one bottle, like suddenly that culture started growing. Then he was like, okay, that bottle is of DNA. He started putting the DNA, but growth was not there. Then he realized that bottle was quite old. So maybe the degraded DNA is there. That's how they reached to, because cytokinin is ultimately what? Adenine derivative. That's how they reached to that chemical compound. So that's how the things are related. Okay. So, natural cytokinin. So kinetin is not the natural plant hormone. It is basically the artificially discovered one, right? So later naturally occurring cytokinin were identified in plants, and the first one discovered was zeatin. Zeatin, because it is obtained from the plant Z maize, right? So it is isolated from corn kernels, Z maize. So today many natural and synthetic auxins are known. Am I clear? Am I clear? Am I clear? So auxin alone cannot sustain cell division, right? It needs the additional factor. So Skoog and Miller, 1955, they discovered kinetin, first a cytokinin from herring sperm DNA, which was artificial, proved existence of this substance. These are the short notes, of course. So first natural one is zeatin from the maize kernels. So they are the adenine derivatives which promote cell division, even organ formation, and this cytokinin, very important function of cytokinin. It delays senescence, which is known as Richmond-Lang effect. So if I will be the examiner, I'll put this question that what exactly is Richmond-Lang effect? So Richmond-Lang effect mane, it is delaying the senescence. Is that clear? Is that clear? So next is the discovery of ethylene, which is the gaseous plant hormone. So early observation mein beta, matlab it is very interesting that it is interesting, but this part is also interesting and important. You might get question from this part this year. Next, discovery of ethylene. So as early as 1800s, it's good, huh? People noticed that the street lamps, because at that time they were using the coal gas, so they were noticing that the plants which are producing nearby to that street lamps, they were showing leaf abscission. The leaves they were shedding early, and stem, it was getting thickened. They were like, okay, there might be some chemical which is coming out from the coal and which is causing that. Okay, fair enough. Then Cousin's experiment, if you will check the data, too many things are there, but I tried to keep it with in accordance with the NCERT. So Cousin's experiment we will discuss, but here one more scientist, just Dmitry Neljuvov. He was a Russian scientist. He studied that pea seedling was showing abnormal growth in the lab. He discovered the triple response. So beta, triple response is again a very important thing. This triple response is actually the bioassay for ethylene, right? Whenever, wherever you read the word triple response, always remember ethylene. So triple response is like stem is not elongated. Just stems, they grow like this vertically. It was not elongated. Rather, its diameter was increasing. It was getting thickened, right? Stem was getting thickened, and then horizontal, obviously horizontal growth, and then it was forming some, hook-like structure, right? So he observed that illuminating gas, it was doing that, but this active compound was identified as ethylene. So when I'm talking about the Cousin's experiment here, right, I'll come to that NCERT part here. So but firstly, I'll tell you what happened in journal. Just confirmation of ethylene as a plant growth hormone, because initially they were thinking that coal gas is releasing that ethylene that is causing all that effect. But later on, they identified that ethylene is actually released by the plant itself. Okay, so R. Jain demonstrated that the ripe bananas and other fruit, they also release this ethylene gas. So he basically confirmed that ethylene was naturally produced by plant. It is not just an external pollutant, because initially it was just considered as the external pollutant. This I gave you the coal gas example. Okay, so it was identified as first gaseous plant hormone. Later studies showed its role in fruit ripening, leaf abscission, flowering, and stress responses as well. Am I clear? And stress responses as well. So I would like to specify this thing from the NCERT itself so that you don't get confused. Just during H.H. Cousins 1910. H.H. Cousins 1910, they confirmed the release of a volatile substance from ripened oranges that hasten, that speed up the growth of the stored unripe bananas. So later on it was identified by ethylene. So yes, in the NCERT, the scientist name is given. You should remember this, even the year. Okay, that is important for you. But the information I have shared, this is to specify that at least you should know what actually happened. And it is very interesting to know that it is the coal gas street lamp where coal gas was used. Nearby that, they observed it, that this is happening. And then they, try to identify it, and that's how the things went. Okay? Okay? No, it is the apical hook there, not the tendril. And now for the Auxin, during mid-1960s, three independent researchers, they were not working together. One was working somewhere else. So they find out three inhibitor. They purified and chemical characterized three inhibitor, which were later considered as the same and was named as abscisic acid. So one named it as inhibitor B, one named it as abscission 2, and one named it as dormant. So it was actually the same compound. So later all three were chemically identical. It was named abscisic acid, which is also the stress hormone, which is also referred to as anti-transparent because it prevents the transpiration. Is that clear? So it is anti-transparent. It also prevents what? Transpiration. Okay. So triple response in ethylene, I would like to specify it. Very important it is. So it is the bioassay of ethylene, right? It is the bioassay for ethylene. There are others also, but the important one is this. So what exactly is this triple response? Wherever you hear the word triple response, just remark ethylene. But firstly read the question carefully. So inhibition of stem elongation. It will, and it is also a way. Just say, let's suppose a seed is growing. There is a hurdle there, right? So how that seed will grow? So its seedling now will not grow, will not elongate, will show the horizontal growth. That will result in the thickening of the stem. Is that clear? Is that clear? So stem or the hypocotyle growth slows down. Stem or the hypocotyle growth slows down. That will reduce cell elongation. Seedling becomes shorter and thicker. Right, seedling will be shorter, but it will be thicker. So actually the stem swelling, which we are saying horizontal growth, yeah, radial expansion occurs. So stem diameter increases, right? Lateral expansion of cells is there. Stem will get thickened and then it will lose its vertical growth. It will bend or grow horizontally. So this will help that seedling to push through soil more effectively. And not just this, then because it is growing, first of all, instead of vertically, horizontally, stem is getting thickened, second response. Then also there is a formation of apical hook. At tip, there is a kind of thick hook-like structure will also develop. Okay, so its significance is it helps seedling emerge through the soil by protecting the shoot apical meristem. So short, thick stem will prevent the damage. Horizontal growth will help the seedling to path, which is showing the least resistance. So it's, as I said, diagnostic test and also the bioassay test. Got it? Chaliye, next part. ABA discovery. So stick to what we have discussed. So yeah, scientists noticed that plants produce certain inhibitor that can cause dormancy and leaf abscission, that is shedding of leaves. So they were thought to be different, but later identified as same. So you know that 3 independent researchers worked on it. There's a Frederick Edicott and colleagues. They study abscission of cotton fruits. Then they named it abscission to, right? Philip Waring and colleagues, they studied the dormancy in woody plants and buds. They isolated this and called it dormancy. So one was studying abscission, dormancy, and later on, right? Later on, you know that they all were the same compound. So stick to the NCERT part here. Here also you can, you have to remember this event was the one who isolated the Auxin from the oat seedling. So oat scientific name is Avena. But Darwin and his son, they worked on canary grass, Phalaris canarensis. Okay, so that's how you have to remember. Baki, let's talk about their functions as well from the NCERT and extra as well. Okay, so then we will be done with this chapter, right? So matlab another 20 minutes or 30 minutes and this chapter will be over. So after this, we will go for the photosynthesis, okay? After this, what are we going to start? We are going to start the photosynthesis. So photosynthesis is again, because I'm not writing it by myself. It's not like that I cannot, but yeah, just to save the time. Okay, so it's a marathon. I don't want to compromise the content. So I thought, why to write everything, right? So I'm just putting the slides so that, you know, we can just save the time. Later on, I'll share these notes. So next is the Auxin. You already know Greek it means to grow. So first it was, right, isolated from the human urine. Okay, so it is indole 3-acetic acid and there are other natural and synthetic auxins as well. So they are generally produced by growing stem and roots. Main site I told you that is stem, but yeah, it is in root as well. So from there they will migrate to the regions of their action. Okay, so IBA and IAA, that is indole 3-butyric acid and indole 3-acetic acid, is derived from plants. We have naphthalene acetic acid, dichlorophenoxyacetic acid, even we have 2,4,5-trichlorophenoxyacetic acid. They are the synthetic auxins. So they help to initiate rooting in stem cuttings. General things I'm explaining because it is totally memory based. So auxin promote flowering in pineapple. So prevent fruit and leaf drop at early stages. Matlab it will not allow the fruit and the leaf to drop at the early stages, but in the mature leaf, they will be like, okay, you just fall down so that nutrient can be mobilized to other growing part. So in most higher plants, growing apical bud inhibits the growth of lateral bud. So this is apical dominance. So this is NCERT, but how will I simplify it? I'll come to this. So first of all, I told you already, it is the amino acid tryptophan which is required for their formation. Zinc ion is also very important. And secondly, here, when I talk about auxin, you remember this, it shows the polar transport. Here we will talk about the synthetic and the natural auxin. And in case of auxin, A for auxin, A for apical dominance. This is something which you have to remember in the case of auxin, right? These are the bullet points that I have mentioned. So let's start this. Let's discuss that. So auxin mein, yes, you have the category, the natural one which we are getting from the plants, right? So natural auxin is there and we have synthetic auxin. Natural auxin is there and the synthetic auxin is there. So natural mein, as you know, IAA, indole 3 acetic acid, and indole 3 butyric acid is also there. But let me tell you students, IBA comes in both the categories. We'll discuss that also. IBA comes in both the categories. So synthetic auxin mein you have NAA, firstly you write it, then I'll explain it. This is important. And yes, this one is also important because it comes in both the categories. So this one is basically a weedicide. It kills weeds, dicot weeds, right? It kills dicot weeds. So you first write it, then I'll explain it as well. So this information is important, okay? Done, beta? Quick. Done, beta? So first of all, IBA here. IBA here is the most common auxin. Now IBA, indole 3 butyric acid is in both because it is naturally produced in plants, right? It is naturally produced in plants, right? But I won't say that in too much of large quantity, but is also synthesized. It is also synthesized artificially, right? It is also synthesized artificially and excessively used in the agriculture. That's why we are keeping it in both the categories. Okay, so when it comes to the most common type of auxin, it is your indole 3 acetic acid. IBA can come in both the categories, right? right? IBA, done beta. IBA will go in both the categories. So these are the synthetic auxins. So 2,4-D dichlorophenoxyacetic acid, it is actually a weedicide, kills weed. And to be very specific, it will kill the dicotyledons. This point is clear. Now the next point is what? Next point is what? Next point is that do you know auxin is the plant hormone that shows the polar transport? Do you know that? This is also important. Auxin, right? You will not talk about it in any other case. It shows the polar transport. Polar transport means it is unidirectional, yes or no? Yes or no? Now what's the meaning? Just say in stem. In stem you will see it will move from tip to base. Yes or no? It will move from, in the case of, yes please, matlab it shows the polar transport, that's what I'm saying. So in shoots, yeah, in stem, it will move from tip to the base. So tip is apex here, to the base it is moving, right? So in that case, you will call it as what? You will call it as basipetal transport, right? You will call it as what? You will call it as basipetal transport. And in roots, what will happen? It will move from, yes please, in roots also there is a movement. Matlab mainly it is synthesized in stem, but in roots also. So it moves from root tip, right? Moves from root tip to upwards. So here we will see acropetal transport. What will we see? We will see acropetal transport. Is that clear? We will see acropetal transport. Is that clear? So why there is such polar transport here? Yeah, why there is one direction, right? One directional transport, unidirectional, yeah, one directional transport. Why is it so? Anybody? Anybody? So auxin movement is in one direction only through plant cells. So one direction only through plant cells. Done. So this is what we have. So first of all, beta, there is the unequal distribution of the carrier proteins, right? Right? Yes, please. देखो फर्स्ट ऑफ ऑल ऑक्सिन, राइट? ऑक्सिन का जो ट्रांसपोर्ट है, द ट्रांसपोर्ट ऑक्सिन ट्रांसपोर्ट is actually beta an active transport, right? It is an active transport, means you have to use the ATP for its movement, and its transport is through carrier proteins. It is not randomly या directly it is crossing the membrane. You need the carrier proteins here, जैसे कि I would say pin protein, not required for you, right? But just for your knowledge. So it will actually enter there in the protonated form, and that's how it will go. So these carrier proteins, now they are present on the one side of the membrane. So their distribution is actually uneven, right? Their distribution is actually what? Their distribution even is uneven. Are you getting my point? So such type of distribution they have. So yes, in their case, what you people are going to see that there will be, right, the polar transport in their case, right, beta? So even the chemiosmotic mechanism is there. So polar transport is there because of these carrier proteins. They are located asymmetrically. Okay, so auxin move directionally from cell to cell, right? It can move directionally from cell to cell. Okay, so because its distribution is uneven. Done? So this is what you need to know about the auxin as well. So now we will see its discuss its function. So very first thing that even I have specified is the apical dominance. So auxin shows that. So what exactly is happening is the apical dominance. Just say, let's suppose there is a tip at the bud. Oh, sorry, there is a bud at the tip. So this bud is not allowing the lateral buds to grow. This is because here, obviously, you know, auxin is also developing. So it is inhibiting the inhibiting the growth of lateral buds. Right? It is not allowing the growth of lateral buds. So this is actually what? Apical dominance. So if we remove, if we remove this bud, if we remove this, right? If we decapitate it, then what will happen? The lateral buds will grow. And the another way is its counter action is by cytokinin. So basically, if we give cytokinin, then these lateral buds can grow. This is what you have to remember here. So now I will discuss it again, right? Because you know a lot of things now. We have discussed it separately as well. So yes, we do have the firstly, auxin was isolated in the human urine, right? This can be asked as a question, number one. Number two is, right, we have natural and synthetic one, told you already, right? So auxin is very helpful to initiate rooting in the stem cutting. So in tissue culturing, we need the ratio of auxin and cytokinin for this organogenesis. So it initiate the rooting. This is important. This is important in the stem cutting. So, and auxin also promote flowering in pineapple. So here, even if you are not able to remember the extra points, whatsoever is written in NCERT, that is important for you. So yes, it will prevent fruit and leaf drop at early stages. At early stages, it will not allow them to fall down, but later on, when in the older mature leaves, it promotes the abscission, right? Because then the nutrients will go to the other parts. So in most higher plants, the growing apical bud, they inhibit the growth of the lateral bud. This is also apical dominance, important it is. So when we decapitate the shoot tips, right, usually results in the growth of lateral buds. So it is widely applied in tea plantations and hedge making. Hedge making, you know, just say in the parks or the gardens that you visit, you will see that, you know, a proper type of like some plants, they are forming the kind of boundary there, that hedges. You have checked it. So this is used for that hedge making, right? This thing is used for that hedge making. Now, auxin also induce parthenocarpy, important. Again, whatsoever is in NCERT, that example is important. So example may we have the tomatoes. What do we have? Tomatoes. So parthenocarpy mane without, fruit is developing without fertilization. Fruit is developing without fertilization. So if without fertilization, if there is a fruit, it means it will not have seed. So parthenocarpy mane seedless fruit. That results in what? Seedless fruit. So it is used as a herbicide, a weedicide, because it kills the dicot weeds. So 2,4-D is the example, again a PYQ. So it will not affect the mature monocot plants. And for making weed-free lawn by gardeners, it is used. So it also controls xylem ka differentiation and help in cell division. This is important. This is important because already told you. So gibberellin, you know that again, terpenes are going to make it. So we know that more than 100 gibberellins are reported. So if you will ask me for gibberellin, just for this part, auxin, we discussed the apical dominance. Here you will talk about the bolting, right? Here you will talk about the bolting effect. This is the important one in this case, right? This is the important one in this case. In the case of what? In the case of gibberellin. What will I talk about? I'll talk about the bolting. So what exactly is the bolting? When you see the rosette plants, right? Rosette plants, mane jaise you see so many leaves are there, right? Their internode length is quite short. So many leaves are there. So in their case, if you will give them a gibberellin, right? So they will show the increase in the internode length. So here I would like to specify, firstly we will go as per this. They are denoted as, you know, many gibberellinic acids are obtained. So GA1, GA2, GA3 are there. So they are one of the most gibberellins to be discovered, and they remain the most intensively studied form. So all gibberellic acids are acidic. This is important. They all are. That's why you are saying gibberellic acid. So they are acidic. They produce a wide range of responses in the plants. So they cause increase in the length of axis and used in the growth of the length of used in the plants For what? For increasing this grape stalks. So each and every point is important here when it comes to their function. So they also they also cause fruit like apples to elongate and to improve its shape, right? To elongate and to improve its shape, right? Then they also delay senescence, but actually, no doubt, because this is a promoter, it is delaying senescence, but that delaying senescence, mainly it is the function of your cytokinin acid, that is also referred to as Richmond-Lang effect. So thus the fruits can be left on the tree for longer period to extend the market period. So they also speed up the malting process in the brewing industry. So this thing that they have mentioned, right, that their ability to cause an increase in the length of axis, right? So overall, this bolting effect, you really need to remember, right? Baki sugar cane, they stores carbohydrated sugar. So when we spray the sugarcane crop with the gibberellins, they increase the length of the stem. So yield will be better, right? As 20 tons per acre. Then spraying juvenile conifers, cone-bearing plants, gymnosperms, with the GAs, they will speed up their maturity period. Early seed production will be there. So this word here, very important function it is. It promotes bolting. Gibberell, जैसे your Auxin, apical dominance. Here, bolting. That is internode elongation just before flowering in your rosette plants like your beet, cabbage, and many plants with this rosette habitat. So do read it once. And if there is any doubt, do let me know. Yes? Done. So rosette habitat, I hope you are clear that here what does it means? That internode length is short. What is short? Internode length is short such that leaves appear. It will be like you will see that leaves are growing in a group, in a cluster, in a circular manner. Such as leaves appear to grow in a circular manner. They appears like that because internode is quite short, right? Internode is quite short. Okay, done. So they appear like that they are growing in a circular manner. Okay. So gibberellin, if it will be given, the internode will elongate. Any doubts? There is one thing that is vernalization. It used to be there in your syllabus, but now it is deleted. Vernalization means low temperature treatment. Listen to me very carefully. I am talking about vernalization. This vernalization means it is the low temperature treatment. Okay? It is what? It is the low temperature treatment. Matlab here, there is the exposure of plants or their seeds, right? Exposure of plants or seeds to what? To the low temperature, because this is the cold treatment actually, to the low temperature so that they can induce flowering or the stem elongation. To induce flowering or stem elongation. So I'll say that it is, there are various spring varieties, winter varieties. In the case of wheat, we use it, even in cabbage, etc., etc. These are the examples that we have. Now, if instead of giving them the cold treatment, right? So this gibberellic acid can replace this cold treatment. Instead of giving the cold treatment or low temperature treatment, you can expose them to, you can provide them the gibberellic acid, right? That will replace that, okay? Okay, that will replace that. Done? Done? Okay, next is cytokinin, which helps in the cell division. So they have specific effects on So yes, here, what would I like to specify? Richmond-Lang effect. Richmond-Lang effect. So what does it mean? It means delay in senescence. It means delay in senescence. Is that clear? So it is the main function of your cytokinin. Is that clear? Done, beta? So cytokinins, you know that it is basically discovered as kinetin, but kinetin is not natural, it is synthetic, right? So they are the modified form of adenine, which is a purine, and it is from the autoclaved herring sperm DNA. So kinetin, as I said, doesn't occur naturally. So we have seen these cell division like property, but naturally occurring cytokinin is your zeatin. So it is from corn kernel, Z maize, and the coconut milk. So since the discovery of zeatin, many naturally occurring cytokinin and some synthetic compounds that can promote the cell division, they have been identified. So natural one is your this zeatin. Keep it in your mind. Where do you find them? Where the cell division is rapid, right? Jैसे root apices, developing shoot buds, young fruits. So they produce new leaves, chloroplast in leaves, lateral shoot growth, and even the adventitious root formation. So yes, cytokinins, they also help in each and every line as a PYQ here. So they also overcome the, right? It is the apical dominance. So they promote nutrient mobilization that can delay the leaf senescence. Very important it is. Is that clear? So cytokinin and the auxins, they are used in tissue culturing. Are you getting my point? Cytokinin and auxin, they are used in tissue culturing. Is that clear? Is that clear? Sure. Done, beta? So they both are taken in a ratio that will help, right? So if we go for high auxin, right? High auxin, low cytokinin. This is how they play a role in kinin. This is how they, so they will help, it will help in root formation. This is important. And now if you take high cytokinin, but kinin, but low auxin, then it will help in shoot formation. So that's what you people can remember here. Got that? Chalo. So ethylene, the gaseous hormone, in the case of ethylene, we know that it helps in fruit ripening, okay? So that is a very important role here. And in the case of ethylene, do remember the triple response, okay? Do remember the triple response. Is that clear? Is that clear? Done? One minute. Chaliye, so next is ethylene. So ethylene is what? It's a simple gaseous PGR. It is synthesized in large amount by tissues which are undergoing senescence. So senescence mane, right, the process of aging. Senescence mane, the process of what? It is the process of aging. This is what senescence is, and in fruit ripening it helps. So ethylene, you know that horizontal growth of seedling, swelling of axis, apical hook formation in dicot seedling. So this is actually the triple response that I was talking, okay? It is actually the triple response. So ethylene also promotes senescence and abscission of plant organ, specially, right? Senescence is process of aging, and abscission, you know that what will happen, the leaves will fall down. So it is also used in that. So highly effective in fruit ripening. It enhances the respiration rate in the fruits. You know that we call them as climatic fruits. So during the fruit ripening, obviously the respiration rate will be more. So this increase in rate of respiration is respiratory climactic. It is what? It is the respiratory climactic. So such fruits where the high, right, respiration rate is climactic fruits they are. I'll add one, I'll discuss one point here from the this NCERT exemplar question. That's an interesting one. So ethylene will also help in, this is the role as a promoter, that it breaks seed and bud dormancy, right? So it initiate the germination. You have to remember the example. This is very important. In peanut seeds, if there is a sprouting of potato tubers, ethylene also promote internode or petiole elongation in deep water rice plants. So it helps leaves the upper part of the shoot to remain above water, right? In the case of rice, it is the ethylene which is used. So it also helps in the of growth root and root growth and root hair formation, right? So absorptive area will increase, right? So here one more function, just say, see, in pineapple, if you remember, related to pineapple, we also studied the function of auxin as well. So that's how you have to draw the comparison, right? That's how you have to write it in your short notes. Just here, dekho. In the case of auxin, we discussed this thing now, that it is going to promote flowering in pineapple. Auxin promotes flowering in pineapple. And here also it is used to, so auxin promote, right? Focus on the statement. Promote flowering in pineapple. Promote flowering in pineapple. But it initiate flowering and also synchronize it fruit set. So that's how you can relate their formation. So even induce flowering in mango. So it is having many function. So source of ethylene in agriculture is ethiphen. It is SEPA, right? This is used. So ethiphen, it is CEPA, SEPA, ethiphen. So it is an aqueous solution which is readily absorbed and transported within the plant. It releases ethylene slowly. So this is ethiphen. Okay, this is what? This is ethiphen. Okay. right? So it is the source of ethylene that is used in agriculture. Okay, so it is synthetic plant growth regulator. Done? Done? So it is 2-chloroethyl phosphonic acid. 2-chloroethyl phosphonic acid. So this is SEPA. This is SEPA. Is that clear? This is SEPA. Understood? So do remember it. So it is the synthetic PGR. Okay? Right? It is basically the source of what? It is the source of your ethylene. So 2-chloroethyl phosphonic acid, and it speed up the fruit ripening in tomatoes. Right? Tomato Auxin promotes parthenocopy. That's how, with examples, you should be clear which hormone is doing what. And apples and accelerate abscission in flower and fruits. And the thinning of cotton, cherry, and walnut is also done by this. So female flowers may, in cucumber, it promotes femaleness. That is, there will be more female flowers, which means there will be more seed. So yield will be better. So next is abscisic acid. You know that it regulates abscission and dormancy. So it is having a role on plant growth and development. So it is an inhibitor. So ABA inhibits seed germination. It stops the seed germination because at that time the conditions are not suitable for that seed. That is why, right? So it stimulates the closure of stomata, right? So if stomata is closed, transpiration will be prevented. So it is anti-transpirant. So it will be done when plant is under the water stress. Okay, water stress. So this is stress hormone. So ABA plays an important role in seed development, its maturation and dormancy. So it also withstands desiccation and other factors which are unfavorable for growth. So ABA act as an antagonist of GA. Antagonist means opposite. They have the opposite function. So you know that even in our bodies, some hormones, they work antagonistically, some hormones, they work in favor agonistically. So this is what you have to remember. So we can summarize this part. So some roles could be complementary or antagonistic. Some could be individualistic or synergistic. Matlab, some hormones are working alone, some are moving together. Right, some are moving together. Just allow me 1 minute. Done. So some are working individually, some are working together, right? So number of events other than, yes, similarly, number of events in the life of a plant where more than one PGR interact to affect that event is dormancy in seeds, right? More than one hormone is required in buds, abscission, senescence, or apical dominance. Okay, so PGR is basically the example of intrinsic control. Along with that, we know that the genetic control, extrinsic factors like temperature, pressure, water, all of it, they are important, beta. So vernalization, which I have already explained, is the cold treatment. If the word is given in NCERT, you should know. So which hormone can replace vernalization? It is GA, that is gibberellic acid, flowering, dormancy, and all of that things are there. So this is all about this chapter, and I have even added the flowcharts as well, just which one is whose derivative, right? And other than that, the functions are also mentioned. Just apical dominance is basically here in the case of Auxin. So though we have already discussed, but still you can see that just malic hydrazide, Na, they will keep lateral buds of tomato under dormant. So this is the role of Auxin here. Then in the case of Gibberellin, stem or internode elongation is there. Elongation of genetically dwarf plant. If plant is genetically dwarf, we can still go for the giving them the gibberellic treatment. It can make it tall. Seed germination, because it is it will synthesize the hydrolyzing enzymes like alpha amylases, like lipases, proteases, and they will help in seed germination, right, by breaking that endosperm. So increase the length of grape stalk as well as improve the apple fruit shape. So this will also speed up the malt formation, malting, help in brewing. So height of the sugar cane, delay the senescence, but mean main delaying of senescence is done by cytokinin. Okay, so cell division is the main function of that cytokinin. Low cytokinin, high auxin, root. High cytokinin, low auxin, shoot. Differentiation already discussed. Apical dominance, Richmond-Lang effect, done. Then post harvesting, this is ethylene, SEPA, 2-chloroethyl phosphonic acid. This part is also over. Okay, beta. So I have added everything here, right, in this form as well. So these are bioassays as well, right? जैसे ethylene के लिए triple response है, cytokinin के लिए tobacco pith cell division. So this is it. Right, this is all about the chapter. So the next chapter will be photosynthesis. So let's start it after a break. So let's start at 3:15. Okay, so you can have your lunch break. Then for next 3.5 hours, right, we will not have any break. Then we will finish the another chapter, that is photosynthesis. Okay, that is photosynthesis. Hi everyone, now am I audible? Am I audible now? Okay, so now let's start the next chapter, that is photosynthesis. Okay. So this is what we are going to start. It is one of the most important chapters in the plant physiology, right? Make sure you understand all the concepts here, right? It's very easy as well, okay? It's literally very easy, okay? So let's start the next chapter, that is photosynthesis. Okay, just a minute, guys. Okay, so let's start the photosynthesis. That's the chapter name. So let's get started. We'll start with its definition, and you know its definition very well, and even in the lower grades you guys have discussed that photo, right? Synthesis means what? Synthesis means what? Synthesis means formation. And here you are talking about the photosynthesis, that is, in the presence of light, there is the formation. Formation of what? The formation of food, right? This is what we have to discuss here in detail, that how these plants, they are autotrophic, how do they form the fruit? So all that points we have to cover. So is that clear? Done? So what sort of process it is? Matlab, is it oxidation? Is it a reduction? Or is it the redox reaction? What kind of process it is? Yes. What kind of process it is? A physico-chemical process. Yes. What else? What else? And what is the meaning of this physico-chemical process? What is the meaning of being a physico-chemical process? Yes, Irene? Anybody? देखो, in general, right? Yes, you are right that when we are talking about the photosynthesis, it is a physico-chemical process. It is true, right? It is a redox reaction. It is a process in which there is the buildup of something, that is, there is the anabolism in this process. Definitely, we are going to discuss that things. We will discuss that how here the oxidation of water and the reduction of CO2 will occur. So it is the oxidation of water. It is the oxidation of water which will give us what? Which will give us the oxygen. So all that basic things, I hope you all are well aware of. So if we have to define it, yes, it is a physico-chemical process or a photobiochemical process, right? It is a physico-chemical या फिर photo biochemical process. But now, before discussing it, yeah, before going forward, so what exactly is the physico-chemical? So when you are talking about the physico-chemical, firstly you are considering the physical properties here. So physico means physical properties, like I will consider everything like the temperature, the pressure, etc, etc, right? The temperature, the pressure, I have to consider the density, I have to consider the solubility. I'm not talking about the photosynthesis in general. If I'm talking physico-chemical process, so when it comes to the physical process, so I have to consider temperature, pressure, density, solubility, etc, etc. And when it comes to the chemical process, then we talk about the pH, we talk about the oxidation, we talk about the reactivity, all that things we consider here. Now in the case of photosynthesis, because it is also this physico-chemical process, so what is happening here, in which, right, in which what will happen, guys? So in which organic compounds are synthesized, in which organic compounds are synthesized. And when I'm saying organic compounds are synthesized, I'm talking about the carbohydrates there. I'm talking about the starch here, isn't it? Isn't it? So it's a physicochemical process in which organic compounds are synthesized, but they are synthesized from the inorganic raw material. How are they synthesized? They are synthesized from the inorganic raw materials like what? Do we need to consider carbon dioxide here? Yes, definitely. Do we need to consider water here? Definitely. So they are the one which are helping in the synthesis of the organic compound, right? And in the, how will it be done? It will be done in the presence of light, right? In the presence of light and pigments. In the presence of light and pigments, we will see, right? There will be the right, photosynthesis will occur. There will be the synthesis of the carbon. There will be the synthesis of the carbohydrate. So oxygen is, what will happen to the oxygen? So oxygen is released as a, right, oxygen is released as an end product or the byproduct here, right? As an end product or the byproduct, right, beta? Right? Right? And it is not just the only product or one of the products, Mane. It is not just one product here that we have to discuss. It is one of the products here. Is that clear? Is that clear? So ultimately what these plants are doing, ultimately, so these plants, beta, they are, right, these plants are converting light energy to chemical energy. Yes or no? Yes or no? So it's a, so plants are, that's why they are also considered as transducers, because they are converting one form of energy to another, right? So overall, what is photosynthesis? So as I said, in the photosynthesis, it is a redox reaction, right? It is a redox reaction. Here you will see that your CO2 will be reduced, and here we will see that the water will be oxidized. That's what we are going to see here. CO2 is reduced, water is oxidized here. So yes, it is the redox reaction, right? So water, so oxidation, right, and the reduction of CO2 will occur. Is that clear? Is that clear? Sure. Done. So the one, so its oxidation is occurring. So it provides, obviously, H positive and electron. This one is going to accept them, right? So basically CO2 reduction will give us carbohydrate here, the sugar here, right, beta? It will give us the carbohydrate here, the sugar here, and this water's oxidation here will help in the, will release the oxygen. So if instead of water, if let's suppose you are using H2S in for this photosynthesis, then obviously you won't get the oxygen, because oxygen is coming out from this oxidizable compound. If it is the water, then the oxygen will come out. So overall this photosynthesis is anabolic, that means build up, right? It means synthesis is taking place. So if synthesis is taking place, so it is going to be endogonic reaction, that is energy is used here, right? The energy is used here. This is what we have to study here. Am I clear? Am I clear? Then we will talk about the experiment. So this is what you have to discuss here. Got it? This is what you have to discuss here. Okay, so photosynthesis, a redox reaction, CO2 ka reduction, water ka oxidation. CO2 reduction will give you the carbohydrate, or I would say the sugar, and here the oxidation of CO2. So overall, when you read about the photosynthesis, we have to start with the experiments indeed. So overall, if I talk about the mechanism of photosynthesis, your mechanism of photosynthesis. So what do you study here? You are going to talk about the light reaction and the dark reaction. So for that, we really need to understand the structure of chloroplast as well. So we will have the light reaction as well as the dark reaction. So light reaction, beta, it will take place in the thylakoid membrane. Don't worry, I'll teach you the structure of chloroplast as well, and this one will occur in the stroma. So it is the dark reaction in which actually the CO2 reduction will occur, right? In these two steps we have to discuss it, and here we will see oxygen will form, water will oxygen will release, water will form, oxygen will release, and here itself we will discuss that there will be the formation of ATP, there will be the formation of NADPH2 in the case of your photosynthesis, right? So these assimilatory part, these they will be used in dark reaction for the carbon dioxide reduction. So that's what we have to discuss here, okay? Okay, done. So before starting this, we have to discuss about certain experiments first, right? We have to discuss about the experiments first. That also I have added. Okay, just allow me 1 minute. So let's start it, okay? Okay, so already I told you about the definition. I told you what exactly is going to happen. Yes, I forgot to write the reaction. So 6CO2, 12H2, it's a balanced reaction. Sunlight is required, chlorophyll is required. You will get the glucose, right? You will get the glucose, and we know that glucose will form starch. So starch is actually a polysaccharide. It is basically a polymer of glucose itself. So oxidation of H2O, reduction of CO2 will occur. It is anabolic, that is synthesizing, and endogenic, basically energy will be absorbed here, right? So let's move to the next part where we have to discuss the experiment. So first of all, let's have a look. So experiments that are going to prove that these are the things that we need for photosynthesis. You know that for photosynthesis, chlorophyll, this pigment is required. You know that light energy is required, carbon dioxide, all that things are required. But now what we have to do, we have to focus on the, right, we have to focus on the this part, the experiments part. So very simple is variegated leaf experiment. Matlab, this type of leaf, you will call it as variegated, you know, having different colors. There's a green part is there, or this other color is there. So variegated leaf experiment mein what they did, very simple it is, right? It's a very simple experiment, variegated leaf experiment. They took a variegated leaf, or you can take any leaf, you can cover it with the black paper. Now why are you doing it? You are testing it for the starch, right? You're testing it for the starch. Starch is what? Polysaccharide of glucose, right? If glucose will form, the starch will form. We know it very well. So observation may, what is there when we observe this thing, that starch is formed only in the green, the chlorophyll containing part. Like let's suppose either you can take a variegated leaf in which the green part will show you the starch presence, and you can take a leaf, you can cover it with a black paper, and you will see the part which was exposed to light, which was green, then it will have the, right, it will have this, you can say that starch. So this is what they checked. So photosynthesis is taking place in the green parts. or of the leaf in the presence of light. So they are important. So next is the moles half leaf experiment. One is variegated leaf experiment. Next is the moles half leaf experiment. So what is happening here? You will take a part of the leaf and then you are going to put it in a test tube or beaker, whatever you want to show. You will put a part of it in the test tube and then it will be out on the outer side. Now here you have KOH. What do you have? KOH. Now, what are we going to see here? See, you will put KOH. KOH is doing what? It absorbs CO2. It's a strong alkali. It is going to absorb CO2 beta, while the other half is exposed to air. Now, what we have observed, that the part which is exposed, that is positive for starch, right? The part which was exposed, which was on the outer side, here we had what? The starch. Right, the part which is inside the KOH, it is negative. There is no starch there. Why? Because this KOH is going to absorb the CO2. Why is it so? Because this KOH is going to absorb the carbon dioxide. And this carbon dioxide is actually important for the photosynthesis. Right? It is actually important for what? It is actually important for the photosynthesis, right? Now, how can we check the starch? The iodine test is there, right? The leaf is spread out and the iodine solution is poured on it. So you will see wherever the starch will be there, because the presence of starch, if there is starch, you are putting the iodine, right? If there is starch, you are putting the iodine, it will turn into bluish color, like bluish black color. And if starch is absent, it will remain just brown or yellow. Now, another question here is that why in this experiment we have used the KOH, which is potassium hydroxide. It is the caustic potash. Right, potassium hydroxide or the caustic potash. So beta, it absorbs CO2. You have read it, but why is it so? Why not any other alkali, right? So it is basically a strong alkali, strong base. But when you talk about the CO2, it is an acidic oxide, right? KOH is what? It's a strong alkali. CO2 is what? It is an acidic oxide. When they are coming in contact with each other, so this KOH is going to react with it. You will see K2CO3 and water. So why are we going for this alkali only? Why can't we go for other alkalis? One thing is, it is easy to conduct experiment with that. The reaction is not going to be that aggressive that we cannot control that. So hey, first thing is, you will see highly soluble in water. It dissolves easily and uniformly, so it will efficiently absorb the carbon dioxide. Beta, nobody will tell you why are you going for the KOH here. Very strong base will react with it, right? So no free traces of carbon dioxide will be available, and we won't get any byproduct here. The solution will be clear and this is not going to interfere with the experiment, right? And as I said, in the schools, colleges, it is easy to conduct it. It is very easy to conduct it. It's not like a very, aggressive reaction where something can happen. So KOH is preferred because byproducts are not there, will clearly do its job. So half of the leaf exposed to CO2 will have starch. You know that with the iodine test, we can check that, okay? So what is the magnitude of the photosynthesis, guys? So it produces more than 170 billion tons of dry matter annually. Right, more than 170 billion tons of dry matter annually, and it's about 7 into 10 raised to power 13 kg of CO2 is fixed through photosynthesis. So that's what you have to remember, okay? Right, so let's go for the early experiment. So here I'll give you a trick, right? Matlab it works for me, I don't know about you. So it's PIST CN RHK. Matlab that's how I remember this. PIST CN RHK. PIST-CN-RHK, that's how I genuinely remember this. Now, what is this PIST-CN-RHK? Right, what is this? Like it's JP. I'll tell you. So J, firstly, it's Joseph Priestley. You have Jen Ingenhouse. You have Julius Wonsack. You have T.W. Engelmann. You have Cornelius Van Niel. And then last is, the scientist name is not given in NCERT, Ruben, Hassed or Kamen. So these are the experiments given in your book, and yes, in this sequence they are given. Right, that's how they were conducted. So basically, Joseph Priestley, Jenningenhaus, Julius von Sack, T.W. Engelmann, then you have Cornelius van Niel and Ruben Hasse then come in. So we will start with the Joseph Priestley, 1733 to 1804. Yes, for the year, you can simply refer to the NCERT and you can just remember that, okay? You can just remember that because it's tough to remember the years. So he performed experiments on green plants, right? How he conducted his experiment? He performed experiment on green plants, and what were his observations? His observations were simple. He kept a candle in a closed space, right? Just say, let's suppose he took the bell jar, he placed a candle there, right? Soon, which will get extinguished, number one. Now, he placed a mouse here as well. Obviously, mouse will collapse. In a bell jar it is. Yes, in a bell jar it is. So it will be collapsed here. So he concluded that the burning candles, the candles which are burning there and the breathing animals, they are doing something to the air. They are damaging it. Is that clear? They are damaging it. Then what about the experiment? So next to it is he placed a mint plant there. Matlab in one experiment, there was the mouse. For me, it's the mouse right now. It's your candle. So candle will get extinguished, mouse will die. Okay. Now what is happening? You are placing a mint plant. Why are we preferring the mint plant? Actually, mint plant is a sciophyte. What is the meaning of a sciophyte? Sciophyte means it's a shade plant, right? It's a shade plant. So look at the condition here in the experiments. What are you doing here? What are you doing here? You are just putting a plant in the shade. Why? Like you have kept a plant. Vedant, thank you so much, but please do not spam. Okay. So you are putting a plant in a bell jar, right? In a bell jar. Obviously, too much of light and all that type of plant you are not going to prefer, right? So basically, he preferred the mint plant, which is a sciophyte shade plant. Even it doesn't need too much of sunlight. So he placed it with a burning candle in a closed jar. So candle is extinguished after some time, but after around 27 days, he relit the candle inside the jar with the mint plant. So candle burned again. And a mouse also survived when kept in the jar with the mint plant. Matlab, in the second experiment, if in simple words I have to tell you, they kept a plant, they kept a mouse and a burning candle there. But this time mouse survived, right? This time mouse survived, right? Done. So plants, they restore. So what they concluded, that plant, they restore the air that animal and burning candle has removed. That's why in first case the candle extinguished, mouse died. So later Lavoisier identified this as oxygen. At that time it was not identified. Now next is Jenningenhaus. So he repeated the Priestley's experiment, right? He repeated this Priestley's experiment, but with sunlight versus darkness. He repeated that experiment, but what he preferred sunlight versus darkness. And here in this experiment, right here in this experiment, you can see, no doubt here is this. So this experiment was performed as per NCERT in 1770. So just keep it in your mind, okay? Just keep it in your mind. Done. So next is Jenningenhaus. So as per NCERT, if I go, so they have mentioned what? They have mentioned that he conducted this experiment in this particular year. Is that clear? Is that clear? Sure? 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 So then he performed it. So he repeated that. So he showed that sunlight is very important for the plants to purify the air. So when bright sunlight was there, small bubbles were formed around that green parts, right, of the aquatic plant. When sunlight was available, then he went for what? For the green parts. Like for, in the case of that aquatic plant, some bubbles were there. You know that these bubbles were later, at that time, no, they were not aware, but later they were identified as oxygen, but darkness may no bubbles were there. So they concluded that it is only the green part of the plant which releases the oxygen. Am I clear? It is only the green part which can release the oxygen. So light is essential for this purpose. So he repeated the same experiment but in sunlight, as well as in darkness. Then comes the Julius von Sachs, right beta? He provided the evidence that growing plants, they can produce glucose in their green cells. So yes, right, first case may you guys have noticed that plants, they restore the oxygen. This is what they said. Second case may we have seen the sunlight is important. And now in the third case, what we have noticed here, guys, what we have noticed here, that how that glucose can be produced in the green cells. So glucose, we know that it is stored as starch. Starch is nothing but the polysaccharide of glucose, right? It's a polysaccharide, it's a polymer of glucose actually, right? So here the green substances, you know that green color is due to this pigment chlorophyll. So we know that it is in special body. Later it was called as chloroplast. So there we will see that starch will be stored. Is that clear? Now the next experiment is very important. It is an important experiment, right? It is an genuinely a very important experiment it is. So kindly focus here. So here they are saying, right, this experiment was conducted to find out that which wavelength of light is most effective. Actually here this experiment has given us the first action spectrum. Just here what was used, green algae, cladophora. In some books, yes, even the Spirogyra is also mentioned, but you will go for what? You will go for this cladophora, which is a green algae, which is a filamentous one. Then aerobic bacteria is there, right? So they are basically attracted to oxygen. They are oxygen seeking bacteria. What sort of bacteria are they? They are the oxygen seeking bacteria. They need the oxygen, right? So a prism is used which will split the white light into its 7 components. We know it very well. So what was required? The green algae, prism, and along with that aerobic bacteria which will get attracted towards the oxygen seeking bacteria. Now what the scientists do? Angelman, he placed a filament of cladophora in a suspension of aerobic bacteria. Like you are taking the water, in that water you have the this green algae, and here you are also having the aerobic bacteria. Now what are we doing? We are providing it the light. Just let's suppose you have the prism. White light will be passed. It will split into its 7 components. Yes or no? In physics also you read about it, right? It will split into what? It will split into its components. Yes or no? Yes or no? Right? Done. So next is, he then passed the white light through a prism, split it into different colors. I told you, you know that we'll get the Vibgyor here, right? Violet, indigo, blue, green, yellow, orange, and red. Then with the algal filament, it was illuminated with this spectrum of light. So the algae carried out photosynthesis, was releasing the oxygen. So what was observed? Okay, diagram is not added here. So it was observed that aerobic bacteria, that oxygen seeking bacteria, it was clustered, right? It was clustered, grouped around the algal filament part where the red and the blue light was present, right? And why is it so? Why red and blue? Because we will study it that these two, right, they are the most effective for photosynthesis. Out of blue and red is more effective. So obviously if this light is suitable for the photosynthesis, so wherever algae will get these two lights, obviously at that place only algae is going to release the oxygen. Yes or no? At that place only the algae is going to release the oxygen. So obviously if it is oxygen seeking bacteria, it will go to that place. Yes or no? It will go to that place. Is that clear? Is that clear? So as I said, the bacteria mostly it is accumulated in these two, and very few bacteria gathered in the green region, right? Right. So in general, we'll also discuss it further. So green light, right? When you talk about the green light, so this one is least effective for photosynthesis. This one is least effective for photosynthesis. Yes or no? Yes or no? Write down this thing. Quick. Quick, students. Yes, please. Done. So green light is the least effective. Why? You know that in the case of leaves, देखो, ultimately why are we able to see any color? जैसे if we are saying that apple is red in color, leaves are green in color, because they are reflecting green color. They are reflecting green. Rest all are absorbed. They are reflecting green. So obviously plants, they are green in color. Chlorophyll is giving the green color. Means rest all the colors, they are absorbed. Right? Yeah, lights, they are absorbed. And the green light is being reflected. Right? Green light is just being reflected here. So what you have to remember, that rate of photosynthesis is maximum in blue and red light. This is important. And it is least effective in green light. Another important thing, because green light is mostly reflected by chlorophyll. So this is the first action spectrum of photosynthesis and a very important experiment. So this experiment tells us about the this action spectrum of photosynthesis. So why do we call it as action spectrum? Because this action spectrum is telling us about the rate of photosynthesis in different wavelength of light. Right? It is telling us nothing but the rate of photosynthesis in different wavelengths of light. Right? That's why we got to know that it is the red and the blue which is highly affected. Is that clear? Is that clear? Done. So next is Cornelius Van Niel experiment. But before Van Niel, what was the thought process? Right? Before Van Niel, what was the thought process? This is something that you have to remember. So people used to believe that oxygen is released during photosynthesis comes from CO2. Oxygen is released during photosynthesis comes from CO2. Actually, it was the belief system. Done? Done? Right? So Cornelius Van Niel, he was a microbiologist who worked on photosynthetic bacteria, and he was the one who gave a revolutionary idea, because before him, his experiment, people used to think that CO2 is the one from where the oxygen is coming out. Is that clear? Is that clear? So Cornelius Van Niel, he was a microbiologist. Which bacteria he studied? Purple and green sulfur bacteria. He studied purple and green sulfur bacteria, number one. Now, observation mein, photosynthesis, it's a light dependent reaction. It needs the sunlight. So he observed that photosynthesis, which is a light dependent reaction. What sort of reaction it is? It's a light dependent reaction where hydrogen from a suitable donor is reducing carbon dioxide to carbohydrates. It's a light dependent reaction where hydrogen from a suitable donor is reducing CO2 to the carbohydrate. So here you guys can see, the compound which we need to be oxidizable, CO2, light, and this is what the equation is. So in green plants, what is used? Let's suppose if instead of H2O, right, H2O, here they have mentioned 2HA, you can even write it in this way, H2A, right? We can also write it in this way. Yeah, carbon dioxide is there. So CH2O, what is it showing? It is showing the carbohydrate. It is showing the carbohydrate, and then 2A is coming out. So because here it is A is coming out, right? A you can consider any element here, right? Let's suppose if I will use H2S, then sulfur will come out. It is not the oxygen. I'm using H2O. That's why oxygen is coming out, right? That's why oxygen is coming out. So this is what you have to understand, that here that photosynthesis is a light dependent reaction where hydrogen from a suitable donor, it reduces CO2 to carbohydrates. Am I clear? It reduces CO2 to carbohydrates. Am I clear? Am I clear? Sure. So this is the general equation. So in green plants, it is the water, which is the hydrogen donor. So it is oxidized to oxygen. And in sulfur bacteria, beta, it is the H2S, which is the hydrogen donor. So sulfur or sulfate is produced, not oxygen. So people used to believe, so it is like you are challenging a belief system. Ki CO2 is giving us the oxygen, right? CO2 is giving us the oxygen. But here, what are they saying? They are saying, no, it's not the CO2. His experiment told us that it is from the oxidizable compound, right? It is from the oxidizable compound. Is that clear? So if H2O is used, water will come out, but if H2S is used, it is a sulfur that will come out, okay? So photosynthesis is a light-dependent redox reaction. Oxygen released in photosynthesis comes from water, not from CO2. So different organisms used different hydrogen donors. That's why, beta, we always, they go, it's not mandatory that if you just say, we talk about the bacteria, right? We say that bacteria is performing photosynthesis, right? But not all bacteria, even if they are performing photosynthesis, not all bacteria, all rather photosynthetic bacteria produces oxygen, yeah, evolve oxygen, right? So this type of photosynthesis is N-oxygenic photosynthesis. What sort of photosynthesis it is? It is N-oxygenic photosynthesis. Are you getting my point? It is N-oxygenic photosynthesis. So if they are releasing oxygen, then it is going to be the oxygenic photosynthesis. Is that clear? Is that clear? Done? Done? Any doubts? Sure. So this idea was later proved experimentally by Ruben, Hassad and Kamen. These were the name of 3 scientists actually. So see, done beta. So they used radioactive isotope of oxygen, right? What they used, radioactive isotope of oxygen, right? O18. So because this was the Venn Niel experiment told us that it is the water. If water is used, then the oxygen will come out. So they conducted an experiment by using the radioactive isotope, right? So because of Venn Niel's work, right, the modern equation of photosynthesis that we study, we got to know about it. And what about this experiment? They used chlorella. Chlorella, you know that it's a green algae. It's a unicellular green algae. Do you remember that? It's a unicellular green algae. They kept it in water, but that water was having the heavy radioactive and the heavy isotope of oxygen, O18, not O16, right? So when they eliminated this, oxygen was released. They tested that oxygen. Means, just say, let's suppose chlorella. You know that even if we started living in an area where in water or in our food, if uranium is there, then that uranium will get incorporated into our system as well, because we are feeding such kind of things. So that's why it is always said that our company is also important, right? It is also important what we eat, what we think, right? All the things are interrelated. So here I'm saying that chlorella, what they did with that chlorella, they placed it in the water, right? They placed it in the water. The water, in that water, they were not using O16, but O18 isotope was there. Then obviously, because it is photosynthetic, it is going to release the oxygen. They checked that, so that was O18. So conclusion is oxygen released during photosynthesis, it comes from splitting of water, not CO2. So they proved that thing. Are you getting my point? They proved that thing. Is that clear? Is that clear? Done. So this is all about the experiments. Now we will study the structure of the chloroplast. So if there is any doubt so far, then do let me know. Then do let me know quick. Then do let me know if there is any doubt. Done. So next, let's study the chloroplast, which is a type of plastid. You know that from proplastid, basically the plastids form, they can get transformed into one form to another. So if I talk about the structure of chloroplast, right? If I discuss the structure of chloroplast, you know it very well that here also we will have an outer membrane, right? Inner membrane. So because why? Because it is a double membrane bound organelle, yes or no? So outer membrane is there, inner membrane is there, and in between these two, you people will see periplastidal space is also there, right? Yeah, intermembranal space is also there. It's a double membrane bound organelle, so you have to keep in your mind. Plus we see here that there will be a membrane bound sac like structure, and that sac like structure they will pile up. one over another, one over another, one over another, like this, like this. This is what we have to study here, right? Before understanding all that mechanism and everything, you should know that what exactly this chloroplast is made up of. What do we study there? So yes, it's a double membrane bound organelle, right? This fact is clear that outer membrane is more permeable than inner membrane, right? Outer membrane is more permeable than inner membrane. Here also you will study that outer membrane is having the porins. Porins are also present on it. They are on the mitochondria. They are also present here. And now when you are talking about the inner membrane, in the case of inner membrane, you'll see that it is semi-permeable, yeah, selectively permeable in nature, right? So in this case, you will see, right, there is a membrane bound sac-like structure. So this one membrane bound sac-like structure, can you look at it? This is a membrane, right? So this structure is actually known as thylakoid. What do we call it, students? We call it as thylakoid. So just say you have the coins, you are piling them 1 2 3 4 5 6, same way. Just say, let's suppose if I have the books, 1 book 2 3, I'm stacking it, right? We are piling them up, I'm stacking it. So same way here also you will see 1 2 3, like it will be like this. So they are forming what? They are forming granum, right? So granum is singular, right? So grana is plural, granum is singular and grana is plural, right? So first thing is this. Secondly, in between these two, you have what? You have intergranal thylakoid. You have intergranal thylakoid, which is linking them. It is also known as fret lamella, right? It is also known as stroma thylakoid. This is having different names. Okay. So here in this chloroplast, stroma is what? Stroma is the matrix of this chloroplast. It is the place where dark reaction occurs. It is the place where you will find all the enzymes for dark reaction. And moreover, we know it very well that this chloroplast is also having circular DNA. It is also having 70S ribosome, right? So it is basically, chloroplast is also a semi-autonomous organelle. What sort of organelle it is? It is semi-autonomous organelle, right? It is having its own DNA. It is having its own protein synthesizing machinery. Okay, so these are some structures. So granum, right? So thylakoid, this membrane will pile up over each other. So here you will see, let's suppose if here I'll draw it. This is your thylakoid. Inside is its lumen. So this one will be thylakoid membrane. So actually in this thylakoid membrane, right, in this thylakoid membrane, there are different protein complexes that you will see here, right? There are different protein complexes that you people are going to see here. And that protein complexes, right, yeah, this area is actually the site for light reaction, right? Initially, I told you what light reaction is, what dark reaction is. So this is what you will see here. Any doubts? Any doubts? Quick, tell me, any doubts? Any doubts? Sure? Done? Done? So these are the things that you have to remember. One by one, we will understand it. You know that chloroplast, you know that mitochondria, they both are semi-autonomous organelle. We also discussed the endosymbiotic theory when it comes to their origin, that there was a eukaryotic cell. It has engulfed a small cell, small bacterial cell, right? And in that bacterial cell, that enlarged cell, that small cell has started living it has shown a symbiotic relationship, right? That small cell, just in case of mitochondria, we say that small cell produces the energy. Now a large cell has engulfed it, it started living in that cell. That cell is like, I will not destroy you, you give me the energy. Same for this chloroplast as well. A photosynthetic cell was there, it was engulfed by a large cell. Do read about the endosymbiotic theory. So this is the chloroplast structure, right? There are other things, I'm telling you this part, we will discuss it. So we have to discuss the structure in detail, right? Just say I'll tell you that this is having the membranous part, right, stroma part. So you can look at the diagram, and this diagram is from the NCERT. Okay, so outer inner membrane, stroma lamella, which is also known as stroma thylakoid, which is also known as fret lamella, which is also known as intergranal, like because it is linking to granum, so intergranal thylakoid. So yes, in the NCERT do highlight these words. So this one is grana, so stroma, where the dark site of dark reaction, I would say. It is a site of dark reaction. So ribosome is 70S, right? So starch granules, this one is for starch, and this one is the lipid droplet, right? So it is having its own circular DNA as well, right? And here you also talk about these lipid droplets are plastoglobuli. So enzymes for the dark reaction are also there in this stroma. This part is clear. This part is clear. Diagram clear. So let's suppose they can give you this diagram. They can ask you, what is the name of this droplet? Yeah, what is this? So you should know what are they saying, right? You should know the diagrams here. Okay, so you can see that this is how we are going to read it. Already explained you this, but still I'm explaining it again. So you know that granum, how that granum will form when the thylakoids, they will stack over each other. So you will see this is the thylakoid membrane here. This is the lumen here. So in this thylakoid membrane, you will see that there is the chloroplast. There is the there is the membrane. This is the thylakoid. It is its lumen. Just keep it in your mind. Lumen. And here these protein complexes are there. So this is the site of light reaction. It is the place where light reaction will occur. So light reaction, beta, right? It gives out oxygen. It gives ATP. It gives NADPH. So this is the site from where we will get it. So you can imagine this diagram, like you can remember this diagram in this way. You know that we have the granum here. So obviously, if I'm talking about an individual thylakoid, so in the surrounding, what you will have stroma. So do remember the diagram, very important it is. So membranous system having grana, having stroma lamella. So grana and stroma lamella, they are making it. So beta, in algae, right? In algae, you study that it is without grana. Like in algae, the chloroplast is not having grana. It is having the thylakoid, but that thylakoid, they are not piled up to form the grana. Do you know that? Do you know that? They say in algae, yes, we do read that in algae, the chloroplast is agranal, right? So thylakoid is present there because algae is also photosynthetic. But that thylakoids, when they will pile up, na, right? When they will stack over each other, then they are forming the granum. But that is the thing that you will not see here. So yes, this membranous system, be it grana, right, or the stromal lamella, they are the one that will trap the light energy. They are the one that will synthesize ATP and NADPH. So NADPH is nicotinamide adenine dinucleotide phosphate, right? So it forms during this anabolic process. In respiration, you will see NAD. So phosphate. Uh-oh. Done? So it is NADP here, okay? So fluid stroma, what will it contain? Enzymatic reaction where carbon dioxide is reduced into sugar and then to starch, not directly light driven. Actually, this reaction is also the light independent reaction. It doesn't need the light reaction directly, but is dependent on the products of light reaction like your ATP, NADPH. Okay, so dark reaction, yeah, carbon reaction, yeah, this dark reaction, carbon reaction it is. Even here, the light reaction in grana. So here the oxidation of water, which will give O2, and ATP and NADPH will form. Is that clear? Is that clear? Sure? Sure? Done? Sure? Any doubt? Chaliye. So the next one is alignment of chloroplast, okay? Next one is what? Next one is the alignment of chloroplast. Again, but yeah, they have mentioned it. Yes, please. They have mentioned it there, okay? Okay. So chloroplast align inside mesophyll cell. What are these mesophyll cells? Beta, when you study a leaf, right? When you study a leaf, so in that leaf, what you people will see? In that leaf, what you people will see? When you see this anatomy, na, you know that we have the epidermis, right? And in between this epidermis, this ground tissue. In the case of leaves, ground tissue is what? Right? This mesophyll, right? Mesophyll is the word that we are using for the ground tissue in the case of leaves. Yes or no? Yes or no? So, conditions and alignment. So, high light intensity, ye dekho, this high light intensity, can you see that? This is going to be parallel to the incident light. Yes or no? This is parallel to the incident light. Can you see that? This is important. Yes, everybody. So here, how your chloroplast is aligned rather. Yes, you can see that it is parallel to the light intensity. So don't you think it is just present near to these walls here? Right? Yeah, it is near to these lateral walls here. So what's the word that we are using for it? It is peristrophe. So if in the paper the question is, if the light is of high intensity, where do you find, right? Where do you find the chloroplast? You will find it parallel to the incident light near to the lateral walls. If light intensity is low, then the word is epistrophe. So you can say that it is arranged perpendicular to the incident light. So check out in your NCERT, you will find this, right, beta? So now it is perpendicular to the incident light. The word is epistrophe. And if there is moderate light, then the distribution is chloroplast is randomly placed, right? Then they are randomly placed. Is that clear? Then they are what? Then they are randomly placed. Am I clear? Sure. So it is apostrophe. Okay. So these words are important. This information is important. Yes, please. This information is literally important. Okay. Done, beta. So this is the alignment of chloroplast. So next is the pigments involved in photosynthesis, a very, very, very important topic. I'll provide you the extra information as well. And trust me, it is literally important for you, okay? So guys, see, I can see majority of students है ना, here, they are just chit-chatting. That's quite distracting, right? Matlab, I don't know why are you wasting your time here. You guys are having the social media accounts. You people have social media. You people have so many things. Why are you fooling your parents? Okay? Right. So stop doing it. Otherwise, I have to end the class. I don't have any option. If you will keep chit-chatting, I'll just end the class. I'll prefer to sleep then. Okay? So when we don't take class, then students are like, "Arey, one shots they are not taking, they are not fulfilling their promises, they are not doing this and doing that. Now this is what you are doing. So why should I take the class? I'll prefer to sleep then. Better." So either stop chit chatting, or I will ask my team to block you guys, or I'll directly end the class. That will be better. That will be better. Okay? करो करो बेटा, आशिकी करो, आशिकी करो. 3 मई को आशिकी होगी और उसके बाद भी आशिकी अच्छी होगी. Good. Keep it up. Idiots. So now the next is photosynthetic pigments. So obviously we are saying that chloroplast is required for the photosynthesis. It is having the pigment. So what exactly these pigments are doing? Basically, they are the proteins itself. They are the one which can trap the light. If they will not trap the light, then how the photosynthesis will occur? Obviously, right? How that photosynthesis will occur, right? Yes or no? So special molecules. So photosynthetic pigments are what? Photosynthetic pigments are the special molecules that will absorb, that will transmit, and that will reflect different colors of light in the visible spectrum. So I'm repeating this part again. They are the special molecules. They are the special molecules that are going to absorb, that are going to transmit, and that are going to reflect different colors of light in the visible spectrum. Is that clear? Different colors of light in the visible spectrum. So pigments, right, you know it very well. The pigments appear in the color they reflect. Like if I'm saying it is green in color, means it is reflecting green color. If it is red in color, it is reflecting red color. So it's like that, right? Yes, please. It is like that. So they use this color and they use the color they absorb, right? So they are reflecting in the color in which they are appearing, means they are reflecting that color. Others they are absorbing. So overall, you know that out of incident radiation, if it is 1 to 10, like out of that 100% of incident radiation, you know that it is just the 2 to 10% which is used by the plant for the photosynthesis. So we know that we have the photosynthetically active radiations. So they are in 400 to 700 nanometer. So this is used for the photosynthesis. So when it comes to the pigment, we will talk about the chlorophylls, carotenoid, phycobilins. Okay, chlorophylls, carotenoid, phycobilins. In the previous marathon, I have completed the plant kingdom. I have discussed the pigments there also. So please relate it, please understand it. So we have the chlorophylls, we have the carotenoids, we have the phycobilins. These are the different types of pigment. Some are soluble in lipids, some are soluble in water. So that's what we have to discuss. And obviously we will start with the chlorophylls, and we know that their color is green colored pigment, means they are reflecting green color. And for the chlorophyll, light is very important for their synthesis. I'm highlighting the important points here, beta. Light is very important for their synthesis. It's not mandatory that for every, right, it is not mandatory that for every pigment you need the light, right? You need the light for its synthesis. Done. So some are with synthesized without requiring the light, but chlorophyll is one of them. Chlorophyll is one of them which need the light. So it is soluble in organic solvent. You should know which one is soluble in water solvents. In water, yeah, which one is soluble in organic solvents. So yes, this is the one which is soluble in the organic solvents. Is that clear? Is that clear? Right? So organic solvents, do you remember their examples? Yes. जैसे ether is there, benzene is there, right? Right, in chemistry also you read it. So these are the organic solvents. Na, जैसे ether, it can be chloroform, benzene, alcohol, etc. So usually the one which are soluble in organic solvents, usually beta you will see, yeah, mostly I would say they are the non-polar, right, or slightly polar. Is that clear? So let's see this. So first of all, chlorophyll A, which is the major, or I would say universal photosynthetic pigment, right, which is the major या फिर universal photosynthetic pigment. In all photosynthetic organisms, you are going to find chlorophyll A. It is that important. If you remember, when we were discussing the algae, even in green algae, right, green, brown and red algae, if you remember, right? We have studied the green algae, brown algae, and the red algae. Yes, please. If you remember, in all the three, chlorophyll A, chlorophyll A, chlorophyll A was there. In green algae, AB, in brown algae, AC, and in red algae, it was AD, if you remember, right? So A, right? So chlorophyll A is the major or the universal photosynthetic pigment, beta. So it is basically in all oxygen liberating photosynthetic organisms. So in chromatogram, you will see its color is blue green. This is important to remember. It is going to be blue green, and formula is C55H72O5N4Mg. So this is a repeated PYQ. Central metal ion, that's very important. Which one is the central metal ion in chlorophyll? Central metal ion is Mg, right? It is Mg. Magnesium is the central metal ion, very important it is. And which one is required for biosynthesis of chlorophyll? Because these are two different things. So biosynthesis of chlorophyll will have what? Will have iron. Is that clear? Will have iron. So these are two important questions. So here, right, in chlorophyll B, I'll discuss the structure here. It is the accessory pigment or the shield pigment, right? Just say chlorophyll A, right? In a leaf, you will not see that only chlorophylls are there. Chlorophylls, carotenoids, and phycobilins, etc., etc. These are the different pigments that we have there. And this chlorophyll A is the main pigment. It is the one, which is in reaction center. We will discuss the photosynthesis, photosystems. You'll get to know about it in a better way. So now here in this case, this chlorophyll B, yeah, other pigments, they are the shield, they are the accessory pigments helping in the photosynthesis, helping that chlorophyll A, protecting that chlorophyll A from getting photo-oxidized by the light. So we will discuss it. So this chlorophyll B you will see in euglenoids, right? Even in green algae it is there. Higher plants it is having this. So it is yellowish green in chromatogram. So formula is, देखो, C55H72O5N4Mg. Mg is there. Here, C55, but H is less. Because chlorophyll A, this is a very important point. How do you differentiate their structure? Chlorophyll A is having methyl group in its second pyrrole ring, but here you will see CHO group in its second pyrrole ring. Accordingly, formula is also modified. Let me show you the structure here. So this is what you have to understand here. So this is the structure of the chlorophyll, right? So this one, first of all, this one is we also compare that if we see the chlorophyll, we see that its structure is tadpole-like, right? Its structure is tadpole-like. There is a head, right? And then there is a tail like this, like this. So it is the tetra means 4. It is tetrapyrrole head, right? And there is a phytole tail. Tail is what? Tail is phytole. Tetrapyrrole head, and there is a phytole tail. Tetrapyrrole head and phytole tail is there. So just have a look. You have the central metal ion magnesium, number 1. You will see these rings here, right? So this is going to be first, just we write it in the quadrants, no? Second, third, and the fourth. So second pyrrole ring, that's what I told you, right? In the second pyrrole ring, you people will see it here. You will see methyl group is there. And now in chlorophyll B, only one thing is different, that you will see aldehyde group here. This is very important. So this is your phytole tail. Magnesium is the central metal ion. So magnesium is what? It's central metal ion. Keep it in your mind. It's the central metal ion, and iron is required for biosynthesis of chlorophyll. Is that clear? This one is required for biosynthesis of chlorophyll. So one is central metal ion, one is required for biosynthesis. Both the things are equally important. So please pay attention here. Yes, please pay attention here. Done? Sure? Done. So tetrapyrrole ring and the phytol tail. So here you can see chlorophyll A as well. So you have magnesium. So 1, it's 2, it's 3, it's 4. So you can see this. And, sorry, my bad. Dekha? This one is the second one. Okay, so this one is the second one here. Just ये देखो CH2, CH3. That is the second one here. So below that, this one is third, and then comes the fourth one. Okay, right? So the structure needs to be very clear. So always remember your chlorophyll A is the major pigment, right? So chlorophyll B is basically kind of assisting it. This is blue-green in chromatogram, right? This is another difference. Chlorophyll A is what? It is blue-green in chromatogram, and this one, chlorophyll B, is what? It is yellowish-green in chromatogram. You need to remember both the things. So we are using this thing again and again, right? There's a chromatogram. Do you know what exactly it is? Do you know what is chromatography? Anybody? Do you know what is chromatography? Anybody here in the class? What is chromatography? Beta, it's a lab technique which is used to separate different components in a mixture. Okay, which is used to separate different components in a mixture. So basically, if you talk about the leaves, in leaves we do not just have a green color. There are different pigments. So with the help of chromatography, and to be more precise, with the help of paper chromatography, we can separate that. We in the ink, just let's suppose if you have blue color, right, which is you have the blue color mein you can also separate it. You'll get to know which different components are present there, right beta? Right? So basically we have a, right, so we have a stationary phase in the chromatography, and as the name is suggesting, it is a stationary phase which will not move. We will also talk about the mobile phase which will move. Here you will talk about the solvent that will move here beta, and obviously in the stationary phase it is stationary, it is the material that does not move, right? So substances, they have different speeds, so they will get separated from each other. Is that clear? Is that clear? Right? So paper chromatography, this is what you study here for separating these chlorophyll pigments. So paper chromatography mein beta, what will happen in paper chromatography? In paper chromatography, a paper is going to act as a stationary phase, right? There is a paper that will act as what? That will act as a stationary phase. That's what you have to remember. Understood? That is what you have to remember. Understood? Done? Done? So it's a very simple method for separating pigments. You check the retention factor here, right? We calculate that distance traveled by the substance, distance traveled by the solvent. That is how you can even check the this retention factor there, but obviously not required for you. So we just put, just say, let's suppose you will take the leaves, you will make their mixture, right? Then you will do what? You will take a paper, you will have the solvent. I don't know whether you have performed it in lab or not. You'll put that paper, the solvent is also there. You are going to put a spot on the chromatography paper, right? On a chromatography paper. So solvent will slowly move upward by what? We know that it is basically the capillary action, yes or no? So different substances, they will move, right? They will move different distances, and in that way, basically, right, from that spot, you will see that how will they separate. Is that clear? Is that clear? So different pigments separate by them, and we can also check the retention factor. So that's why in paper, when we do this technique, paper chromatography, what color we are getting on that paper chromatograph, that's what we are discussing. Ki blue green hai ya fir yellow green hai. That's it. So now, as I told you, the structure looks like a tadpole. You have the tetrapyrrole ring, the porphyrin head, I would say. Hydrophilic in nature it is, right? So it is tetrapyrrole structure with magnesium at center. So magnesium is the central metal ion here, again emphasizing it. Phytol tail, this one is hydrophobic, so that one will stay embedded in that lipid bilayer of thylakoid membrane. Remember, So this is thylakoid membrane, this is the lumen. Here you will have these pigment molecules. Understood? So synthesis ke liye, the precursor is protochlorophyll and it starts from the glycine. Do you remember glycine? The simplest amino acid. Yes, the simplest amino acid is much needed for this process. Succinyl coenzyme A is required. So succinyl coenzyme A is basically in the Krebs cycle. So you will see this is also used here. So glycine, you need iron, you need light, protochlorophyll, then there will be the chlorophyll. So this iron point is very important. Central metal ion is very important. Yes, this glycine, simplest amino acid, it is required here. That is also important. Is that clear? Is that clear? So this information is literally very important. Okay. So next is the lipid soluble pigment, that is carotenoid, another important part. So beta, pigments are really very important. Is that clear? Pigments are actually, they are really very important. Okay, they are really very important. Is that clear? Right, so they are soluble in what? They are lipid soluble, right? So it can basically, they are lipid soluble, matlab in fats, oil, they can get dissolved, right? Yes, please. So one thing that you have to remember, all the lipid soluble things, you can dissolve them in organic solvent, but all organic solvent soluble things cannot, right? They may not get dissolved in the organic, this fats, lipid soluble things. Yeah, in lipid basically. Okay, so usually the substance which get dissolved here, they are the non-polar, they can pass through, obviously, because we are saying it is lipid soluble, they can cross the membrane as well. So that's how you can remember these things. But I think you know them better than me, you study the chemistry, hai na? Okay. Mm. Done. So in green and the non-green plant part, both will contain carotenoids. They can absorb light energy. They also transfer it to the chlorophyll A. Is that clear? They also transfer it to the chlorophyll A. So it acts as an accessory pigment. So it protect the chlorophyll from photooxidation, point number one, by squelching the nascent oxygen. So it is the shield pigment. They are protecting that chlorophyll A to not to get photooxidized, right? Right, beta? So they basically do what? Right? They will squeeze that nascent oxygen, will convert it into harmless O2 so that this, they can protect the chlorophyll A. So yes, chlorophyll B is also the example of accessory pigment. Your this carotenoids, they are the accessory pigment. Main is the chlorophyll A. We'll see, we'll learn that how the electron will get excited from this chlorophyll A. So that's what you have to study. Is that clear? Is that clear? Sure. Done. So this is the shield pigment, yellow to yellow orange, right? Pigments they are found inside the chromoplast as well as in the chloroplast. So they are not present in the eubacteria. Understood? Understood? So yes, they are the most stable pigment. So this is again a PYQ beta, that light is not required for the synthesis of carotenoids, right? So chemically, if I talk about their structure, it is terpenes, the hydrocarbons with conjugated double bonds, right? Hydrocarbons with conjugated double bond, terpene. We studied them in the case of gibberellin as well. So lipid soluble, important point, they don't need light for their biosynthesis, but yes, for chlorophyll you need that. They are very stable, yellow to yellow orange pigments they are. So even in the chromoplast you will get it, even in the chloroplast you will get it. Am I clear? Am I clear? So here types me, you will talk about carotenes, right? So yellow to, here what I said, overall your carotenoids. What I just said here, overall in the carotenoids, yellow to yellow orange. Yellow to yellow orange. So this is yellow orange colored. They are common in plants, just carrot and all, they have this. Just alpha carotene, beta carotene, I know you all know about it, right? So xanthophylls, they are yellowish, they are yellow colored. So they are yellow colored, they are oxygen containing derivative of carotenes. So here lutein, right? That the autumn leaves, that autumn color, why? Because of lutein. So it's common in plant, responsible for autumn color. Diatoxanthin, diatoms में present है. Fucoxanthin, brown algae. Right, fucoxanthin, brown algae. Brown algae is brown due to this fucoxanthin. So again, it is an example of carotenoid. To be more specific, xanthophylls. Right, to be more specific, xanthophylls. Okay, you know that beta carotenes, you people have discussed them in the case of even in the beet, even in the carrot, we have studied that they have their color because of these pigments. Is that clear? Is that clear? Yes, please. Any doubts? Any doubts? So beta carotene is very common, right? So the color of carrot is due to this beta carotene. So overall carotenoids, they do not need, right? Overall carotenoids, they do not, yes, please, they do not need what? They do not need these light for their synthesis. So they are accessory pigment, right? They will make photosynthesis more efficient by absorbing extra wavelength. Jैसे chlorophyll A, they can absorb, yeah, chlorophylls, they can absorb in a certain region. The other lights are also there. They will absorb the light of different wavelength. Ultimately, to whom they are going to pass? To the chlorophyll A. We'll study the photosystems. I'll tell you that there will be a reaction center with chlorophyll A. And in the surrounding, we will talk about the accessory pigments as well. Right? We will talk about the accessory pigments as well. So we will see that chlorophyll A will also absorb the sunlight. There are other pigments. They will also absorb different wavelengths of light. And ultimately, they all will pass it to chlorophyll A. And that will help in excitation of the electron. Right? That will help in excitation of electron from it. Right? That's how we will study the light reaction. That's how you will study the photosystems. So chlorophyll A is that universal or the major pigment in the reaction center. So rest all are the part of light harvesting complex. They are the shield pigment. They are the accessory pigments. Is that clear? So they make photosynthesis more efficient by absorbing extra wavelength. So it protects chlorophyll A from photooxidation. As I told you already, will convert the nascent Auxin into the harmless O2. Beta carotene, it's the precursor of vitamin A. That is also important. Even in class 12, the golden rice that you talk about, which is a GMO, that is genetically modified organism, sorry, genetically modified plant, there also we discuss that this is the source of beta carotene. So next is phycobilins. They are water-soluble pigments. Important it is. Right? This one is not soluble in organic or the lipid soluble. They are water soluble. So they do not have that magnesium and phytol tail. Is that clear? So phycocyanin blue, phycoerythrin red. You remember red algae is red due to this phycoerythrin. So that's an example of phycobilins. So phycobilins are water soluble. A very, very, very important statement. Is that clear? It's a very important statement. Is that clear? Is that clear? So it found exclusively in blue-green algae and even in red algae. So they are also the accessory pigments. Now you know the function of accessory pigments. Yes or no? Now you know it. So just have a look. If there is any doubt, do let me know. Quick. Chaliye, next part is, so structure for chlorophyll done. So now here you can see this is a very important topic and a very important PYQ as well. So by paper chromatography, we can separate these pigments. So chlorophyll A, you know that blue-green or bright blue-green, bright green or blue-green, yellow-green chlorophyll B. Xanthophylls will show the yellow color and carotens will go for, or I would say carotenoids actually will go for yellow to yellow orange. So that's what you have to remember here. Now, the next is, you know that wavelength, they ranges from, overall I told you 400 to 700 as 390 to 760, right? So they can be resolved into different colors. So basically what we know already from the experiment also, it is the red and blue which are highly effective for photosynthesis, right? They are very effective. If I have to compare blue and red, okay, so red light which is above 700 nanometer, if the light which is above 700 nanometer, you will call it as far red. You should know these terms. If it is less than 390 nanometer, below violet, so you will call them as UV rays. Beyond red, that is even above 760 nanometer, it is infrared rays. So photosynthetically active radiations may wavelength ranges somewhere around 400 to 700. So this part is used in photosynthesis. So you can see the different wavelengths of light as well. There's a violet ka 390 to 430 nanometer is there. Blue ya indigo, early workers will call it this, 430 to 470. So here you can see green, the one which is least effective, 500 to 580. So red color is there, so you should know which one is far red or infrared. So red ke liye you people can see 660 to 760, right beta? So that's what you have to remember. So these wavelengths of light, they are very effective for the photosynthesis. Is that clear? Least effective is green. So leaves appear green because they do not absorb green light, they reflect or transmit it. Number one. Blue and red region most effective. Blue light will carry more energy. Red light, देखो, this is also extra information, but the important one. Blue light, it carries more energy. It carries more energy. Which one? Blue light. But when it comes to red light, it is having less energy, but it is most efficient in photosynthesis. It is most efficient in photosynthesis. Is that clear? Is that clear? So blue light, it carries more energy. Red light carries less energy, more efficient out of these two. Green light, least effective. So under a tree canopy, right? I think you should know this. Under a tree canopy, mostly green light passes through, right? So therefore, plants growing under that show low photosynthetic rate because green light, they are reflecting green light. So the one which are growing under that, they will get it and they will have the low photosynthetic rate. Is that clear? Is that clear? So the next topic that we have to study is the absorption and the action spectrum. So up to this part, all clear? Up to this part, all clear? Guys, up to this part, all clear? Sure? Are you sure? Done? Any doubt so far? Done. So blue light somewhere around 430 to 450 nanometer. Red light overall 640 to 680 nanometer. So these wavelengths are absorbed the most and they provide maximum energy for the photosynthesis. Is that clear? Is that clear? Sure? Done, beta? Done, beta? So even you should know, this one is having less energy. Red light is having less energy and even it is having the longer wavelength. I hope you know what wavelength is, right? So this one is having the less energy and shorter wavelength. So do remember such things also. So let's go to the next part here. So next part may we have the absorption spectrum. So absorption spectrum, somewhere you already know this absorption spectrum. How come you know that? It is talking about the like which which light pigment is absorbing which pigment is absorbing which light. Means it is a curve which will show wavelength of light absorbed by the pigments. What is it? It is the curve that is showing wavelength of light absorbed by the pigments. Right? It is a curve that is showing which wavelength of light is absorbed by the pigments. So it is measured by spectrophotometer. Is that clear? So it is the curve which is showing the wavelength of light absorbed by the pigments. So it is measured by what? It is measured by spectrophotometer. So chlorophyll A, chlorophyll B, that absorb blue and red light most strongly. And these carotene, they absorb blue light. Okay, so basically red light most effective, but blue light is also absorbed more efficiently. Now guys, if I have to discuss, just here blue region it is the red light ka region. So from these spectrums also you should know what are they trying to tell you. First of all, ye dekho chlorophyll A, blue green, yellow green, carotenoids, yellow, right? Look at this, you should know how to depict it. You should know about it, how to depict it. So it is basically the absorbance of light by chloroplast pigment. So you can see that if I stick to this part only, right, to the NCERT. So these carotenoids, right, just here it is written, they are absorbing blue light. They are absorbing blue light. We have written this. They are showing no peak in this red region. But your chlorophyll B, your chlorophyll A, they are showing in the peak in both the regions. But I am not able to, I will not find, as per this diagram, we cannot see any peak there in the red region. Moreover, if I have to depict this part only, chlorophyll A absorb more blue than red light. Am I right? And chlorophyll B also absorb, you tell me, these points are correct or not, more blue than red light. Then chlorophyll B absorb more blue light than chlorophyll A. Am I right? And chlorophyll A absorb more red light than chlorophyll B. So as per that graph, I'm explaining you everything. So tell me, is it correct or not? Yes, please. Yes, please. Any doubts? Done. So carotenoids, I'm not just talking about carotenes, I'm talking about the carotenoids. So beta, carotenoids, do you know that this is for your information, right? This is for your information that these carotenoids, right? Look at them. Look at them. So these carotenoids, they absorb in 400 to 500 nanometer and maximum. They will absorb 400 to 500 nanometer, and here maximum absorption is in 450 to 480 nanometer range. That's how it is. So this is about the carotenoid, and this is the benefit, right? This is the benefit of having, different pigments, different accessory pigments. Just say, that chlorophyll A and B, maximum in blue and the red light, right? Blue and the red light. But here, these other pigments, they will also absorb the light wavelengths of different light of different wavelengths, and then ultimately they will provide it to whom? To the chlorophyll A. So that is also that is how this is also beneficial. Is that clear? So tell me if there is any doubt here. Quick. Quick. Done? Okay. So this is the action spectrum, right, that we even discussed in that TW Angelman experiment. So in action spectrum, what you have to discuss, this curve is showing the rate of photosynthesis at different wavelengths. What do we see here? We see the rate of photosynthesis here at different wavelengths. So it shows that photosynthesis mainly occur in blue and red region. So this is, see, look at this. Look at this. And why do we say that the red is most effective? Dekho, blue is absorbed more. It is having more energy, shorter wavelength. We know it very well, right? In comparison to that, look at its rate. Red is absorbed less. In comparison to that, look at its rate, right? So this graph only, it is showing me the rate here only, right? That maximum is in blue and in the red. Look at the absorption. Red is absorbed less, but it is having more, the rate is more here. So is that clear? So the graph, the diagrams all need to be clear to you. So wavelength of light in nanometers is given here, 400, 500, 600, 700. So this is the part that you should be knowing. Any doubts? Any doubts? Done, beta? Done, bache? Sure. Chaliye, next is this. So this graph, these diagrams, they need to be very clear. Okay, so next point is quantum yield and quantum requirement. And then let's move to the next part. So next is what? Quantum yield and quantum requirement. Okay, quantum yield and quantum requirement. So what is this quantum yield? It is it is yield, like how much oxygen is evolved per quantum of light. So it is 1/8. And requirement is number of light quanta which are needed for. So basically, if I have to make one O2, how much light quanta do I need? Their value is 8. So that's why, because this is the requirement, so yield as per that will be 1/8. It is also important information, right? It is also an important information. Is that clear? Is that clear? Is that clear? Chaliye, so let's move to the next part here. Okay, so after this, let's talk about the light reaction. So up to this part, all clear, right? So quantum yield and quantum requirement done. Acha, so here I had this part. Just a minute. Okay, guys, experiment part was also there in the previous PPT. No, it was not there. Chalo, it's okay. Done. So pigments clear. So let's start the mechanism of photosynthesis. Right, let's start the mechanism of photosynthesis. Let's talk about it. So any doubts so far? Okay, chaliye. So already I told you it is the light reaction, which is also known as Hill reaction beta. It is also known as photochemical phase, right? And we have the another is dark reaction, which is also the carbon reaction, right? It is also light independent reaction, and its another name is biochemical phase. Is that clear? Its another name is what? It is biochemical phase. So that is what we have to discuss. So Hill reaction, photochemical phase, carbon reaction, light independent reaction, and the biochemical phase. So this is what we have to discuss. So ultimately in light reaction, what is happening? Matlab in two parts we are discussing it. We know that light reaction will take place in the thylakoid membrane. Where will that light reaction occur? It will occur in the thylakoid membrane. And where do you see the dark reaction? Stroma, right? Keep it very clear in your mind. So in light reaction, ultimately what will happen? Light absorption, then water will split. Splitting of water, we know that will release oxygen, right? Splitting of water will release oxygen. Then the ATP and NADPH, which are high energy intermediates, they will form, and these are the one which are required in the dark reaction. So why do we say that the dark reaction is the light independent reaction? No doubt it's like word dark is a misnomer term here, right? Word dark is a misnomer term here. It doesn't mean that this reaction required the darkness to occur. No, it's like it doesn't depend on light directly. It doesn't need light directly, but it needs the product of light reaction. For dark reaction, I need ATP, I need NADPH, right? In dark reaction, students, you study that C3C4 cycle. Okay, so basically here for them, I need ATP, I need NADPH. It doesn't need light directly, but it needs the products of light reaction. It needs the product of light reaction. So if there will be no product of light reaction, guys, then obviously it will not occur. Is that clear? Is that clear? So basically, to understand the light reaction, we need to understand the photosystems. So here we will talk about Emerson and Arnold worked on chlorella, which gave the concept of two photosystem pigments. So first of all, just have a look. Dekho. One thing that you have to remember overall, I'll talk about the experiment. This red and blue light, they are very effective for the photosynthesis. They are literally very effective for the photosynthesis, both of them. Right, and even in the red light, we'll see the different wavelength concept as well. Just to see, first of all, let's talk about the red drop effect and the immersion effect. Immersion effect, enhancement effect. They go, red drop effect is saying that when chloroplasts are illuminated with far red, I told you what is far red. When the wavelength is above 680 nanometer, then it is far red. Right, matlab around 700 nanometer we are giving, yeah, above 680 nanometer when we are giving, we are saying it is the far red. So the quantum yield of the photosynthesis, matlab that oxygen production, it drops sharply, right? So this is basically the red drop effect. But how do we get to know it? We'll come to that. Then there is one more thing, that is the immersion effect or the enhancement effect. So in 1957, Emerson observed that if red light around 680 nanometer and even, but here it was far red, and the far red that is above 700 nanometer. Matlab you are giving 680 or 700, around 700 or 7 above that, they were given together, the rate of photosynthesis was greater, right, than the sum of their separate effect. Matlab if I'm giving these lights separately and then I'm conducting the photosynthesis, the rate was lesser, but when I'm doing it collectively, the rate was better. So it is immersion enhancement effect, and it led to the discovery of the two photosystems. Am I clear? Am I clear? Right, because of it, we know about two photosystems. Is that clear? Is that clear? Done? So when light will exceed 680 nanometer, you know that the photosynthesis efficiency will decrease. But when you are giving the red light around 680 nanometer, far right around, right, rather than here, just say around 700 nanometer, around 700 nanometer. So you will see that the rate will increase. Is that clear? So red light alone will do moderate photosynthesis, far red will do low photosynthesis, both will give the better rate. So they tell us about the discovery of the photosystems. Is that clear? Is that clear? Is that clear? Yes or no? Done. So these effects, so when it is around 680 nanometer, yeah, far red light, which is right, which is around 700 nanometer. So this is what we see. So this phenomena is called Emerson enhancement effect, right? This phenomena is called Emerson enhancement effect. So two photosystems, be it PS2, PS1, they work cooperatively in the light reaction, right? Matlab from this discovery, we got to know about the photosystems. So red dwarf effect revealed a fall in efficiency with only far red light. So immersion enhancement effect tells us that both are required for this maximum efficiency. So let's discuss what these photosystems are, and then let's talk about it further. Okay, then let's understand what is photosystem 1, what is photosystem 2. So as I was explaining you earlier as well, they are nothing but they are the group of pigments. Is that clear? Is that clear? Just a minute, guys. Done? चलिए, so photosystems में बेटा we discuss two things, right? Overall in photosystems, before understanding the photosystem 1 and 2, let's discuss the photosystems. So in photosystems, what you people will see that there is going to be a reaction center, and other than that there will be the light harvesting complex, which you also call as, right, antenna complex. What is this? This is a light harvesting complex, which is also known as antenna complex. Is that clear? Which is also known as what? Antenna complex. Is that clear? So actually these are the group of pigments. Just imagine it. This is your reaction center. In reaction center, you will only see chlorophyll A, this major photosynthetic pigment. But here you will have other than chlorophyll A, other pigments you will see other than chlorophyll A. So these are the other pigments which are present here. Is that clear? These are the other pigments which are present here. Is that clear? Is that clear? Let's suppose these are the group of pigments. Now you know that it is the chlorophyll A. Ultimately, what will we see in the light reaction? Sunlight will be absorbed. It has to emit the electron. That electron will go here and there, right? So they will produce the energy. That will form ATP, right? That is how we will see or understand the things. So chlorophyll A is having certain wavelength that it will absorb. So there are other pigments also. So these other pigments, they can absorb different wavelength, but ultimately they also pass it to the reaction center, because to exude the electron from the reaction center, more energy is required, right? So this part is making the LHC or the antenna complex. This is the reaction center. This is together making photosystem. So why do we say photosystem 1 and photosystem 2? So when we are saying photosystem 1 and photosystem 2, right? When we are saying here photosystem 1 and photosystem 2, so their names are not as per their function. Names are not as per their function. Names are not as per their function, but as per their order of discovery. Basically, you know that from this immersion effect or from this red drop effect, we got to know that electrons has to move. So electrons, how do they move? Ultimately, beta, they move from PS2 to PS1. This is what you have to remember. A very important point, right? When it comes to the electron movement is from PS2 to PS1. So name PS1 and PS2 is not as per the order of their discovery. Not as per their function, as per order of their discovery. PS1 was discovered first, PS2 later. But actually how the electrons will move, they move like this. So why PS1, why PS2? Because in PS1, you have in reaction center, the chlorophyll A, it shows the maximum absorption at 700 nanometer, but in PS2, it shows at 680 nanometer. Understood? At 680 nanometer. So that is how it works. Is that clear? Done? Okay. So I hope it's done, because it is one of the most important topics. So here you people can see it, right? That every photosystem has a reaction center having chlorophyll A, accessory pigments, which is antenna pigment or LHC. So function is to absorb light energy and to transfer it to the reaction center. I hope it's clear. So reaction center mein beta you have PS1, right? You have PS1, you have PS2. So here chlorophyll A, it will show peak at 700 nanometer. So we denote it like P700, right? PS2, chlorophyll A, Here it is showing peak at 680 nanometer. So you are representing it like this. So this one is playing role in oxygen evolution. This one is playing role here. So we will study about that. So where are they located? Now we will discuss this part. So when I was teaching you the chloroplast structure, I didn't talk about the oppressed and the non-oppressed regions. Now we will discuss it. Is that clear? So they will tell me about the location of their location and the differences in them. Is that clear? Yes? Done? Okay. Yes, please. Done. So look at the grana here, right? Granum here. So we will talk about two types of membranes here. One is oppressed region, right? Oppressed membrane. Another one is non-oppressed. This is important and you should know where will you find which particular membrane. So one word here that we are writing is oppressed and non-oppressed, right? So they are, one second. So they are basically that membranes which are tightly stacked, okay? And they are not tightly stacked. In simple words, if I have to explain you this, so oppressed region, right? As the name itself is suggesting. So they are the membranes which are in the tightly stacked part, and the non-oppressed one are not. Just say here it will be rich with PS2, right? You will find the PS2 here and you will find too much of LHCs here. So oppressed membrane and non-oppressed membrane. Just say look at this diagram. So these membranes which are in between, which are tightly stacked, just say here, dekho, they have marked it as the oppressed regions, right? Oppressed regions. Can you see that? They are the oppressed regions. Right, so here they are rich with beta PS2 and a lot of light harvesting complexes. Now you can see these are tightly stacked, right? These are tightly stacked, so they are the oppressed region. PS2 is in the oppressed region, a very important point. And this one, because it is not tightly stacked, so you will call it as non-oppressed membranes. Right, non-oppressed membranes. So non-oppressed membrane may you will have PS1. Their location also vary. So if I draw one granum and the another granum here, right? So you will see that there is the intergranal thylakoid, right? So you will see here, you will only see PS1, right? Here this side will show you PS1. This will also, matlab non-oppressed part will show you basically PS1. In between you will have PS2. So yes, their location is also very important, right? Where will you find, where do you see which one is very, very, very important here, right, beta? So here you can see this diagram in very simple words. It is explaining. So non-oppressed mane, non-stacked mane, exposed surface. Is that clear? So this one is not stacked, exposed surface. So this one is stacked, right? This one is stacked part. Is that clear? Is that clear? Right, so non-oppressed are loosely arranged, so they have PS1, right? They have PS1 mainly, okay? So they will help in ATP as well as NADP formation. They have ATP synthase as well, right? So edges of grana and the complete stroma lamella will have it. Okay, so non-oppressed may mainly you will see PS1, oppressed will have PS2. Done? Okay, so this is important, okay? This location and position is important. So let's see the differences in PS1 and PS2. So PS1 is located at non-oppressed part of grana thylakoid and stroma lamella, right? Non-oppressed part. माने exposed surface. Okay, माने exposed surface. Reaction center P700, it is having the reducing nature, will reduce NADP plus, means it is very important for NADP formation. So there are two processes that you have to study, cyclic and non-cyclic photophosphorylation. Here you will find it in both. Okay, you will find it in both. Okay, but here PS2, you will find it in non-exposed part or the part which is tightly stacked. Okay, so reaction center P680, oxidizing nature, oxidizes H2O, very important point. So this one will only participate in non-cyclic photophosphorylation. So we also call it as Z scheme, if you remember, right? So we will study it here. So here you guys can see, right, the photosynthetic molecules. You'll see the primary acceptor here. So that's how everything is connected. Am I clear? That's how everything is connected. Am I clear? Am I clear? So let's study the light reaction. Let's study the Z scheme. Let's study that mechanism in detail. So if you guys are having any doubt here, do let me know. Do let me know if you guys are having any doubt. Sure? Sure? So it is one of the most important topics. Okay, it is genuinely, it is one of the most important topics. And once we are done with the light reaction, dark reaction is not at all a problem. It's going to be quite easy. So in next, I believe in 40 minutes, we can finish this part. Okay, yeah, chalo, theek hai, maximum 1 hour, otherwise in 40 minutes, we will finish it. So any doubts? Okay, so let's move to the next part here. So you guys can see this is the chloroplast trauma, right? This is the chloroplast trauma. So this is what you will see. They go thylakoid membrane, because we are going to talk about the light reaction. So firstly, I'll teach you some basics, then we'll go ahead. So this is the thylakoid membrane, and obviously within it, you will have lumen. Outside it, you will have stroma. We know it very well. These are the protein pigment molecules which are present here. Just you will see, because here the process will start from PS2. So PS2 to PS1, we have to pass the electrons, right? So it is again a type of electron transport chain only. We also discussed that in mitochondria, but it is also like that. Electron will pass from one pigment molecule, one protein to another. And when you will pass this, the energy is used that will help in the movement of protons, and that will ultimately help in the synthesis of ATP. Just if you will see here, PS2 is there, right? You will talk about the plastoquinones then. Yeah, quinones then I would say. Then you will talk about what? Right, we will talk about the pheophytin. Firstly, pheophytin is there, plastoquinones are there, cytochrome B6F complex, plastocyanin, PS1, rhodopsin. So all that things are arranged here. One more diagram to explain you this. Yeah, this is how the protein molecules are present. So you have PS2, you have different other carriers in between, right? And that's how they are going to move. It's so electrons, again and again, I'm repeating this part. Electrons will move from PS2 to PS1. This is the first thing here, right? This is the first thing here. In between, these are other electron carriers, right? Right? So look at this, the way things are present. So you have this PS2, firstly focus on the diagram, then I'll simplify it obviously. Focus on the PS2 here, right? Focus on the PS2 here. So now when you focus on this PS2, look at the position here. Inside it is having one oxygen evolving complex as well, right? Diagram is important, beta. You have oxygen evolving complex as well. Then you will talk about pheophytin, you will talk about plastoquinone, the plastoquinone pool that they are showing here, right? Then you will talk about the cytochrome B6F complex, you will talk about the plastocyanin. Look at the position, where are they present, right? You will talk about the PS1, right? You will talk about unknown electron acceptors. Finally, the ferrodoxin, this FNR mane ferrodoxin NADP reductase. What does it mean? Ferrodoxin NADP reductase. FD is ferrodoxin here. So yes, you have to remember them. You should know where are they placed. Just say plastocyanin is towards lumen. Hana, towards lumen side it is. Right? Firstly, just keep it in your mind. Ferrodoxin is towards stroma side. So firstly, keep it in your mind, then we will discuss it. Okay? Yes. Then we will discuss it. Quick. Done, quick. Just have a look of this diagram, then we will discuss it in detail. Oh. Yes, everyone. Done, beta? So diagram needs to be very, very clear here. Quick. Right? Diagram needs to be very clear here. देखो, overall, after listening to me, you will be able to understand it from the NCERT as well. But in general, I'm telling you, just we are going to talk about in the light reaction, we will be discussing non-cyclic photophosphorylation. And we have also discussed one more thing. The one is non-cyclic photophosphorylation. And another point that we have here is the cyclic photophosphorylation. So let me explain you this, right? And then let's understand that what exactly is happening. Ultimately, that will be the thing. Light will be absorbed, splitting of water, oxygen will be released, ATP and ADP will form. Ultimate thing is that, but actually what is happening, that's what I'm going to talk about. So when it comes to the non-cyclic photophosphorylation, if it is a non-cyclic process, in non-cyclic process, let's suppose there is A, you're starting from A, you're going to B, you're going to C, you're going to D, right? You're going to E, something like this. Now, if it is the cyclic process, if you are starting from A, you have to come back to A only. Means A to B, you are going B to C, C to D, D to E, E to back to A. That is the cyclic process. This is the non-cyclic photophosphorylation, non-cyclic process. Now, the next word is photophosphorylation. That what exactly is phosphorylation? Firstly, focus on the word phosphorylation. So phosphorylation, right? Addition of phosphate, like when it is ADP plus inorganic phosphate, it is giving you ATP. This is phosphorylation. But if I'm saying photo, mane photons, mane light. So in the presence of light, when there is the formation of ATP, in the presence of light, when there is the formation of ATP, that is the photophosphorylation. That is the photophosphorylation. Is that clear? Right? Now, three types of phosphorylation we will study. One is this. One is substrate level phosphorylation. One is this. Another is what? Another is substrate level phosphorylation. And one is oxidative phosphorylation. Are you getting my point? And one is what? One is oxidative One is oxidative phosphorylation. That's what you have. Now, when it is substrate level phosphorylation, like a particular substrate is giving its phosphate, yeah, helping in the formation of ADP to ATP. So this one we study in the respiration. We will study in glycolysis. We will study in Krebs cycle. Oxidative phosphorylation, mane this oxidation, mane this oxidation reduction, जैसे chemiosmotic hypothesis you discuss. There also you talk about the ATP formation. So that's how we relate everything. Okay, so three type of phosphorylation is there, and here you are talking about, because it is photosynthesis, we will talk about photophosphorylation, that in the presence of light, how that ATP will form. So this is actually beta Z scheme. This is actually beta Z scheme, right? That we have to discuss. Okay, Z scheme, zigzag it is, right? So substrate level phosphorylation is there, oxidative phosphorylation is there, that oxidation energy is ultimately helping there. So basically, that's what we have to discuss here, okay? Done, beta? Okay. Okay, redox. So now next is redox potential that we have to study. So I hope these basic terms are clear. So in non-cyclic photophosphorylation, we will talk about both. We will discuss PS2 as well as PS1. Third or fourth time I'm explaining that electron will go from PS2 to PS1 here. In cyclic photophosphorylation, you will only talk about PS1. In non-cyclic photophosphorylation, you will see ATP will form, you will see NADPH will form, you will see oxygen will release. But here what will you see? Here you will see only ATP forms, right? Only ATP forms, nothing else. Is that clear? Only ATP forms, nothing else. Is that clear? Is that clear? So this is what we have to understand here. Got it? Chali. So before starting this Z scheme, right, let's talk about the redox potential. Let's discuss that part and then I'll teach you what you have to remember, and then one by one we will focus on everything here. So what are we going to discuss? We'll be talking about the redox potential. So what do you know about the redox potential? Done? Okay, so what is the redox potential? Anybody? Anybody? Matlab, it is the tendency, है ना? It is the tendency of an atom or a molecule, to gain or to lose electrons. That is what this redox potential is, right? That you know that gaining of electron will come in what? Reduction. Losing is basically to get oxidized, yes or no? So if it gains electron means it will be reduced, है ना? If it is losing electron, mane it will be oxidized. So this is what redox potential is. Firstly, tell me, is that clear? Firstly, tell me, is that clear? Yes or no? So if there is positive redox potential, there is positive redox potential as well as negative. So if there is positive redox potential, mane the one, it is positive now. So it means the substance will, right here the substance will accept electrons very easily. Substance accept electrons easily. Matlab easily it is accepting the electron, right? It is easily accepting the electron. If it is accepting the electron, no doubt it is getting what? It is, because it is accepting electron, it is getting reduced, but it can accept electron. Like there is one entity here, this is another. This one is having more tendency to accept the electron. It is positive redox potential. Mane, it is a very strong oxidizing agent. Na? It simply means that it is a very strong oxidizing agent. It is a very strong oxidizing agent. Like it can oxidize the other compound and can accept its electrons. So same way you will have negative redox potential, right? Same way you will have negative redox potential. So here what is happening? Substance, right? This one is easily accepting the electron. So the one which is having negative redox potential will be losing the electrons. Yes or no? Because it is the opposite here. Yes or no? So it will be substance that will lose the electron easily, isn't it? Substance that donates electrons easily. So if it donates electron easily, then obviously what will you see? That it will be a strong reducing agent. It will be a strong reducing agent. Am I clear? It will be a strong reducing agent. Yes or no? Yes or no? Done? Okay. Any doubts? So ultimately, how the electrons will flow? So electrons flow from, right? Electrons flow from lower redox potential to, है ना, that मतलब that is understandable. So it will be from lower redox potential to higher redox potential. Lower redox potential to higher redox potential. Is that clear? So it is from lower redox potential to higher redox potential. That is how the things should be now. So this is what you have to remember. So electrons will move from lower redox potential to the higher redox potential, and this is the thing which will help in ATP or NADPH formation, right beta? So now understand this part, right? If electrons, because if something is having lower माने negative redox potential है ना, lower माने negative redox potential, yes or no? And so this one is positive redox potential. So this is higher redox potential. Is that clear? So that's how the electrons flow. That's how the electrons flow, right? Right? Yes or no? Okay? Yes or no? Okay, so no doubt if I'm saying lower to higher, it is not always means beta that from negative. Lower value always doesn't mean that it will be in the negative only. But in general, if I have to explain you in this, 1 second, sorry. Yes, my bad. Huh. So if I have to explain this, matlab in general, in context of this, if I have to explain, then obviously, let's suppose if one entity is having less redox potential, so matlab, so lesser to higher, this is how the electrons are moving. This is having a lower, it is having higher redox potential, it can accept electrons at a, it will accept electrons more easily. So obviously that's how the electrons should flow. So this is basically the downhill movement in this case. So do not confuse it with the active transport or the passive transport, because in passive transport you say that things are moving from their higher concentration to lower. So don't confuse it ki ma'am higher to lower redox potential, if it will go, then it is that that type of movement, that passive movement, yeah, down the gradient, yeah, fir I would say the along the concentration gradient. It is simple. If there is an entity which can accept electron easily, there is another entity in front of that, it is losing electron easily. Obviously, if A is the entity, which can easily lose electron in front of B, then obviously electron will move from A to B. So A is having what? Because it is donating electron easily, right? It is donating electron easily, so it is having negative redox potential, and then this one which is accepting it more easily, it will be having the positive redox potential. Is that clear? Is that clear? So that's how you have to relate it. Is that clear? Is that clear? So firstly tell me this, is it clear or not? Because in this electron transport pathway, that's what ultimately you have to study. Right, so basic rule is that the electrons will move from lower redox potential to the higher. So lower one will be more negative, right? Lower one will be more negative, and this one is more negative, obviously. So that is why it is able to give the electron to a substance with higher redox potential. Higher redox potential mane which will be having more positive redox potential. So if this is the flow, then it is the downhill. Like let's suppose if there is anything going against this thing, then it will be uphill. Just we study in the case of active transport, that let's suppose if something is moving from its lower concentration to higher, we call it as against the concentration gradient, and we call it as uphill. But here what's the case? Here you have to focus on the redox potential thing. In terms of redox potential, what is happening, right? What is happening here, that it is more negative, and this one is the more positive. This one is the lower redox potential, higher. So higher redox potential in front of this will obviously deserve more electrons. Firstly tell me this part clear, then I'll start the Z scheme. Quick guys, then I will start the Z scheme. Quick. Done? Chaliye, let's continue, guys. So next is the Z scheme that we have to discuss or we have to understand. Understand one thing, both are photosystems, both can absorb the light simultaneously, right? But I have to explain you something. So I am explaining you it in this particular way. This is PS2 towards the lumen side. Towards what? Towards the lumen side, you will see there is OEC, that is oxygen evolving complex, which is attached to it. Is that clear? There is oxygen evolving complex which is attached to it. Is that clear? Is that clear? Right? Guys, am I audible? Yes? Chalo, so let's understand that. Okay, so first of all, there is PS2. In the thylakoid lumen, we also have PS1, right? We also have PS1, right? And they both are absorbing light energy. It's not like that they are not absorbing. They both are absorbing light energy simultaneously. It's not like that first light will go to PS2, then light will go to PS1. No, that's not going to happen. Are you getting my point? That's not going to happen. Okay? Done? Are you getting my point? Are you getting my point? Yes, please. Are you getting my point? So that's something that you have to keep in your mind. This PS2 is there, light energy will fall on it, right? So oxygen evolving complex is also attached to it, right? Oxygen evolving complex is also attached to it. You people can see this very clearly. So that's how the things are. Now, in between, we are having other electron carriers as well. Right, just say you will see there will be there will be pheophytin, there will be plastoquinone, all the things are there, right? So now understand this. Just say from this part I will be starting. The electrons will move as per the redox potential. So let's start with this PS2. PS2 will, right? PS2 will absorb the light energy. You know that light harvesting complexes will also pass the energy to this reaction center, and from that reaction center, the light from that reaction centers, we know that ultimately the electron, it will emit, the electron will exude. Electron exude. So if electron will exude here, right, you know that what will happen. Matlab, if electron, it is ejecting electron, it is exuding electron, mane it is getting oxidized. This part clear? This part clear? Yes or no? So light energy of a particular wavelength will be absorbed by these reaction centers. So you know that it will get photo excited, it will exude an electron by gaining the energy, right? So it will get PS to get photo excited, right? And exude electron. Done, beta? Exude electron, and obviously they need a certain wavelength of light to do that. Yes or no? Yes or no? Yes or no? Done, beta? Done? So no doubt when you will see the diagrams, this is how we show now. This thing, this is showing that photo excited PS2. Who's going to accept that electron? When I explain it, just to make it clear, I write the pheophytin, but here just to make it clear to remember, then comes the pheophytin, electron carrier. Then comes what? Pheophytin, right? I'll discuss it as uphill or downhill. Then the electron will pass to plastoquinone, right? Plastoquinone, which we denote as PQ. Tell me, up to this part all clear that how the electrons are flowing? How the electrons are flowing? Is it clear or not? Is it clear or not? Then you have pheophytin, you have plastoquinone, that is PQ. You have plastoquinone, that is PQ. Is that clear? Sure? Sure? Quickly tell me. Quickly tell me. Yes, please. Done, beta? Now, this plastoquinone, right? I'll write it here. This plastoquinone, remember, it is not just an electron carrier, but also a proton acceptor. This is something that we also study in respiration in plants chapter during ETC. We also study that. So whenever you talk about the PQ, PQ will become PQH2. So this is plastoquinone, right? And this is plastohydroquinone. This is plastoquinone, then it is plastohydroquinone, which we also call as plastoquinol, right? Which we also call as plastoquinol. Is that clear? Which we also call as plastoquinol. Am I clear? Am I clear? So you have plastoquinone here, you have plastohydroquinone here, yeah, plastoquinol here. This part is clear. PQ, PQH2. That is what we have here. Here, no doubt, a Q cycle will operate. For sure, a Q cycle will definitely operate here, indeed. Right? We are not going to talk about it right now. So here, remember, ultimately where these things are located. Where these things are located. I'll discuss it very slowly so that you all can understand that this is happening in the thylakoid membrane. So you have pheophytin, you right, which is almost closely associated here. You have plastoquinone, right? So this plastoquinone is no doubt accepting the electron, but it will also accept the proton. It will become PQH2. Then it will pass its electron to whom? It will pass it to cytochrome B6F complex. And when it will pass the electrons here, H positive will be passed to the lumen. Just like ETC that we have to discuss in respiration. But what I want to tell you here, one by one I'm discussing all the steps, then I will write few points which you have to remember. One thing is, after accepting the light energy of suitable wavelength, it will get excited, it will eject its electron, that will be accepted by pheophytin, number one. Now if it has given the electron, it is oxidized. Now, if it has received the electron, it will be reduced. So overall in this process, you will see that ultimately what is happening? Oxidation, reduction, oxidation, reduction, oxidation, reduction. It is going on here. Oxidation, reduction, oxidation, reduction, oxidation, reduction. It is going on here. Is that clear? Is that clear? Is that clear? Yes or no? So oxidation, reduction, oxidation, reduction, oxidation reduction is going on here. If pheophytin is accepting the electron, it is getting reduced. This one is getting oxidized. When it will pass its electron to plastoquinone, it is getting reduced and this one is getting oxidized. So plastoquinone will become plastohydroquinone, right? Which is not just an electron acceptor, but also the proton carrier. So plastohydroquinone means what? That it is the reduced one. And obviously you also call it as plastoquinol. Quinol. Here a Q cycle operates. That is not what I'm discussing right now. So I'm telling you it will also accept the proton from stroma. And when it will pass its electron further, it will pass its electron further to cytochrome B6F complex. So while passing this electron here, it will pass this H positive that it has picked up from stroma into lumen, right? Into lumen. Then it will pass its electrons to plastocyanin. Right now I'm not marking it as per the positions. In general, I'm writing it. Now, why do we call it as a Z scheme? Because here you will see a kind of zigzag pattern when it comes to what? Their redox potential. Now, up to this part, the flow of electrons clear. The flow of electrons clear. Now I'll tell you, let's suppose if this PS2 is getting oxidized, it has passed its electron to pheophytin, it is getting oxidized. But we have to, for the continuation of this thing, we have to bring it back to the ground state. right? We cannot be in excited state always. We cannot be in excited state always. Yes or no? We can not be in excited state always. Yes or no? Yes or no? Yes or no? Yes or no? It is like that. It is like that. है ना? So this OEC will help. What is this OEC? Oxygen evolving complex. Beta, in this complex, what will happen? There is the photolysis of water. There is the photolysis of water that will occur here. Right? What will occur here? There is the photolysis of water. Is that clear? Is that clear? So this photolysis of water will occur here, beta. So photolysis means breakdown of water in the presence of light. So if here there will be 2H2O, this reaction is very important. So you will see 4H positive, 4 electron, and there is the evolution of oxygen. So what does it means, right? Please keep it in your mind. Done, beta? Keep it in your mind. So here, what you have to remember, it's manganese, calcium, and chloride, right? Manganese, calcium, and chloride. So these are the ions that are used here. Is that clear? Is that clear? So for the photolysis of water, for this particular reaction, what is happening? For this particular reaction, beta, these are the ions that are used. I have a trick for it. The trick is Man Ka College. That's how I remember. Man Ka College. That's how I remember. Right, beta? Beta, Shivansh Patel, what will you get? What do you want to know? What are you thinking? That you will type such kind of stuff, and what are you trying to show here? Right? You are if you're thinking that your such comments can demotivate me, or I'll end my lecture, or I'll feel embarrassed, I'm sorry, I'm not. Because people like you, they know just one thing, that how to type the rubbish things in the chat section, right? You have that sick mentality. You have that, I would say that it is showing that about your upbringings as well, right? I am in front of camera. Otherwise, I would have reverted you, and in your language, I would have answered this to you. Trust me on that. I am in front of camera and I have to maintain the integrity. If you have the guts now, then face some lady, say the same thing, and then you will see what kind of slap you are going to get. Trust me on that. Right? You are that type. You guys are having such mindset ki, yeah, theek hai, we cannot, do something good in our life. So what should we do? Okay, online platforms, yeah, teaching platforms, yeah, Instagram, let's do such kind of rubbish things. You're just satisfying your ego. You're sick. You are just showing your sick personality here, right? Sick mindset here. So trust me, if you really have the confidence, talk to a lady, right? Make sure you talk to a strong lady, ask the same thing, and then you will hear the voice of that slap, right? And trust me, after that, you won't be able to hear anything, right? So don't just do such kind of things in front of me at least, right? And trust me, I'm telling you this thing, I'm in front of camera, otherwise I would have answered you in your language. And beta, वो जवाब ना इंग्लिश में नहीं आता, वो बहुत गंदी पंजाबी में आता. घटिया लोग, घटिया के पेरेंट्स को क्या ही गाली दूं. वैसे लग तो यही रहा है घटिया पेरेंट्स की, बेटा घटिया औलाद हो आप. So now guys, look at it. OEC माने oxygen evolving complex, right? OEC माने oxygen evolving complex. Is that clear? So here there is the photolysis of water. So if I'm saying 2H2O, keep it in your mind. If you will go for 1H2O, then you will get half O2. If you will go for 2H2O, then you will get complete oxygen molecule. Is that clear? So it is 4H positive, 4 electrons, oxygen, and it is Man Ka College. That's what you have to remember, and just keep it in your mind, right? When these electrons, see, we have the electrons, we have the protons here. So these electrons, they will be given to this PS2, and it will come back to the ground state. Are you getting my point? It will come back to what? It will come back to the ground state. Is that clear? Is that clear? So if somebody will ask you that PS2, if it gets excited, right? How will we keep it to the ground state then, right? Obviously, we have to provide the electrons from what? From the photolysis of water. That's the answer here. Right? So if PS2 is excited, how will it come back to the ground state? After the photolysis of water, after the breakdown of water by oxygen evolving complex, which is having these ions which need that, and there is an enzyme water oxidase as well. So these protons, again, they will be in the lumen, and where that electrons will go? To this PS2. So pheophytin will accept the electron, will get reduced, will pass the electron to the plastoquinone, which will then get reduced. So this plastoquinone, it will also accept the H positive from the stroma. That's what you have to remember. But here, rest everything is better. Only this electron transport is uphill transport. Only this one. Please keep it in your mind. Right? Means when we are giving the electrons from PS2 to this primary electron acceptor, that is your pheophytin, then it is uphill transport. It means it's not like that pheophytin is having, the higher redox potential, right? It is having lower, but still it is getting the electrons. So it is the uphill transport. Only this thing is the uphill transport. Am I clear? Am I clear? Sure. Am I clear? Any doubts? Any doubts? Done, beta? Done? So only this is uphill, the rest all is downhill. Okay? Okay? So pheophytin is the primary electron acceptor here. So it is itself a chlorophyll derivative that lacks magnesium in the porphyrin ring, right? So it is what? It's a chlorophyll derivative that lacks Mg2 positive in porphyrin head, right? Porphyrin head. Am I clear? So that's what you have to remember. So only in this case you will see uphill transport, rest all will be the downhill transport. So up to this part all clear. Now cytochrome B6F is passing the electrons to plastocyanin. It is located towards lumen side. I hope you know that. It is located towards lumen side. Right? Now it will pass the electrons to whom? To PS1. But guys, I told you already, PS1 is also absorbing the light energy. PS1 is also ejecting the electron. It will pass it to some unknown electron acceptor A, and then finally it will pass to the ferrodoxin. Right? To which part it will pass? It will pass it to the ferrodoxin. And when you talk about this ferrodoxin, just a minute. So here, so PS1 will pass the electrons to what? To ferrodoxin FD. So with this FNR is located. So yes, you really need to know what is this FNR. So FD here, I'm writing it. It is ferrodoxin, right? It is ferrodoxin, and this FNR stands for ferrodoxin NADP reductase. It stands for ferrodoxin NADP reductase. Okay, so this is towards the stroma side, right? And now here in the stroma you have reduced NADP, right? It will get the electron, it will pick up the proton from the surrounding, it will become NADPH2. Am I clear? It will become NADPH2. So I want to make it clear that you should know that, see, when PS2 was excited, how will it come back to the ground state? It will accept the electron from photolysis of water. What about PS1? Who's providing it the electron? Plastocyanin. It is plastocyanin. So that is what, and that is how the flow is. So if I have to write the points here, I'll write PS2 passes electron to PS1, number one. Number two, PS2 comes to ground state by photolysis of water. By what? By photolysis of water. Is that clear? Number 3, plastoquinone is not just an electron carrier, but also a hydrogen acceptor. Then PS1 comes to ground state by accepting electron from plastocyanin. So cyanin, blue color, it contains the copper, right? Cyanin word. So from plastocyanin, which ultimately is coming from PS2 rather, which ultimately is coming from PS2. Now, the 5th point is plastocyanin located towards lumen side. Done. Plastocyanin is located towards lumen side. Done. Anything extra? FD or this FNR, right? It helps in NADH. The NADP plus is not reduced. This is oxidized. By mistake, I think I said reduced here. Nahi, this is oxidized. So it will be reduced. Is that clear? It will be reduced, right? So helps in NADP reduction. Okay? That's his name. So ultimately in this process, what is happening? Right? Ultimately what is happening? We are basically trying to maintain a proton gradient. How come proton gradient is maintained? Simple. You have PS2 here. When it is the OEC, right? That is here also. There is the splitting of water. So that splitting of water is also making the protons, no. Where will they go? Lumen. Your plastoquinone, right? It will also pass the proton, right? When your NADP, oxidized one, when it is getting reduced, from where it is picking up the proton? From stroma. So during this process, ultimately what is happening, right? Inside, H positive increase. If H positive increase, we know that there will be low pH, right? And in stroma, what will happen? In stroma, you will see H positive decrease. So if H positive decrease, what will happen? The pH will increase, right? The pH will increase. Am I clear? So this gradient is important because it will help us to make the ATP from ATP synthase. Firstly, tell me this part is clear. This part is clear. So as per electrochemical, this redox potential, I would say the things are going. I'll show you from the pictures as well. This is the oxidized one, then pheophytin, which is referred to as primary acceptor beta. Then you have the plastoquinone, which will become plastohydroquinone, then cytochrome B6F complex, plastocyanin, right? Their positions should be very clear. So then unknown electron acceptor AZ, and then finally ferredoxin, and finally to NADP. Just have a look. Just have a look. If there is any doubt, do let me know, right? Do let me know. Yes, they can even ask you just ferredoxin, which one is containing ferredoxin, iron sulfur it is having. So where do you find that also? So such type of questions can also come. So you tell me if it is clear to you or not. You tell me if it is clear to you or not. Understood? So next is, you can see that, right? You can see it here. So you have P680, like it is getting oxidized, pheophytin is there. Then you will see the quinone part also, right? Then finally you will see, obviously we are not going in that detail. This is true. You have quinone A, B, plastoquinone, that will become plastohydroquinone. But whatever I have taught you, just stick to that. That will be more than sufficient to clear this clear the questions on the basis of this topic. So then comes the cytochrome B6 cytochrome F complex, then to plastocyanin, then to P700, then photosystem 1. So that will pass through this unknown electron acceptors A, and finally it will go to the ferrodoxin, which is towards again the stroma side, and finally to NADP. So I hope this part is clear, right? I have made it very clear to you. And even not just this, that's why I started with, right? That's why I started with what? I started with the, right? This part and the redox potential part. So you tell me if there is any doubt here. Quick, quick. Done, beta? Okay, so if you will see that, ma'am, here we cannot see the pheophytin location. Actually, pheophytin is somewhere around that reaction center. Like it is closely associated with that reaction center wala part of this photosystem too. Is that clear? Is that clear? So it will pick up the electron, it will pick up that electrons and then will pass to the plastoquinone. Understood? Any doubts here? Any doubts here? And trust me, if it is clear to you, then you will very easily, you people will understand the next portion, that is the what do we say, etc in the Z scheme. That is also very easy. Is that clear? Is that clear? Right? So plastoquinone is there, and remember it is lipid soluble. Rest everything you know about it, but yeah, okay, you can also mention that it is lipid soluble. Am I clear? Yes or no? Am I clear? Okay. Done. So you should know this pathway, okay? So it accept 2 electron, 2 protons, it becomes plastoquinol. It is also the hydrogen carrier. It will pass the electrons to cytochrome B6F complex, okay? Any doubts? Any doubts? Done, beta? So this is plastocyanin. Always remember it is copper containing, right? Electron carrier. So this one is what? It is copper containing electron carrier. Is that clear? Okay. Any doubts? So yes, this is also important. This is also important. I wish you are clear with it. So 2H2O will give you 1O2. If it is only H2O, it will just give you half O2. So let's revise it from the notes as well. So see this, yes, these are special quinones that come in between. Then there is this pool, but okay, fine, I am not touching that. So cytochrome B6F complex is there. So it is also iron sulfur protein, and that ferrodoxin, before that also iron sulfur protein is there. So do remember that. Okay, do remember that. So we'll share the notes in the Telegram group as well, and I have mentioned some few, quickly asked points as well. So let's get started. Let's start with the light reaction. Okay, let's start with what? Let's start with the light reaction. Am I clear? So begins as soon as the light falls on green plants, right? Matlab, we have already discussed it. Now we will just revise it from these points. Then I will teach you the cyclic photophosphorylation and finally the dark reaction part. Is that clear? So you can see pigments in PS1 and PS2, they absorb the specific wavelength. They transfer that energy to reaction center, which will get excited, which will emit high energy electrons, which will be accepted by the nearby electron carriers. So non-cyclic photophosphorylation will include both, cyclic will include one, already discussed. So firstly, light will excite PS2, energy will be absorbed by reaction center, Pe680 will become photooxidized, right? It will release electrons, it will gain the energy, and electrons will be accepted by pheophytin, then will go to the quinone discussed. So this P680, because it is oxidized, it got that plus charge, so it will again come back to the ground state by photolysis of water. So lost electrons of beta, your P680, is replaced from water molecule. A very important point. Is that clear? So this is photooxidation. Your photolysis occurs in oxygen evolving complex, right? So it will do splitting of water. Water oxidase is the enzyme, and this is Manka College. Again, another important point, told you already. So here you guys can see the reaction as well. So evolution of one molecule of oxygen requires 2 molecules of water. So these 4 protons and 4 electrons are there. Electrons will be accepted by your photosystem too. And what about protons? They will be into the thylakoid lumen. They will increase H positive concentration inside and will decrease the pH. Important it is. Now how that electron will flow? So PS2 excited, the electron will be captured by quinone, but firstly it will pass it to pheophytin, then to plastoquinone, then to this, right, which is a copper containing pigment. Understood? Right? So the oxidation reduction is going on. So cytochrome will pass the electron. So it is the PAC which will deliver it to the photosystem 1. So you can see PS1 is also absorbing. It is also transferring it to the reaction center. It is also getting excited. So it will pass it to primary acceptor A. Some unknown electron acceptor is there. And ultimately, right, they'll pass to ferrodoxin. So this ferrodoxin is stromos. It is iron sulfur protein. That is also important. Okay? So it is located to outer surface. I told you locations are also very important. So reduced ferrodoxin will donate the electrons to NADP, nicotinamide adenine dinucleotide phosphate, which is oxidized. So with the help of enzyme FNR, ferrodoxin NADP reductase. So if this part is clear to you, chemiosmotic hypothesis will be easily clear. Okay, so in the case of plastoquinone as well, remember here it is the mobile electron carrier. Mobile electron carrier mane it is movable. So pick 2 electron from PS2 by a pheophytin, also 2 protons from stroma. So it forms plastoquinol ya plastohydroquinone. So it will deliver its 2 electrons to cytochrome. But again, beta, here also the Q cycle is there, which we also discuss in ETC. 1 electron will pass to plastocyanin here via the cytochrome F, then to PS1, and other electron will come back through cytochrome B6 to this another PQ molecule in the QP. Because here also in 2 cycles, because we have to make the this particular carrier again, right? So again it needs basically in that Q pool 1 electron, or like it, you cannot say that immediately it will give its electron, will get reduced or will get I would say will get oxidized. The next one will get reduced. So here basically in steps the Q cycle will be there. So yes, here also semiquinone will form, then that will, finally the PQ will be there. So by transferring the electron, PQH2 will release 2H into the thylakoid lumen. That will help in the proton gradient. So recycling, this recycling is basically done by Q cycle. So semiquinone, PQ negative is again reduced. When another PQH2 comes in, it completes the cycle. Is that clear? Done. So pheophytin, told you already, right? This excites an electron from ground state to high energy. The excited electron is transferred to primary acceptor pheophytin. But here, low energy to high energy, matlab electrons will be excited like that, but here this is the uphill transport. Rest everything is downhill, I told you, but here you will talk about the uphill transport. So when PS2 is giving the electron to pheophytin, the primary electron acceptor, so it is uphill transport. It means pheophytin is not having that positive redox potential, or this one is negative, right? So that it is getting the electron. No, it is the vice versa here. PS2 reaction center has more negative potential, yeah, high reducing power, right? Very important it is. This one is having more negative potential, that is high reducing power, right? And pheophytin has more positive potential, that is less reducing or more oxidizing power. So under normal condition, we know that the electron would not spontaneously passes from this to this, but light energy will give the electron an energy boost. That's why it is passing against its natural gradient. So that's why it is uphill, because it is against its natural gradient. Any doubts? Any doubts? Done, beta? Done? Sure. So next is the cyclic photophosphorylation. Already told you what is the context here. We'll discuss it as well, right? So in the cyclic photophosphorylation, only ATP will form. Why? Because your PS1 is not associated with the oxygen evolution, right? So PS, and here, because where will this cyclic photophosphorylation? Stroma lamella. Stroma lamella. So this is very important. It lacks PS2 as well as FNR. Understood, right? So ferredoxin reducing substance, matlab PS1 will pass the electrons to ferredoxin reducing substance, then that will pass the electrons to ferredoxin, then it will come back to cytochrome B6, then to cytochrome F, then to plastocyanin, and then back to the PS1. Right? So here in this only PS1 works. Important point. Where is it present? Stroma lamella. Second important point. So electrons ejected from the reaction center of PS1 come back to this third important point. So no doubt at every step, oxidation reduction of electron carrier occur, which will release the energy, and this energy is actually used for creating the proton gradient for ATP formation. So ATP is formed here. So in stroma lamella, see, that were the bullet points. That are the bullet points. It lacks PS2 as well as FNR. Is that clear? So there is no FNR, no NADPH formation. So here only you will see the light beyond 680 nanometer is available for the excitation. Am I clear? Am I clear? Done, beta? So in cyclic ETS, right? In cyclic ETS, no oxygen evolution occurs because there is no photolysis of water, only ATP is formed, right? So keep it in your mind. So here also one more diagram, PS1, then to FRS. What is this FRS? I'm writing it here, beta. FRS stands for ferredoxin NADP reductase. Ferredoxin reducing substance, my bad. Ferredoxin reducing substance. Keep it in your mind. FD here means ferredoxin. So yes, energy is provided for the proton gradient, but not like this. So cytochrome B6, then plastoquinone, plastohydroquinone, back to cytochrome F, back to plastocyanin, okay? Okay, so this is the complete process which is written here. Next is the chemi-osmotic hypothesis given by Peter Michel. If things are clear here in this particular topic, right, this particular topic, then right, we will even in respiration you will see, you will find the things very easy. Okay, okay, so this diagram you already know that. So obviously inside, because there is more H positive, so there is high electrochemical potential gradient. Outside there is low electrochemical potential gradient. Water breakdown is taking place here. Half O2, half O2 is there. Keep it in your mind. So plastoquinone, and yes, plastoquinone is the mobile electron carrier here. This one is actually mobile. So it is also picking up H positive. It is making plastohydroquinone, then cytochrome B6. So when it is passing the electron here, H positive here, H positive from water, right? So plastocyanin and this. So this is also picking H positive. So decreasing the H positive in stroma. So already I have explained it. So this is your what? This is your ATP synthase, right? And here, you remember ATP synthase, oxysome. So here you are calling it as CF1, CF not CF1 particle, okay? Right? CF not CF1 particle. So I will see it like this. So this part is CF naught. This part is CF1. This is going to be base. Right, this is going to be head plus stalk. Head plus stalk. Done, beta. So this CF naught is traversing, right? It is in the traversing, it is in the membrane, within the membrane. And where will you get the CF1? In stroma. Where will you find it, beta? You'll see it in stroma. Am I clear? Am I clear? Done? Okay, sure. So what are the requirements, right? What are the, trust me, now you will find it easy if you understand what light reaction is, and next dark reaction is very, very easy. Overall, it is very easy, right? Chalo. So let's focus here. What are the requirements of chemiosmosis? You need a membrane, thylakoid membrane, ETS carrier that will pump or move H positive, high H positive inside lumen, that is proton gradient, which is a very important point, which is a repeated PYQ as well, right? So ATP synthase, which is CF not CF1 complex. This is another important point. Am I clear? So ATP formed by breakdown of proton gradient. Please do solve the PYQs. You will find this statement as such. ATP is formed by breakdown of proton gradient. So NADPH formed by reduction of NADP. So both are used in the Calvin cycle. That's why we say the products of light reaction are important for the dark reaction. So that will help in CO2 fixation and sugar synthesis. Am I clear? So this will explain how ATP is formed. Just like in respiration, ATP synthesis is coupled to a proton gradient. Here also, beta, it will happen. There you talk about the mitochondria. Here you will talk about the thylakoid membrane. So remember, in when we will compare it with the respiration, जैसे mitochondria में, in the case of mitochondria, if you remember, right, that ultimately that proton gradient, right, the more protons will be developed here in intermembrane space. In this intermembrane space, you will have proton gradient. Right? You will have proton gradient. Now, but here in the chloroplast, more H positive will be collected here in the lumen. Here in intermembrane space, more H positive will be accumulated during this electron transport state. Here in thylakoid lumen, right? In thylakoid lumen, more H positive will be accumulated during this process. Any doubts? Any doubts? Done, beta? Okay, chaliye. So the next part here is, right, it is the NCERT that I'm going to simplify here, that the splitting of water take place on the inner side of the membrane. So beta, if concept is clear, trust me, there is no need to cram here. This part is literally very easy. So protons or hydrogen ions that will produce will be accumulated within the lumen. So as electron moves through photosystems, photons are transported across the, right, these protons, not photons, the protons are transported across the membrane, because the primary acceptor of electron, which is located towards the outer side of the membrane, will transfer its electron not to an electron carrier but to a hydrogen carrier. So here beta, they are actually talking about your plastoquinone. Please keep it on your mind, right? So it will also remove a proton from the stroma, and then it will pass on the electron carrier on the inner side of the, pass to the next electron carrier, and then it will pass the proton to the inner side of the membrane. So NADP reductase enzyme is located on the stroma side. Another important point, right? Another important point it is that the NADP reductase enzyme is located on the stroma side of the membrane. So along with electrons that come from the acceptor of PS1, protons are necessary for the reduction of NADP to NADPH, right? So they are also removed from the stroma. Is that clear? So hence within the chloroplast, protons in the stroma decreases, in the lumen they will increase. So this is the proton gradient. So lumen mein beta low pH, acidic, stroma mein high pH. So this proton motive force will help to make the ATP with the help of that ATP synthase. So CF naught beta, it is embedded in thylakoid membrane. It is forming a transmembrane channel. So this is because when protons, they are passing through a channel, they are obviously they cannot cross the membrane directly, but they are passing as per their concentration gradient. That's why the name here is chemi-osmotic, right? So osmosis, you know that the things will pass from there. The solvent will pass from its high concentration to the low concentration. So it is chemi-osmotic, right? So obviously these H positive is creating the gradient here. So because no doubt the protons will pass from the CF naught, from this ATP synthase, right? So it is passing through a channel. So that's why the word here is facilitated diffusion of protons, right? That's why the word here is facilitated diffusion of the protons. So CF1 protrudes on the stroma side. Conformational changes will occur here, right? So protons, when it will flow, the energy will be provided for ATP synthesis. So that's how the protons will move, and the release energy will drives ATP formation by CF1. Am I clear? Sure? Sure. So this energy is used to pump the protons to create a gradient, and this gradient is helping to make the ATP. Okay? Okay. So along with the NADPH produced by the movement of electrons, ATP will also be used immediately in the biosynthetic reactions, that is in the dark reaction. Is that clear? Biosynthetic reaction in the dark reaction. So that's the next point that we have to discuss. Next B, and it's quite easy. So up to this part all clear? Yes, up to this part all clear. So beta, the mechanism is literally important, and if concepts are clear, you are not going to find it difficult at all. So do let me know. Done? Okay. Done. So next is the biosynthetic phase, which we are calling as phase of photosynthesis, dark reaction, chemical dark reaction, light independent reaction, even the Blackman reaction, even the carbon reaction, right? Because here the CO2 is getting reduced. So it is also the carbon reaction here. Glucose will form basically. So it doesn't directly depend on light, but you know that if there is no ATP, no NADPH, this will not occur. Okay, so when light is not available, this biosynthetic process, it continues for some time until, you know, they finish that ATP and NADPH, right, and then stops. So if light becomes available, the process starts. So it is dark word is the misnomer term. It doesn't mean that it is working in a dark condition. It's just that it doesn't need the light directly. And where will it occur? Stroma of the chloroplast, a very important point. Understood? A very important point. So here, beta, we will talk about C3 cycle, Calvin cycle, right? It is the Calvin cycle. C4 pathway is there, Hatch and Slack pathway on the name of their discoverer. And we will also talk about the CAM pathway, that is crassulacean acid metabolism. So this is something that you will understand in the case of what? This is something that you will understand in the case of succulents. Okay, so we will start with the C3 cycle, which is also the Calvin-Benson experiment, which is also what? Which is the Calvin-Benson experiment. We will finish this part and then you will have a break, and then after that, right, then you will have a break, and after that, what will we start? What will we start? After that, we will start respiration, the last chapter. Okay? Okay? Sure? Done? So, biosynthetic phase of the photosynthesis, also called the dark reaction, the chemical dark reaction, light independent reaction, carbon reaction, told you already. Acha, this is done. Repeated slide. So, Calvin experiment, let's talk about that. So, it is Calvin-Benson experiment also, and we also call it as C3 cycle. Why C3 cycle? Because the first stable product, here is first stable product here is a 3-carbon containing compound. What's that? It's a 3-carbon containing compound. Am I clear? What's that, beta? It's a 3-carbon containing compound. Is that clear? And what is it? It is phospho, 3-phosphoglycerate, or you can write it as 3-PGA. That's why it is the C3 cycle, because it is having the 3-carbon. It is the first stable product is a 3-carbon containing compound. So it's 3-PGA, right? So it is, sorry, one second. So that's what you have to remember. Just allow me 1 minute. Okay. So next point here is, so do not confuse PGA and 3-PGA or PGAL or G3P. They are different. Why? Because PGA, you are talking about phosphoglycerate or glyceric acid. So here the functional group is a carboxylic acid. Okay. And in the case of this PGAL, PGAL stands for phosphoglyceraldehyde. We discuss it in glycolysis. So it is phosphoglyceraldehyde. So here the functional group is, right, phosphoglyceraldehyde, or you can call it as glyceraldehyde 3-phosphate, right, glyceraldehyde 3-phosphate. So here the functional group is aldehyde. Okay, Here the functional group is aldehyde, and in the case of 3-PGA, the functional group is phosphoglyceric acid. So why it is a C3 cycle? Because the first stable product is a 3-carbon containing compound. In between, yes, we do have unstable product, but when it comes to the stable product, it is 3-PGA, okay? So in the experiment, they took the green algae, that is chlorella. Obviously, it is a unicellular green algae, right? It is easy to culture, right? So here they use the radioactive carbon isotope. Okay, why? Because they need to check that how that carbon dioxide is getting fixed, right? Like what are the compounds they are making. So if they will make the if they'll use the radioactive substance, then obviously they can just trace the presence of that radioactive substance in that particular algae. So radioactive carbon isotope 14C in the form of 14CO2. So radioautography to trace this, to track this rather, labeled carbon in the intermediate. So they checked it that, okay, fine, when CO2 entered, what CO2 has formed, next compound, next compound, next compound, like this. Okay. And why do they use chlorella that much for such experiments? Because as I said, it is unicellular, it is easy to culture, right? It is easy to culture, it is photosynthetic as well. Okay, so what they did first, so algae were eliminated, it was given this radioactive carbon so that carbon fixation could occur. Then the radioactive carbon quickly got incorporated into the compounds of this pathway, right? At different time intervals, in seconds, they were killing the algae, right? They were stopping their metabolism, and by drooping them into boiling alcohol. So basically, after every step, they were putting that algae in the boiling alcohol so that the cells can die, so that the metabolism can stop, and they were just checking out that compounds, that which compounds were made and in which particular which pathway was there and what is the sequence, everything they checked. So then chromatography and autoradiography. So compounds, they were separated by 2D paper chromatography. So a photographic plate was placed on the chromatogram. Dark spot showed where the radioactive carbon was present. That's how they traced it. So now from this particular cycle, from this experiment, they got the first compound that got labeled was 3-phosphoglyceric acid or 3-phosphoglycerate, 3-carbon containing compound. Plus, over time, other compounds like triose phosphate, glucose, fructose, ribulose bisphosphate, they were labeled. So it proved that CO2 first enters in a C3 compound. So that's why it is a C3 pathway. Okay. So they have provided the complete oxygen pathway. So this work even earned Melvin Calvin the Nobel Prize in Chemistry in 1961. Okay. 1961. So let's discuss the next point. So let's discuss the main points here. So we have started with the C3 cycle. So it's Calvin cycle or Calvin-Benson cycle. Done. Where is it occurring? The site is stroma. So if it is a cyclic process, always remember, whenever you talk about the cyclic process, if we start from A, we have to come back to A if we want to continue that process, okay? So replenishment of this compound is very important if we really want to continue the process. So here what is happening? What you have to remember that who's going to accept the CO2? So carbon dioxide acceptor. Now, initially they were thinking that if first stable compound is a 3-carbon containing compound and it is CO2, so initially, some other guess were there. Maybe that's a 2-carbon containing, that it's a 2-carbon containing compound which is accepting the CO2. But actually that's not the case. That's not the CO2. That's not the point here. So CO2 acceptor is a 5-carbon containing compound. Actually here, a CO2 acceptor is a 5-carbon containing compound. Are you getting it? Yes, everybody. So what is the name of that compound? They can ask you this question. It is ribulose 1,5-bisphosphate, right? It is ribulose 1,5-bisphosphate. It is the first, it is actually the CO2 acceptor here in the case of your what? In the case of your C3 cycle. So it's a 5-carbon containing compound. So what's the word? It is ribulose. Ribulose. Are you getting my point? What's that? It is ribulose. So it is ribulose 1,5-bisphosphate. Right? Ul. So understand this thing here. If at the place of ribulose you are going to write ribose, that is incorrect. Why is it so? Let me explain you. See, both are 5 carbon containing compound. Both are monosaccharides indeed. But ribose is an aldose. It means here the functional group is an aldehyde. Are you getting my point? It is an aldose. Functional group is an aldehyde. Are you getting my point? This one is a ketose. Functional group is a ketone here. That's the difference. So functional group is a ketone. It's a subterminal group. So in the nomenclature, this ul is written now. This is what this ul is signifying. This is what this ul is signifying. Is that clear? Sure? Done. So this is a 5 carbon containing compound. So this RuBP plus you have the carbon dioxide here, right beta? So what are you getting here? You are actually getting 3PGA, right? Because total it will become C. So here which enzyme is used? A very important enzyme, that is Rubisco. So it is basically ribulose bisphosphate, ribulose bisphosphate carboxylase oxygenase, ribulose bisphosphate carboxylase oxygenase. That is what you have here. Am I clear? So this is ribulose bisphosphate carboxylase oxygenase. Beta, it's the most abundant protein of biosphere. What's that? It's the most abundant protein of biosphere. Is that clear? It's the most abundant protein of biosphere. That's what you have to remember here, right? Right? This is what you have to remember here. So this enzyme, it's very important because it can do both carboxylation, that is addition of CO2, and this enzyme can also do what? Oxygenation, right? So no doubt it will go for the carboxylation first, right? That is the addition of CO2 first. But if let's suppose CO2 is less, it can even do the oxygenation. And in that case, this Rubisco will result in C2 cycle, that is photorespiration. That is a wasteful process for the plant. So this is important. Most abundant enzyme, right, can do both carboxylation as well as oxygenation. So kindly underline this point. They are important, right? So it is the most abundant protein of biosphere. So 5 carbon containing compound accepting 1 CO2, you are getting 3 PGA, which can do both. Am I clear here? Am I clear here? Yes, please. Done? Done? Sure? Any doubts? So this is how it works. So let's talk about the cycle, though there are many, other details also, but stick to the NCERT for this part. So the very first step here is, what? The very first step here is the carboxylation, which means addition of CO2. Addition of CO2. So carbon dioxide along with water. So this is a 5 carbon acceptor here. This is the first CO2 acceptor here, right? And this is 3 PGA, right beta? 2 molecules rather of 3 PGA. So this is the first stable product here. First stable product here. You really need to know that. Okay? You really need to know that. Are you getting my point? Are you getting my point? So then 3-phosphoglycerate is making triose phosphate. The second step here in the C4 cycle is reduction. What is it? Reduction. So beta here 2 ATP, not 1, 2 ATP, 2 NADPH are used, right? And this one, which are oxidized, they will again be reduced. So that is how the continuation of process is maintained. So in this step, there will be 2 ATP, 2 NADPH. So triose phosphate will form the sucrose here, right? You know that even the phloem transport sucrose. Sucrose is an invert sugar, right? It's a disaccharide. It's a disaccharide. Even in the phloem, this is which will be translocated. Okay, so then the regeneration where 1 ATP is used. So now what you have to conclude here, I need to make a glucose. I need to make a glucose. What's the formula of glucose? C6H12O6. That's the formula of glucose, yes or no? Yes or no? Then C6, 6 carbon. So 1 CO2 will not give you this. You need 6 carbon dioxide, right? So 6 CO2 are reduced. Please keep it in your mind to form 1 glucose. So if 6 carbon dioxide are reduced to form 1 glucose, if what? If 6 carbon dioxide are reduced to form 1 glucose molecule, 1 glucose molecule, right? If 6 CO2 are reduced to form 1 glucose molecule, so it means 6 turns of, right? It means 6 turns of C3 cycle are required for production of 1 glucose molecule, right? For production of 1 glucose molecule. Am I clear? For production of 1 glucose molecule. So in each turn, what do you have? Each turn may you are using 3 ATP, 2 NADPH, 2. So for 6 turns, that is for 1 glucose. For 1 glucose, how much you need? You need to multiply it with 6, sorry. So 18 ATP, 12 NADPH are used here. Is that clear? For 1 glucose molecule. So this is important, rather this is very important to remember. Okay, this is very important to remember. Is that clear? Is that clear? This is very, very important to remember, right? This information is damn important. Okay, for 1 turn. Now, the next thing is, let's suppose if the question is, now, what kind of questions can come in paper? It can be how many, in simple words, firstly I'm telling you, how many turns of C3 cycle produce 1 glucose? You should know the answer, right? How many ATPs are used in 3 turns of Calvin cycle in reduction step? So beta, they are talking about the reduction step. So answer will be 2 ATP in each reduction step. So it will be 3 into 2. That is 6 ATP. That's how you have to check. That's how you have to check. Now, how many ATPs are required for synthesizing 6 glucose molecules in C3 cycle? So now let's suppose if somebody is asking me this question, then what can I say? Matlab, how many ATPs? For what? For 6 glucose molecule. For 1 glucose molecule, you know that. For 1 glucose molecule, you know that. Right? It is going to be 18 ATP. Right? So for 6, it should be 6 into 18. है ना? नहीं, नहीं, नहीं. What will be? What should be the answer? For 1 glucose molecules, देखो, 1 glucose molecule is giving you 18 ATP. How come 18? Because 6 turns of this. So if you have to make 6 glucose, if you have to make 6 glucose molecule, so 1 glucose molecule for 6 turns of, this Calvin cycle. So for 6 glucose molecule, it will be, for 1, if it is 6, 6, 6 are 36, matlab 36 turns of, matlab in both the ways you can calculate is turns of Calvin cycle. So per turn 3 ATP, right? So 36 into 3, that is also a way, right? That is also a way. Okay, so in both the ways you guys can calculate it, in both the ways, okay? Okay. Done. So in both the ways you can calculate it. So be very clear when you answer such kind of questions, right? So do read it well. So RUBP, you know that it is a very, very, very important topic here, right? Ribisco, ribulose, bisphosphate, carboxylase, oxygenase, right? Having dual activity. Okay, then PGA is phosphorylated using 2 ATP molecule, then reduced using 2 NADPH. So 18 ATP and 12 NADPH in totality. So if there will be no regeneration step, how that cycle will continue. So these tables, they should be very clear to you. So Ribisco, you know that. Where will we find it? Stroma. It's the main enzyme here, right, beta? It's a very important enzyme here. Most abundant enzyme on earth here, most abundant protein on biosphere. And if somebody asks you, most abundant protein of animal world, right? Most abundant protein of animal world, then you will call it as collagen, right? Then it is the collagen. That's again a PYQ. And collagen is also the most abundant protein of extracellular matrix. That's what you have to keep in your mind, okay? Okay, so act as a primary gateway for inorganic carbon to enter the biosphere, determines the rate of photosynthesis. So carboxylase activity will fix CO2, but oxygenase activity will only be occur when high CO2 but low, high oxygen but low CO2 is there, right? So then Ribisco will go for the oxygenation activity, but this can result in a wasteful process, that is C2 cycle or photorespiration. Am I clear? Am I clear? So next is the beta, next is the C4 cycle that we have to understand, right? And then we will further discuss here, okay? So here, what is important to know? We will discuss the Krenz anatomy. So Krenz here mane read. Okay, so that is what we have to discuss here. Ready? Next is C4 cycle. Any doubt so far? Yes, please. Any doubt so far? Quick. Guys, quick. Sure. Chaliye. So next is the Hatch and Slack pathway, which is also known as dicarboxylic acid cycle. So it is on the name of what? Their discoverer, Hatch and Slack. And why do you call it as C4 pathway? Because here the stable product, first stable product is what? The first stable product here is a 4 carbon containing compound, right? 4 carbon containing compound is the first stable product here. So that is why we are calling it as this. So it is OAA, which means oxaloacetate. Oxaloacetate, that's the first, right? That is what you have here. Okay, that's a first stable compound here. Okay, so it will, where do you see the cycle? Dekho, one thing is clear. In all the plants, you will see C3 cycle, but C4 cycle is not there in all the plants. This is C4 pathway is a carbon fixation pathway. It is present in those plants which are living in hot, dry, and high temperature conditions, right? So it is an adaptation there, right? Those who are living in the hot, dry, and the high, right, high light environment conditions, they will be having this. So Hatch and Slack in 1966 they discovered it. So these are plants are the C4 plants. So the difference in C3 C4 plants is also very important. So because the first stable product of carbon dioxide fixation is a 4 carbon compound here, so that's why we discuss this. Is that clear? So why C4 pathway evolved? I told you, when high temperature and light, this enzyme ribisco, it can bind with the oxygen. We already know that it is having the oxygenase activity as well. So when the temperature is high, CO2 is also less, temperature is high, so it will bind with oxygen instead of CO2. So photorespiration, which I'm also calling as C2 cycle, occurs here. So this process is actually a wasteful process, beta. It is not at all helping this plant. Rather, the carbon dioxide which they have assimilated, it will utilize there, right? Which they have fixed, it will be utilized there, which will be of no use. So no ATP formation, nothing at all. So this is waste for a plant, right? So C4 plants, because they are already living in high temperature areas, right? The conditions are like that there. So they have evolved so that they can minimize the photorespiration by separating carbon dioxide fixation and the Calvin cycle into different. So here, what will you discuss? You'll talk about the Krebs anatomy. Done? Done. So it is the Krebs anatomy that you have to understand first. So it's a special type of anatomy that you will see in such C4 plants, right? So before this, look at this diagram here. Normally, C3 cycle will be in all the plants. So you have C3 plants, just when you look at its cross section, right? When you look at this mesophyll, right? Here you know that vascular bundles are there, which are surrounded by the bundle sheath cells, right? Which are surrounded by what? Which are surrounded by the bundle sheath cells. Can you see that? Which are surrounded by bundle sheath cells. Now here, you know that mesophyll cells, they have a chloroplast. Now when I talk about the C4 plants, they have their chloroplast is dimorphic. Two types of chloroplast is there, right? Dimorphic chloroplast. Large sized chloroplast you will see in the bundle sheath cells. Yes, bundle sheath cells which are having thick impervious walls, right? And here, small sized chloroplast you will see in the mesophyll cells. Now, how will it help here? How will it function here? These bundle sheath cells, as I told you, very thick wall, impervious to gaseous exchange. Here you will see in the bundle sheath cell, there will be C3 cycle. In the mesophyll cells, there will be C4 cycle. And moreover, we will talk about one more enzyme here, that is phosphoenol pyruvate carboxylase, PEPcase. PEPcase, that's what we have to discuss here. We will talk about the PEPcase here, okay? So chloroplast may dimorphic, large or the small chloroplast. So bundle sheath cells, mesophyll cells, C3 cycle in bundle sheath cells, C4 cycle in the mesophyll cells, and they have the enzymes PEPcase. Is that clear? So how are they arranged? So you can just see the diagram. Jaise ye dekho. Look at it. So this arrangement, jaise these are bundle sheath cells here. Around that, look at the mesophyll cells arrangement. So it is just like a wreath. So what exactly is the wreath? Firstly, see the diagram, then I'll tell you. This is a wreath. This is what we call as wreath. So Krenz word is from the wreath itself. There's a, here you have vascular bundle, bundle sheath cells. So surrounding me, you have the cells arranged like that, right? You have the cells arranged like that. Am I clear? So in the surrounding, you guys are having the cells arranged like this. So look at the diagram here, right? This is a C3 cycle, normal vascular bundles. Then you have bundle sheath cell, mesophyll cells. But here, look at these mesophyll cells, how are they arranged? And these are bundle sheath cells, right? Bundle sheath cells with large chloroplast, right? With large chloroplast, with C3 cycle. Why are they showing C3 cycle here, right? Because they have rubisco and lack pepcase, right? These cells lack rubisco and have pepcase. So pepcase is not getting affected with the oxygen concentration in high temperature. So this is the enzyme for C4 cycle. So it is phosphoenol pyruvate carboxylase oxygenase. Carboxylase oxygenase. That's what you have here. Oh, sorry. Phosphoenol pyruvate, my bad. Carboxylase only. Right? So this is for C4 cycle. This enzyme not getting affected with such changes. Am I clear? Right? Okay. Done? So let's discuss this process in detail. जैसे ये देखो. First of all, what is happening? Atmospheric CO2 will enter in the mesophyll cell. Again, I'm repeating. They have pepcase. So with what will happen? C4 cycle. No rubisco. How is it benefiting? Because it is the rubisco which is getting affected with the temperature and that CO2 concentration. Pepcase is having no role, right? So when this carbon dioxide, atmospheric carbon dioxide will enter, there will be the bicarbonate ions. Next, this is the phosphoenol pyruvate. So it's a 3 carbon containing compound. So in this case, you will see the primary acceptor is 3 carbon containing compound. Is that clear? So it will get fixed, beta. You will have the formation of the C4 acid here, right? We will see malic acid or aspartic acid, have patients will discuss it. So this will be transported, the C4, matlab 4 carbon containing acid will be transported to the bundle sheath cell. So now here, right, this C4 acid, it will be decarboxylated. So imagine here the CO2 is getting fixed. It is fixed in the form of that C4 acid. 3 carbon containing phosphoenol pyruvate is basically accepting this thing here, like it is the CO2 acceptor. That C4 acid is transported to the nearby cell through these plasmodisk metal connection. Here it is getting decarboxylated. And overall bundle sheath cells are impervious to gaseous exchange. So it doesn't matter. It doesn't matter outside whatever is the CO2 concentration. Rubisco is not getting harmed. Rubisco is having no issues. Are you getting my point? So Rubisco, what will happen? Decarboxylation will occur, CO2 will go, Calvin cycle will be done. And now the C3 acid, because one CO2 it has a release, C3 acid is there, it will be transported back and it will be regenerated. So when we have to regenerate it, then 2 ATP will be used for regeneration of phosphoenol pyruvate. And no doubt here in this process here, one NADPH is used, here is formed. So it is balanced out. So let me show you this diagram here once again, right? So you remember regeneration will cost 2 ATP each step. So in C3 cycle, so we already know for one cycle, right, 3 ATP, 2 NADPH are used. Done. In C3 cycle, one cycle में 3 ATP and 2 NADPH are used, right? And now, if there is C4 cycle as well, along with this, 2 extra ATP for that regeneration step. Right? So here, for one glucose, you will see total 18 ATP, 12 NADPH, 2 are used in the C3 cycle, plus here add 6 into 2, 12, 12 more ATP. So this is the drawback here. For C4 cycle, you need 30 ATPs, right? And 12 NADPH2. But ठीक है, extra ATPs which forms occasionally during cyclic phosphorylation, they can also be used. So let me show you this cycle. So first of all, there is the Krebs anatomy. Discuss that already with you all. Krebs is a German word for wreath, meaning to the ring-like arrangement of cells. So mesophyll में initial carbon dioxide fixation, bundle sheath cells will go for Calvin cycle. So bundle sheath cells में they are surrounded by mesophyll like a wreath. So chloroplast and C4 leaves are dimorphic. They are of two types. जैसे chloroplast of bundle sheath cell, it is larger, right? It is larger. It is rich in ribosco. Right, chloroplast of bundle sheath cells. It is larger in size, rich in ribosco, but lack phosphoenol pyruvate carboxylase, has starch grains, but lack grana. No grana, but starch grains are there in bundle sheath cells. Because mainly the CO2 fixation, where is it taking place? In bundle sheath cells, so it contains starch grain, but it doesn't have grana. Right? Why? Because that light reaction wala part is majorly in the mesophyll part. So normal type with grana but lack ribosco enzyme. So mesophyll cells perform C4 cycle, that preliminary fixation. Bundle sheath cell will go for C3 cycle. So their cooperation is also there between these two cycles. So it is the cooperative photosynthesis as well. You also call it as cooperative photosynthesis. Is that clear? Is that clear? Sure. So important C4 plants are sugarcane, maize, sorghum, digitaria, etc. etc. Got that? Done. So here you can see carbon dioxide accepted by phosphoenol pyruvate, 3 carbon containing compound, then oxaloacetate, then it will form malate, then pyruvate. So this is the first stable compound here, right? This is the first stable compound here. Told you already, right? This is the first stable product here, right? It is a 4 carbon containing compound, and then finally this one. So we have to regenerate it in order to continue the cycle. So CO2 plus PEP will give you OA, then that will form malate, again a 4 carbon containing compound. This malate will pass to the bundle sheath cell where its decarboxylation will occur and you will get pyruvate. Then that will enter in, CO2 will enter in Calvin cycle, glucose will form, and this pyruvate will go back to mesophyll. It will regenerate PEP using ATP. So 2 ATPs are used here. 2 ATPs are used here, right? So that is it. So either malic acid or aspartic acid can be there. So baki decarboxylation will also occur. So pyruvate plus ATP, this is the reaction. So let's see the energy requirement for one molecule of glucose. 18 ATP in C3 cycle, 12 will be for PP regeneration, right? So total 30. So that's the thing here. But in C4 cycle, what's better, right? Either they will have no, or I would say very less photorespiration, right? Less or even I would say negligible photorespiration in their case. Photorespiration in their case. Is that clear? Is that clear? Done. So key characteristics may no photorespiration, but though we see that very less photorespiration is there because there is a spatial separation. Light reaction occur in mesophyll cells. Important. Right? Dark reaction occur in bundle sheath cells. Another important point. They have higher productivity and yield. Matlab their high temperature and everything is there. So it is like that. So there is no photorespiration. So efficient CO2 uptake. So pepcase is having high affinity for carbon dioxide. Even when CO2 levels are low, they can withstand high temperature, high intensity. So it's a good thing for the dry tropical climates. For the plants living in dry tropical climates, they reduce photorespiration and transpiration and have better water utilization. Bundle sheath cells are impervious to gaseous exchange. That's why they have the high internal CO2 concentration, right? So storage of starch green is there in there, right? So this is the cycle. That's it. So you should know the examples, right? This is very important. Okay, so here you guys can see it very clearly, right? That ATP is formed. Pyruvate to phosphor, ATP is used rather, right? And oxaloacetate to malate, NADPH is used, but that also form when, you know, this malate will undergo decarboxylation. So it is just equal. Okay. Just have a look, guys. Quick. Quick. Done, beta? So let's move to the next part here. So their importance is already discussed, right? So next is, you can see that C3, C4 plants may, right? First product is 3-PGA, 3-carbon oxaloacetate. Primary CO2 acceptor, RUBP, 5-carbon, PEP, 3-carbon, right? Only Ribisco, Pepcase and Ribisco both are used. Site of Calvin cycle, mesophyll cells, but here it will be bundle sheath cell. Photorespiration high, absent, or sometimes if it is very low, though it is absent only. ATP per CO2, 3, there 5. Okay, CO2 fixation single, it will take place in 2 steps, mesophyll plus bundle sheath cells. Just wheat is a C3 plant, but maize is a C4 plant. Again, important. Done. Sure. Okay, so that is what you have to remember. So next is crassulacean acid metabolism. What is this? This is crassulacean acid metabolism. But this will be the process that you will see in the case of succulents. Okay? Right, you should know here. Here also, C3, C4 cycle both are there. But here there will be no Kranz anatomy. Here there is nothing which we have to discuss in detail, but the basics you should know. No Kranz anatomy here. C3, C4 cycles are there, but they are separated not by space, but C3 and C4 cycle are separated by time, right? C3 and C4, right? C3 and C4 cycles are separated by, they are separated by, not by space, but by time. Will occur at different times. Okay, so here you guys can see it was first observed in Kelencho. It is photosynthetic adaptation which is found in succulent plants which are living in hot and dry environment, right? So the main purpose is to minimize the water loss. Okay, so in dry condition, in arid condition, photosynthesis is continued, but the use of water is very good here. The loss of water is prevented. So cactus, Europensia, aloe, bryophyllum, pineapple, they show it. Okay, they show this cycle. So what is happening here? So because if the climate is very hot, so stomata cannot stay open during the day. Right, during the day it cannot stay open, so that can cause excessive transpiration or excessive water loss. We know it very well. So plants still need CO2 for the photosynthesis. So daytime, here daytime means stomata is closed. That's what you have to understand here, right? Done, beta. So CAM pathway mein stomata opens at night. So if it is opening, if it is opening at night, it will absorb carbon dioxide. It will keep them closed during the day to conserve water. Is that clear? So they will open stomata at night, right? So that it can absorb the carbon dioxide, because during daytime it is closed. So carbon dioxide fixation, that is C4-like, and the Calvin cycle occur in the same cell, but it is at the different types, that is night and day. Okay, so matlab daytime mein the CO2 will be fixed, all the processes will be done. Then nighttime when the stomata is closed, then that further cycle will continue. So you can see that nighttime mein carbon dioxide will get fixed into organic acid, C4 cycle. Stomata is open there, so pepcase will be used here. Daytime mein CO2 is released and fixed in that Calvin cycle, because stomata is closed, it will not go outside, it will be used in that cells. Same cell is having both there. So here the C3 C4 cycle is not separated by the space but by the time. So day and night thing will be there. Okay, so the last topic, second last rather, we have the factors also, right? We have the factors also. So what will you see here? So जैसे we have the photorespiration, photosynthetic carbon oxidation cycle, that is C2 cycle. This is what we have. Okay, this is what we have. So I told you it is because of the rubisco. So let's highlight the bullet points here. Or here you have to remember Chintu, Papa, Mummy, chloroplast, peroxisome, mitochondria. These are the three organelles which are used, and that is again a PYQ. Okay, that is again a PYQ. Got that? Done? Yes, please. So photorespiration is the uptake of oxygen, release of CO2. Matlab ultimately what is happening? Respiration mein you take oxygen, you release CO2, right? Photorespiration, so it is occurring in the presence of light, right? In the presence of light. So it mainly occurs due to the dual activity of rubisco, right? We know that its carboxylase activity can fix CO2, but its oxygenate activity will do oxygen fixation. Is that clear? So Chintu, Papa, Mummy, that's the trick. So these are the three organelles which are in which this cycle will operate, and that will be in a sequence. So that's important. That's a PYQ. Okay, that's a PYQ. It is important. So here in chloroplast, your RUBP reacts with oxygen. So now here you will not get two molecules of 3-PGA. Instead, you will have one molecule of, this point is important, rest steps are extra, but this point is important. You will have one 3-PGA, right? And one 2-phosphoglycolate, which is a 2-carbon containing compound. 2-phosphoglycolate it is, right? So this phosphoglycolate dephosphorylated to glycolate, which will in peroxisome oxidize by oxygen. Then here the glyoxylate cycle will be there. So I'm not expecting you to remember this, but yeah, this is important. So glyoxylate will form glycine with the help of enzyme glycoxylate oxidase. Then 2 molecules of glycine, they will form serine. CO2 will be released. Ammonia will also be there, and NADP will be reduced to NADH here because it is respiration. So NADH will be there. So serine will convert it to hydroxypyruvate, glycerate will return to chloroplast. So definitely this detail is not required for you, but yeah, what is required? Organelles, peroxisome, chloroplast, mitochondria, and what exactly is forming there, right? What exactly is forming there? That's important to remember. Done? Done? Sure? So why photorespiration is considered wasteful? Because no ATP or NADPH is produced. CO2 is released, consume ATP to convert intermediate, reduce the photosynthetic efficiency. So that's why we say it's a wasteful process, okay? So high temperature, high oxygen concentration, low CO2 concentration, and bright light causes that. These are the conditions which favor the photorespiration. Understood? Understood? So there is high temperature, there is high oxygen concentration, there is low carbon dioxide concentration, there is bright light. So it prevents the photooxidative damage by dissipating excess light energy, maintain balance in redox reaction. Now the last topic is factors which are affecting photosynthesis, and then we will move to the next chapter, that is respiration, but yeah, after a short break, right? After a short break. Okay. So first of all, law of limiting factors. First of all, we have the law of limiting factors here given by Blackman. So when a process is conditioned so as to its rapidity by number of factors, the rate of process is limited by pace of slowest factor. Matlab, if in a process you have many factors, right, more than one factor, the one which is the slowest one, yeah, which is in less amount, that will decide the rate. Let's suppose if a process is contributed, right? If a process is due to, I would say, many factors, like let's suppose 5 factors are there. Out of that, one is in its lowest concentration, slowest pace. So if I will increase its concentration, then obviously the rate will increase. That's what Blackman's law of limiting factor states. That so here, how can we apply it on photosynthesis? We know photosynthesis depends on multiple factors. So if one is limiting, the rate will decrease. If CO2 is limited, even if light increase, photosynthesis won't increase because it depends on 5 factors. So if out of that one is getting decreased, it doesn't matter that I'll increase the another one. It depends on that 5 factors now. So this one which is in less concentration is deciding the rate, right? So firstly, light. Only 2% of solar light reaching Earth is used in photosynthesis. We know that. So in case of light, its intensity, quality, duration, all of it affect the rate. Right? The intensity of the light, the quality of the light, the duration of the light, everything is going to affect that. So when I'm saying light intensity, right? When I'm saying light intensity, so it is directly proportional to the photosynthesis up to a certain limit. Its intensity, matlab with beyond a saturation point, rate no longer increase. So I'm talking about the intensity. We will also talk about the duration here. Jaise shade plants, skyophyte, they are low light tolerant. Sun plants, heliophyte, bright light. So accordingly, light intensity will affect. Quality mein beta, obviously if red light will be less, then photosynthesis rate will be affected. So if there is red light more, moderate blue, minimum in green, then obviously light quality is better. So longer photo period, more photosynthesis. Hai na? We know that plants, they need different periods of light as well. So there is one more thing, that is the compensation point, that at which CO2 uptake is equals to CO2 release. So no photosynthesis will occur. So yes, you should remember the compensation point, where what is happening, the uptake, carbon dioxide uptake and carbon dioxide release is same. So light saturation point is also there. So intensity at which maximum photosynthesis occurs. So here you guys can see this graph as well. You should know beta what is on x-axis, that is light intensity, what is on y-axis, rate of photosynthesis. So what these A, B, C, D, E are showing, this is not given in NCERT. You can see A is that part of curve where the light is actually a limiting factor. But when it reaches at B, it is only 50% limiting. Now it means some other factors is the limiting one, right? Yes. So B pe it is 50%, C pe it's like other factor is limiting because light is optimum. Down D represent light intensity at which the rate is maximum. So it is the point where your light intensity, it is the light intensity at which your rate is maximum, and E will represent that the rate of photosynthesis under given conditions. So do highlight it quick. Do highlight it quick. Do highlight it, guys. And write it in the NCERT. Quick. Done? Sure. So next is carbon dioxide concentration, from where you can get the question. So when it is the CO2 is the major limiting factor, right? So concentration of CO2 beta, it is very low in the atmosphere, somewhere around 0.03 to 0.04%. So it increase in it up to, right, increase in it up to 0.05% can cause, its concentration is 0.03. If it reaches to 0.05, yes, the carbon dioxide fixation rate will increase. So beyond this level, they can be damaging, right? So we don't need it in too much quantity as well. And you know that it's a greenhouse gas also. So C3 and C4 plant, they respond differently. We know that when the light condition is low, neither group respond to high CO2 condition. At low light condition, when light is low, right, both of them cannot respond to high CO2 condition. But when the light intensity is high, both are responding differently. Just say for an example, both C3 and C4 plants, right, they show increase in the rate of photosynthesis. We know that. But what is important to understand here is that C4 plants, beta, they show saturation at about 360 micro per liter, while C3 respond to increased CO2 concentration and saturation is showing beyond this level. So these values, they are important. Question can come from this part, right? This is important. Now the next is, does current availability of CO2 level is limiting to the C3 plants only? है ना? So the fact that C3 plant respond to higher CO2 concentration, when you increase the CO2 concentration, C3 plants will respond to that. How? By showing increased rate of photosynthesis leading to higher productivity. So the productivity will also be higher here, right? Because the CO2 concentration increasing. So that is why in the greenhouses, right, we can grow some crops because there we are maintaining this level. So C3 plant, they respond to high CO2 concentration by showing increased rate of photosynthesis leading to higher productivity. Okay, so it has been used for some greenhouse crops such as tomatoes and bell pepper, right? Such as tomatoes and bell peppers. So bell pepper, so this example is also important. So they can grow in CO2 enriched atmosphere and the yield will be maximum. So question can come from this part, guys. So Baki, you know that dark reaction, it is enzymatic, hence temperature controlled. Light reaction is temperature sensitive, but less affected compared to dark reaction. Baki C4 plant, they respond better to higher temperature and higher rate of photosynthesis. They have lower temperature optimum and their rate of photosynthesis decline at high temperature. So temperature, you know that for tropical plants, high temperature is optimum. For temperate, low. So it varies from plant to plant. And then this is the optimum range, 20 to 35 degree generally, right? Generally. So C3 plants ke liye 10 to 25, C4, they can work efficient, they have the adaptations for high temperature as well. Is that clear? Is that clear? So do remember this thing, beta. Do remember this thing that from minimum to optimum, rate of photosynthesis doubles for every 10 degree Celsius rise. So from minimum to optimum, so rate of photosynthesis will get double. How? For every 10 degree increase, right, beta? So beyond optimum temperature, rate of photosynthesis declines. Fine, rate of photosynthesis declines. Am I clear? So next factor is of water, right? So though water is one of the reactants in the light reaction, the effect of water as a factor is more that it is overall affecting the plant. Just a plant, if it is under water stress, you know that transpiration will slow down. So that is how water is affecting, right? So it is not directly affecting on photosynthesis, but its stress can cause stomata to close. So if stomata is closed, carbon dioxide availability will also be closed. So in addition to that, water stress also make leaves wilt. So reducing the surface area of the leaves and their metabolic activity as well. So oxygen, high O2 concentration also decreases photosynthesis, which is Barber effect. So more oxygen, less photosynthesis. Pollutants can also affect the photosynthesis. जैसे बेटा peroxyl acetyl nitrate, chlorophenyl dimethylurea. So this is the Hill reaction inhibitor. So this is all about the photosynthesis. Baki beta internal factors mein structure of leaves, accumulation of end products and genetic predispositions are also there. Okay. Okay. So number and size of chloroplast, thickness of cuticle, orientation and age of leaves, internal carbon dioxide concentration, they all affect photosynthesis. And accumulation of end product will also reduce because the end product is there, then obviously the rate will come down. So that is it. This is all about the photosynthesis. Next is the respiration, which we have also discussed in one of my marathons in detail, right? And it is not even lengthy. Okay, so today we will discuss it up to this part only, right? And respiration ke liye I'll pin the link in the comment section, in the description box, and even in the comment section I will pin the link for the respiration. So be sincere, guys, because you are taking this biology part as a joke that it will be doable, but trust me, this time it will not be, right? You will face this thing when in your final exam. So keep practicing, keep learning, and do not miss the conceptual chapters. Okay. So thank you so much, guys. This is all about today's lecture. Bye bye.